Uh, Madam, Fips organizer. I could open with the same one, madam. ನಾನು ಓಪನ್ ಮಾಡಿದೆ ಈಗ. Madam, same link ಏನೇ. I just opened it. Can you see that now if you open it, the organizer, uh, hello? ಮೇಡಮ್ ಒಂದು ಸೆಕೆಂಡ್ ಅವ್ರು ಕಾಲ್ ಮಾಡಿಬಿಟ್ಟು ನಿಮ್ಗೆ ಲಿಂಕ್ ಮಾಡ್ತೀನಿ ಮೇಡಮ್ ಒಂದೇ ನಿಮಿಷ. Madam, good morning. So, our director Pratima Madam is trying to, log in. Her video is there. There is no audio, it seems.
Now I can hear you, Dr. Rao. Now it's fine.
You can hear, Madam? You can hear,
Yes,
Madam?
now I can hear you. I'm going to
Okay.
put on my video.
Yeah, okay.
Yeah.
Yeah, yes. Yeah.
I'm sitting in the middle of the highway, so I don't know how it's going to work, but we shall try.
Yeah, sure, Madam. Sure, Madam. Okay.
Thank you.
Yeah. Thank you, Madam. Thank you.
So thank you, ma'am. Dr. Pratima Murthy, ma'am. I'm Shashi from Director Naipar, Hyderabad. Thanks for joining us and agreeing
Hi,
to
Dr.
deliver
Shashi.
a talk.
Meet you online.
Yeah, and I have heard a lot about you. I sometimes
Thank you.
see you in print media. So welcome. And I think there are another 3 minutes, then we can start. Huh? Yeah. Ma'am, your voice, I think you are mute.
Sure. Can you hear me now?
And now it is fine. Yeah.
Hello?
Yes, ma'am, I can hear you.
Yeah, we can hear you, Madam.
Yeah. We also have another chairperson. We have 2 chairpersons. One is Professor Rao, other is Professor Srinivas. He is Dean ESI College and Hospital in Hyderabad. He is there with us. And Namaskar, Professor Srinivas. Namaskar.
Thank you, ma'am. You connected me to Murthy madam after a long time.
Achha, so you know Murthy ma'am. I think
Way back in 1980, 88, 87. After that, I have not seen her. I've been only hearing about her.
Okay. Sir, if you are alone in the room, you may remove your mask. If you, you know better than me.
Oh, I am in the hospital.
Yeah, okay.
If I don't follow, my fellows will not follow me.
Okay, fine. Yeah. So, ma'am is there, na?
Yes, ma'am. shall we begin with the session with your permission?
Ma'am, I can't see her. Is she there? Dr. Rao, ma'am is there now?
Madam, can you hear us, madam?
But I can't see her, Dr. Pratima.
Pratima madam, she was in.
Yeah, where is she now? Is she there?
She is not there. Okay.
Yeah.
Of course, her login is also my name only is there actually.
Oh, she's there, yes.
Yeah,
So,
she's
ma'am, we can start with your permission, right? Dr. Pratima and Dr. Srinivas.
Yes, please.
Yeah. So, I will request my colleague to start. Please.
To all the participants, respected speakers, and the chairpersons. Today marks another day to be filled with excellent discussions covering new updates of various scientific challenges. We warmly welcome you all back to day 3 of the 9th International Conference of Federation of Indian Physiological Societies 2022, organized by NIPER Hyderabad in association with PSI, SAPI, APPI, and ISCAP. Without taking much of your precious time, we shall now begin with our first session for the day, that is keynote lecture, to be delivered by Dr. Pratima Murthy ma'am and to be chaired by Dr. B.S. Shankar Narayan sir and Professor Srinivas sir. So I request the respected chairpersons to kindly take over.
Professor Srinivas. So,
Namaskara, sir. Namaskara.
Namaskara. Okay. So, ತಾವು introduce ಮಾಡ್ಬಿಡ್ತೀರಾ ಮೇಡಮ್ ಅವ್ರಿಗೆ?
ನೀವು ಮಾಡಿದ್ರೆ ಚೆನ್ನಾಗಿರುತ್ತೆ.
Okay, no problem, no problem. So, very good morning and welcome to the Federation of Indian Physiological Societies Congress. And so we have a great pleasure in inviting and also introducing Professor Pratima Murthy. She is a very distinguished psychiatrist and so she established Center for Addiction Medicine and also various her studies and policies which translated into the addiction medicine regimes and also the better management all over the world. So she is currently heading the NIMANS and as a director. And also she is serving in various organizations in terms of developing the policies for the better management of addiction and various kinds of addiction policies, including tobacco quit line. She is chairing. She also coordinates with various activities of the WHO, and it's a great pleasure to listen to her. And thank you very much for accepting our invitation, madam, and to deliver the keynote address. And so we request on behalf of the Federation of Indian Physiological Societies, and once again we invite you, and over to you, madam.
Thank you very much. I'm just going to share my screen. I hope you can all see my screen.
Yes, madam.
Dr. Shashi, the chairs of this session, Dr. Rao, thank you very much for inviting me to this very prestigious keynote. And I'm sure the last day has gone very well. And what I'm going to talk to you about this morning is the work that has been carried out by my colleagues on neuromodulation at NIMHANS. And I thought this was an interesting topic because, again, for physiologists working in this area, there is a lot that needs to be learned. So I'm going to talk about where we've reached with neuromodulation and how we proceed from here. I'd like to acknowledge colleagues at NIMHANS who've been working on this area for more than a decade now. Dr. Venkat Subramaniam and his team, Dr. Pramod Pal and his team. Venkat works in psychiatry, Pramod works in neurology. We also have Shyam Sundar, who's working on deep brain stimulation. So it's really the work that some of my colleagues have done, which I'm going to talk about and present in the context of reviewing the literature around neuromodulation. So I'm going to talk about how neuromodulation has been effective as a treatment. And I'm going to focus predominantly on electric and magnetic stimulation. We'll talk about how long it's been around, the principles of the various kinds of neuromodulation, the emerging uses, the mechanisms of how neuromodulation seems to work, what opportunities we have in front of us, and the future of neuromodulation. In terms of the early milestones, when we peek into ancient history, we find that this is not new. The concept of electricity as a cure has been in existence for several centuries, and discoveries in medicine have contributed to the use of convulsive therapies. For example, we know malaria therapy for neurosyphilis, for example, was one of the breakthroughs which won Wagner Yorek the Nobel Prize. I'll talk about insulin shock therapy, which was also a way of inducing convulsions, it was thought. Of course, this is the All India Institute of Mental Health and the mental hospital where electroconvulsive therapy had its beginnings. And then we'll go on to the more modern forms of neuromodulation. So as early as AD 47, we know that electric eels were used to cure headaches of Roman emperors. So it was around, using electric treatments, very well known from a very long time. There's a very nice essay on electricity and its usefulness in science. And several people, this was by George Adams, but ever since the discovery of electricity, we've looked at the application of electrical methods into treating various kinds of peripheral disorders of the nervous system. This is what is called a patented magneto electrical treatment. And this electrical apparatus actually delivered electrotherapy to treat various kinds of physical and nervous disorders. It's possibly one of the oldest alternative current apparatus that was used. And this is in our museum. This particular piece is in our museum. And this dates back to the in the 1800s, where this was available and was very popular as a form of treatment. We know that various chemicals were used to cause convulsions and to cure: camphor, cardiazol, as well as ammonium chloride. But one of the breakthroughs was insulin coma therapy, which initially involved inducing coma as well as convulsions as necessary for treatment. Later, convulsions were considered as an unwanted and dangerous side effect. And of course, from our own center, Professor M.V. Govindswamy, who was the first director of the All India Institute of Mental Health, actually wrote papers in the Lancet on insulin shock and its relevance in the tropics. And of course, in tropical climates, there are various important, precautions that need to be taken when such interventions are delivered. And of course, cardiazol and ammonium chloride as proconvulsants actually were associated with a lot of side effects. And therefore, we were constantly looking out for something that was safer for patients. And then came electroshock therapy, Serletti and Binney. There's a very, very interesting history to this, how he watched pigs being convulsed, and that became the emergence of the electroshock therapy. And we know that electric convulsive therapy, in spite of the negative kind of publicity it had in terms of one flew over the cuckoo's nest, is a very, very important treatment to date, which is extremely useful. And it's a time-tested neuromodulation treatment. We've had several administration manuals that our institute has derived. Professor Gangadhar and Professor Janki Ramaya were some of the leads at NIMHANS who developed these treatments. It has established efficacy in severe depression, mania, suicidal states, as well as catatonia. And we also use it in treatment-resistant schizophrenia. ECTs can cause prolonged seizures, prolonged apnea. We know about post-tilt confusion that can occur with ECTs, as well as, of course, acute respiratory syndromes. So we've constantly been looking for neuromodulatory techniques, which are likely to be safer and well tolerated by patients. And that's where the first neuromodulation technique I'm going to talk about comes in, which is direct transcranial magnetic stimulation. And this has its roots from, again, the oldest treatments which were developed by Aldini and all that in terms of electrical stimulation. And you have fairly compact machines which deliver TDCS presently. So when you look at transcranial direct current stimulation, it's a non-invasive, safe technique. that involves the application of low intensity direct current using bioconducting electrodes placed on the scalp. And that leads to polarity specific neuromodulation as well as neuroplasticity, which is the underlying principle of the use of TDCS. We have a very strong neuromodulation program at NIMHANS, and my colleagues have looking at TDCS as an add-on in several disorders: schizophrenia, obsessive compulsive disorder, craving and alcohol use disorders, depression, as well as in other conditions as well, conditions like dementia and so on. We've had experience with child and adolescent populations, adult as well as geriatric populations. It's also been successfully tried in pregnancy, where psychotropic medication may be contraindicated or have significant side effects. One of the very significant places where TDCS has been tried is when there are refractory auditory hallucinations in schizophrenia. And of course, various changes or adaptations in terms of targeted intermittent booster TDCS sessions have occurred, and colleagues have had experience of more than 3,000 sessions without any significant adverse effects. So TDCS in schizophrenia looks at addressing both the negative symptoms by, for example, where there is defective activation of the prefrontal cortex and decrease in acetyl aspartate levels, as well as when looking out for positive symptoms like delusions and hallucinations, which focus on the temporoparietal junction hyperactivity with increased glutamatergic function. So both in positive as well as in negative symptoms, there has been some significant contribution of TDCS as an add-on treatment. And of course, the important thing is that you can do an MRI-guided personalized neurotargeting with the high-density TDCS. So you can actually decide which particular area to stimulate. And of course, the target site, how do you position the device, the orientation, etc., depending upon gyral anatomy, depending on the electric field and the direction of the current flow, various kinds of adaptations can be made in the TDCS. In terms of the neurobiological effects of TDCS, you'll see, and again, as physiologists, you would be interested in the fact that there are ionic shifts in transmembrane protein changes that result in changes in cortical excitability, and that's what TDCS seems to do. Of course, it appears to have effects on the GABAergic and the glutamatergic transmission, and anodal TDCS inhibits GABAergic transmission, whereas cathodic TDCS inhibits glutamatergic transmission at the receptor site. And in terms of BDNF signaling, there are changes that TDCS causes in terms of regulating BDNF through various trafficking signals, as well as local translation and the release of BDNF, which appears to underlie some of the beneficial effects of TDCS. It appears that TDCS has multiple multi-level neurobiological mechanisms of action, including glial, because there is glial involvement, all these various neurophysiological as well as neurochemical changes appear to underlie the impact of TDCS. Now, TDCS clearly has several factors which can affect its effectiveness, depending upon the background of the patient, the gender of the patient, the illness status of the patient, the attentional state and arousal effect of the patient, various physiological states, whether the person is in clear consciousness or undergoing anesthesia, various intrinsic metabolic factors. We don't know how that would affect the patient's response to TDCS. We also know that the stimulation intensity, the size of the coil, the shape of the coil, the montage of the coil, various genetic polymorphisms that may underlie this the patient condition, as well as individual cortical geometries, can all affect the effectiveness of the TDCS. I'll move on to TMS, transcranial magnetic stimulation. And again, it's got almost 4 decades of experience where you looked at activating muscles, the specific motor areas of the brain, but initial ones also caused pain in the process. And therefore, in 1985, Barker demonstrated the ability to stimulate the brain with magnetic stimulation, which could be accomplished with little or no pain. A brief mechanism, look into the mechanism, suggests that there is a high current pulse that is produced by a magnetic coil, and that is placed tangential to the scalp, and the magnetic field can reach up to about 2 Tesla and lasting for about 100 milliseconds. And when you look at the mechanism of TMS, an electrical field is induced perpendicularly to the magnetic field. The voltage of the field appears to excite neurons and induce local currents. And in a homogeneous medium, the spatial change of the electric flow causes the flow in parallel to the plane of the coil, which is predominantly tangential to the brain, and the loops within this with the strongest current will be near the circumference of the coil itself. So therefore, a lot of local changes can be produced. This is the TMS machine used in our neurology, particularly for Parkinson's disease. So recurrent transcranial magnetic stimulation has been used in depression, in schizophrenia, obsessive compulsive disorder, post-traumatic stress disorder, and nicotine dependence in psychiatry. I'll talk about the neurological conditions in a bit, but several psychiatric conditions appear to get benefit from the use of recurrent TMS disorders. When you talk about neurological disorders, apart from Parkinson's disease, stroke, cognition, in spinal cord lesions, in spasticity, in migraine, in various kinds of chronic pain conditions, and in partial epilepsy, you'll see that it can be used both as a diagnostic aid as well as a therapeutic intervention. And therefore, both across psychiatric disorders and neurological disorders, TMS appears to have wide applications. When you look at any of these, you will find, as I mentioned earlier, that there are lots of things that can influence it, from genetics to the person's anatomy and physiology to environmental influences. There's a lot that occurs in terms of biological factors as well as measurement factors, which, accompanied by neuroimaging, looking at various physiological measures, both in stress and in active conditions, as well as the stimulation protocols, can all influence the non-invasive transcranial brain stimulation. There are 3 primary components that have been used in neurology. I'll go to deep brain stimulation, which is an invasive intervention in a bit, but basically the stimulating electrode, the internalized pulse generator, which has a battery pack, and the electrode extender, which are all parts of the transcranial magnetic stimulation and other interventions. So there can be different kinds of stimulation in neurology, from transcranial magnetic stimulation to specific stimulation of the motor cortex and the deep brain stimulation, both of which are invasive. You can actually also stimulate the spinal cord as well as the vagus nerve, which are much more local in terms of their application. I'll just talk about the deep brain stimulation in a little while. So for deep brain stimulation, you'll see that the insertion of the electrodes will have to be done under anesthesia. You can target it with image guidance with a stereotactic frame. And of course, imaging is done to confirm the electrode position. And then you have stimulation. You can do an intraoperative stimulation. I'm sure many of you have seen actually the surgeons demonstrating what actually happens on the table, where a person's the experience of obsessions might actually change, the mood might change, the movement disorder might ameliorate. The same kind of things were also earlier demonstrated by Dr. R.M. Verma with chemothalamotomy. So now you have much more focal electrical stimulation to produce the same results. And then you place electrodes with multiple contacts. So then you have an implanted pulse generator, which is battery run. And of course, the battery lasts for anywhere between 2 to 5 years, and it has to be recharged periodically. A programming will have to be done with a handheld device after the insertion, which involves active contacts with the patient, the polarity, which can either be monopolar or bipolar. There are settings of amplitude and frequency and pulse duration that need to be established. Of course, tolerability, acute effects also are important parameters to monitor. And sometimes it takes a long time for the patient to get comfortable with the DBS settings, so that the patient experiences the benefits without having the side effects. So the mechanism of DBS is a functional lesion of the surrounding tissue, neuronal activation, the depolarization blockade of current-dependent ion channels, changes in the neurotransmitters, synaptic inhibition, and there are also is a disruption of the dysfunctional oscillations in the pathological circuits, leading to benefits. Now, what is the future of neuromodulation? What people anticipate or kind of foretell for the future is that there will be an integration of adaptive network neuromodulation with predictive artificial intelligence, automatically adjusted by the brain as well as with external sensors and controlled via cloud-based applications. So the main key terms here are integration of the adaptive network, using artificial intelligence, the potential for the brain itself to make adjustments, as well as external sensors, and of course, using the cloud-based applications. So you'll see here that typical bioelectronic applications are going to extend far beyond just the central nervous system. Of course, I mean, the cardiac monitors have been, the cardiac pacemakers have been the forerunners of the electric stimulation of the brain. But then you also see that there can be visual prosthesis for blindness, cochlear implants for deafness, epilepsy stimulators to abort epileptic attacks, vagal nerve stimulators for epilepsy and depression. We've also just carried out a vagal nerve stimulation project for a DM dissertation on reducing craving in alcohol use disorders. It's also been tried in opioid use disorders. And then, of course, deep brain stimulators in Parkinson's disease, dystonia, tremors, obsessive compulsive disorder, and epilepsy. And local hypoglossal stimulators for sleep apnea, sacral nerve implants for incontinence, spinal cord implants for chronic pain, tibial nerve stimulators again for incontinence, and of course, focal problems like foot drop using a peroneal nerve stimulator. So there is a lot of scope for using these kind of neuromodulatory techniques, both centrally affected, influencing the central nervous system, or indeed for focal peripheral nerves as well to ameliorate certain conditions. I'm going to leave you with this very, very evocative slide. On the left side is actually a country in South Asia where people lay down on the electric railway tracks. This was carried in a leading magazine, and they felt much better after they laid down on these railway tracks because the low-grade electrical current appeared to cure them of several chronic diseases like chronic pain, headaches, and so on. But obviously not a very safe thing to do, in fact, quite a dangerous thing to do. And therefore, we've known from time immemorial that electrical treatments have great benefits for various kinds of disorders. And now we possibly will have ambulatory neuromodulatory techniques that people can go home and apply to themselves once they've learned to do this. My colleagues are trying to try these ambulatory forms for people with chronic hallucinations so that they learn to use it themselves, their family members learn to use it themselves. Again, in alcohol use disorders, in movement disorders, there's a lot of potential of neuromodulatory techniques. And I'm sure as physiologists in this forum, there will be a lot to study in terms of the underlying mechanisms. So it's a very, very exciting field. And that's why I thought I should talk about neuromodulation in neuropsychiatric disorders. Thank you very much for giving me this opportunity. And thank you again.
So thank you very much, madam. And so the neural plasticity associated with neuromodulation, and so you explicitly and very in detail you have provided an evidence that three different modalities of the stimulus which we use, either TDCS or RTMS or the deep brain stimulation, both in neurological conditions and also in the psychiatric disorders, where the neural plasticity induced by them can have a significant impact on the restoring most of the symptoms associated with it. So brain stimulation seems to be the key factor in controlling various neurological and psychiatric conditions, and also there is a lot of technological development is the key component. As madam mentioned, the artificial intelligence and also having a non-invasive kind of approaches for the stimulation of the various brain regions by inducing the neural plasticity, they are the key things, and the NIMHANS is known to work in that division, in that area. And thanks to Dr. Shashibala Singh also, she is also associated with neural plasticity work, and she's been associated with NIMHANS for more than 2 decades. And so we are working together, madam. As you told you, as you said, that the physiologists, they have a lot more job to do in terms of understanding these mechanisms in great detail. So I request Dr. Professor Srinivas, my co-chairperson, and also share his views, and also if there are any questions in the chat, we will address to you, madam. Professor Srinivas, please.
Thank you, sir. Very enlightened, and I'm very happy personally that I could connect to the people after decades of attachment with Bangalore. I just go with a request to madam that please visit Hyderabad once. Gangadhar sir visited last year. We were very happy to see him after a long time. And it's time that being a new director of the NIMANS, and we would like to see you here, and then maybe some collaborative research between the institutions of ESIC Medical College, NIPER, and NIMANS, ma'am. I guess we can go to the attendees, please. Thank you.
There are questions in the chat box. I think Dr. Rao, you can take.
Okay.
There is one question by Dr. Dharmender.
Yeah, Dr. Dharmender, what is the success rate of DBS in a PD post DBS, madam? So this is in the PD patients? Yeah.
Yeah. So my colleagues have obviously found it extremely effective, as I said, specifically for movement disorders, and in fact, the earliest with chemothalatomy, again, what Dr. R.M. Verma started off as focal things have been, very, very effective with deep brain stimulation. I am not very sure about the exact remission rates in terms of Parkinson's disease, but what we know is that there is a lot of temporary remission that occurs, and possibly you need to keep readjusting the parameters every 6 months or so to make sure that the response sustains. While it is very effective, the major problem with DBS is the cost that is associated. And that's why, it is rather difficult to actually carry out large-scale studies presently because of the cost that is associated and, of course, the complexity of DBS. But it's a very, very promising intervention for Parkinson's disease.
Shashi madam, so you have any further comments?
And thank you for a very enlightening and absorbing presentation. Nowadays, because of this corona post-pandemic, the mental issues are becoming more and more, and NIMHANS being one of the premium institute of India for mental health. I would like your views. How do we sort of tackle this? Because government is also giving a lot of push, and we say rather than physical, the mental issues are going to be much more, and there are a lot of things which are coming even in the print media. How do we sort of train or condition not only ourselves but even our own children and even the aging population, because this is creating a lot of mental imbalances at various places.
Yeah, thanks, madam, for that question. And I think we're going to look for the simple mechanisms of neuromodulation for such patients, for people who are under stress, who are under depression, who are under anxiety. We know, for example, the importance of, exercise and neuroplasticity. Both physical activity, physical therapies, as well as exercise have a lot of role in terms of increasing energy expenditure and can involve various simple tasks. Exercise again can help in these areas. So I think we're going to look at a lot of these aspects of, call them neurostimulation if you will, but various, simple ways of helping people to relax, to meditate, to exercise, to be able to look after their mental health. And, and this is going to be something that we need to teach people from a very, very young age. I think something that COVID has taught us is that we've forgotten. I mean, children have lost the opportunities to go out, to play, to exercise. And therefore, I think we need to revisit these important aspects of physical activity and exercise in terms of neuromodulation, particularly in the context of COVID, and the whole lot in terms of things like relaxation, meditation, mindfulness. Many of those things also have significant neuromodulatory effects. So while we've been only talking about the technical gadget-driven neuromodulation, I think we also need to focus on the simple aspects of neuromodulation, its effect on the opioidergic system, the GABA and the glutamatergic system, the dopaminergic system. So I think, right, we have a continuum of interventions that are going to be possible and relevant to population in the large scale.
Thank you, ma'am. Thank you, ma'am. Ma'am, I would also have a same request. Please visit NIPER when you are visiting ESI.
I'd be delighted to learn from.
Srinivas also wants, and we also want your presence in NIPER.
Thank you. Now, I think there's a lot I need to learn. I'm a novice in this field, and except for some work in the area of addiction, it's all the colleagues who have done amazing work. So it'll be lovely to come as part of a team to both your organization.
Sure. Please do.
Thank you. Yeah. Thank you very much.
Thank you, ma'am.
Professor Srinivas?
There's a question by Dr. Amrita Kulkarni. Can DBS be soul? I just got it. Yeah.
Yes, soul therapy for.
Yeah. Neurodegenerative diseases. Yes, Madam Villa.
Dr. Shashibala, you might be able to answer this as well. But I don't think right now we have any of the neuromodulatory techniques as sole treatments, except, of course, I mean, many of the treatments have been complementary, but perhaps people are looking at it in terms of not only cognitive interventions, but also in terms of consciousness interventions. So I think the jury is still out in terms of its usefulness, but there appears to be potential, and I would think that it's still at the exploratory phase rather than a very established intervention for neurodegenerative disorders.
Yeah, I think speaker already said, ma'am told also that it's an expensive therapy. Economically, it may not be available to everyone. It is not the sole therapy because non-pharmacological interventions, mindfulness, their exercise, yoga, and there are many alternative therapy medicine, which can also which can complement this system.
So Professor G.K. Paul, he has some comments from JIPMER.
No, it was a very, very interesting topic. I was really listening to Madam. Actually, simultaneously, one more program was going on. So I missed the initial part, but it is a wonderful, topic presented by Madam. I really appreciate the work you are doing at NIMHANS. We are doing also at JIPMER, and yoga is very helpful. And Madam knows actually. And it was a wonderful topic. Thank you, Madam, for such a brilliant presentation.
So on behalf of the Federation of Indian Physiological Societies, on behalf of my co-chair, Professor Srinivas, and also the NIPER director, Dr. Shashibala Singh, she is the organizing chair, and the entire team of the Federation of Indian Physiological Societies Congress, we take this opportunity to thank Professor Pratima Murthy for enlightening us with the neuromodulation and also the neural plasticity associated with it, and its role in the better management of neurodegenerative diseases and also the psychiatric diseases. In fact, we are very thankful to you, Madam. Madam is traveling and she stopped in the middle, and so she delivered this talk. And I am sure that, so as requested by Professor Srinivas and Dr. Shashibala Singh, and Madam will be, whenever she is visiting the Hyderabad, she will certainly visit both these organizations as well. So on behalf of the organizing team, and I take this opportunity to thank profusely Professor Pratima Murthy for delivering the keynote address. Thank you very much, Madam.
Thank you very much.
Thank you.
More to the organizers. Yeah.
Thank you so much, Professor Shankar Narayan Rao sir and Professor Srinivas. We would now begin our next symposium, 3D Bioprinting, Future of Animal-Free Research. I would like to call upon Dr. Falguni Pati and Dr. Shanti Moikar to carry forward the symposium.
Thank you very much for handing over the session to us. And myself, Shanti Moikar, and along with me, Professor Falguni Pati from IIT Hyderabad, will be chairing this session. So I would like to take this privilege to introduce Professor Falguni Pati, and who is currently the Associate Professor in Biomedical Engineering Department at IIT Hyderabad. Before that, he completed his PhD from IIT Kharagpur, and thereafter he had postdoc from postdoc South Korea and KTH Sweden. And then he moved back to IIT Hyderabad in 2015, and there he started his independent academic career and established the lab biofabrication and tissue engineering, particularly focusing tissue engineered products for in vitro tissue and organ models using 3D bioprinting technology. And his research interest involves developing new bioinks, bioprinting methods, protocols for productions of tissue and analogous system on demand, and thereafter customize the tissue or organ model. He has designed and developed a new course on biofabrication for the PG program at IIT Hyderabad, which is one of its first kind in India. His research was supported by different prestigious funding agencies, including MHRD, DST, DBT Biotechnology, and he was also the recipient of prestigious Ramalingaswamy Fellowship in 2016. So with this brief introduction, I would like to hand over the session to Professor Pati and also request to enlighten us about the importance of 3D bioprinting. Then we'll go into the session.
Yeah, thank you, Dr. Kaur. Am I audible?
Audible, sir.
Okay, thank you. Thank you very much for the nice introduction. And I'm privileged to be here today with all of you on this topic that is very much important, like the 3D bioprinting. 3D bioprinting has got huge prospect for generating artificial tissues and organs, because over the last 30-40 years, like you see that tissue engineering domain, the researchers, they have been trying very hard to develop artificial tissues and organs, but there are not much success in that domain. So with the advent of this technique called 3D printing in biomedical area, there is a huge prospect, and we have now, there are some success stories where people have developed various tissues, artificial tissues and organs, starting from skin, bone, trachea, and others for different applications, like either for tissue engineering, for implantation, or for also to be used as an in vitro models. So I'm excited today here because we have some outstanding speakers lined up for these sessions. And without further delay, I hope that you are all waiting for the same. Without further delay, I would like to introduce the speakers. So our very first speaker is Dr. Pallab Datta from Naipur, Kolkata, India. He's a very good friend of me. Now, so I short intro, so a very brief bio about Dr. Pallab Datta. So he completed his B.Pharm from University of Pune, and then also MS in pharmaceutics from Naipur Ahmedabad, now Naipur Mohali in 2007. And then he moved to IIT Kharagpur for his PhD. He completed his PhD from IIT Kharagpur School of Medical Science and Technology. Here, so presently he is an assistant professor of pharmaceutics at the Naipur, Kolkata, West Bengal. He has previously served in the Center for Healthcare Science and Technology, Indian Institute of Engineering Science and Technology, Shivpur, Howrah, years, where he established the bioengineering program, biomedical engineering program. His areas of interest are biomaterials, bioprinting, and drug delivery. He is the recipient of BST Inspire Faculty Award. He has conducted sponsored research projects from DST, ICMR, and various other funding agencies. He has also co-authored over 100 publications. Recently, contributions from his group and collaborators have been listed. He has been listed in top 2% cited authors globally in the area of biomedical engineering by the Stanford group. So with this short intro, I would like to welcome Pallab. So Pallab, please, you are welcome to share your screen, and please, over to you. Thank you very much.
Thank you very much, Dr. Falguni. So am I audible?
Yes, you are audible. Yes, you can share your screen. Yeah. Yes.
So once again, thank you very much, Dr. Falguni. It is in fact very intimidating for me to speak on this occasion, where the chair in itself is one of the best exponents of the subject in our country. but still I will try my level best to present whatever little works that we have been able to do, and justify the role of bioprinting in physiological research. I will also like to thank at the outset the organizers of the conference or of this symposium, Dr. Amita Asthana, Professor Shashibala Singh, Madam, and Dr. Shantimay Kaur for giving me an opportunity to speak in this August, in front of this August audience. So the topic of my talk is facile bioink modifications for development of 3D bioprinted constructs. However, being the first, by virtue of being the first speaker of this session, may I take a few minutes to very briefly talk about the basics of the bioprinting, how the bioprinting comes into the picture, how it has evolved. And then after that, I will like to go into some of our works on this bioprinting. And finally, I would like to show that how this technique is essential, very essential for physiological research. So bioprinting is actually one of the manufacturing techniques. In fact, it has come from the word 3D printing, and 3D printing, as you know, is one of the is also synonymous with additive manufacturing. So our conventional manufacturing, in that, let us say we had to make a sculpture, or if we have to give a particular shape into an object, and then in that case mostly the conventional manufacturing processes were subtractive manufacturing. For example, we'll start with a block of raw material and then cut it into a desired shape to give whatever shape of the object that we want. As opposed to this subtractive manufacturing technique came the technique of additive manufacturing, in which the object is built in the form of a layer by layer process. So one after another layers were added to build up the whole process, and that is why it is also called as bottom-up approach compared to the conventional top-down approach. So now in 3D bioprinting or in bioprinting, what it is actually done is that we start with a digital file, which is a blueprint, and this digital file or the 3D design is created in one of the CAD softwares. So we have AutoCAD or you have SolidWorks like that. So various softwares are there. Then from that CAD software, this design is transferred into another software, and where it will show that how the transfer process takes place. So once it is transferred to a second software, the second software slices this object. So let us say I have got a particular shape. So this shape is sliced into different layers. So after this slicing into different layers is completed, then instrument called as a bioprinter or a 3D printer, it then deposits the material, okay, at the different places as defined, or in a single layer as defined by the 3D design. So this is exactly what happens in the case of 3D printing or 3D bioprinting. Now, why this 3D bioprinting is called? Because 3D bioprinting can allow us fabrication of objects using living cells. If you talk about any of the other fabrication methods, let us say casting. So if I am say trying to make a hip implant with titanium or stainless steel as a raw material, by the process of say casting or by the process of say CNC machining. So in each and every case, there is high temperature, high pressure, or such high harsh conditions that are involved, such that we cannot fabricate any material using living cells. So if I have to cast, I have to melt the titanium at around say 1400, 1500 degrees Celsius or so. So our cells will not be alive in that harsh temperature conditions. But bioprinting fabrication technologies allow us to fabricate any object into a desired shape, not only into our desired shape, but using living cells as the raw material. So that is why this has become so important in today's world. So we have got within 3D bioprinting, what we can do is it allows us the precise placement of biomaterials and living cells in a layer by layer fashion with the spatial control onto the fabricated 3D structure. So we have a design as per our choice. We have the materials as that is found in the living organisms. Now this 3D bioprinting like if I am starting from the raw materials, which is made up of say biomaterials and living cell, the whole object, okay, after the bioprinting also can take place. So from the say having our cells as and the extracellular material components like collagen as the raw material, from there after that I am positioning or depositing these things into a desired design or desired shape. After that, the conversion into the tissue or the conversion into the organ can take place either by three approaches. One is in which we do not use any scaffolds or any extracellular matrix. That is the case in which we exploit the natural self-assembly or the natural autonomous self-organization properties of the cell. So once we have got say one cell, it divides into two cells, it goes into four cells, it lays down its own extracellular matrix and then gets the shape or the morphology, okay, as is found in a particular tissue. On the other hand, we can also use a sort of template in which we can produce or we can provide these biomaterials on which the cells can grow and organize into itself. So we can either use only cells or we can use cells plus biomaterials, or we can also use mini tissue building blocks. Okay, so variously known as your spheroids, organoids, or scaffold based techniques. Okay, so these are the different terminologies that are being generally used for the different bioprinting techniques. And this ultimately allows us to get the 3D volumetric shape, which is similar to the ones found in the natural tissues and organs. There are basically four modalities of bioprinting. The one which is most commonly used or which is most accessible is the extrusion-based bioprinting, in which this bio ink, which we call is made up of the cells and the biomaterials, can be extruded by mechanical or pneumatic extrusion, and it is just like depositing, okay, after a extrusion process, just like we write with our pen, or just like the toothpaste are extruded out of the toothpaste container, so or the tube. So in the similar manner, the extrusion of the bio ink and the extrusion will take place as per the design that is fed into the computer. Apart from that, we also have inkjet based. So in inkjet based, the mechanism is similar to that what is found in our normal printers in our offices. So that we have got a thermal or a piezoelectric ink head, so which deposits the bio ink, okay, at the desired places wherein we want to deposit, or at what we have given in the design. So now in the normal printer, it deposits the ink in a two-dimensional pattern in a paper. So if we stack different layers or different papers, then we are going to have a three-dimensional construction of the design that is given. We can also have laser-assisted and stereolithography-based bioprinting, out of which although the laser-assisted bioprinting are quite costlier, but stereolithography-based bioprinters, they can be even more accessible than the laser-assisted one. And in both of these cases, they exploit the curing or the cross-linking of a polymer in the presence of light. So light falls onto that particular areas in which we want the solidification or the curing or the structural consolidation of the material. So what I can have a liquid, okay, so a liquid bath in which I allow the light to fall onto only those specific places where I need the solidification, and as the solidification of the liquid at that desired places is going to take place, I am going to have the object, fabricated object in my hand. Now, all these different bioprinting modalities are not comparable in every aspect. They have got their own strengths and own disadvantages. For example, if you see that in extrusion bioprinting, we typically require very high viscosities of the bio ink, whereas the extrusion bioprinting can also give us very robust bioprinted constructs, which is not possible, okay, with inkjet or other inkjet or even the laser-based bioprinting techniques. The resolutions in the resolution in the laser-based techniques are quite higher compared to the resolution of the extrusion-based bioprinting techniques. So that is why extrusion-based techniques have found more use in research, drug testing, and for making tissue engineered constructs, whereas laser-assisted ones are mostly used where we need very precise deposition of the or very high resolution is the demand of the application. Now, because of their ability to give us structures which can be anatomically complex, just like found inside the body, in which there is a high degree of heterogeneity in the cellular organization. So, because of these advantages, bioprinting has come into the community of tissue engineering as well as for the pharmaceutical research, in which we can generate lot of these in vitro models. We can grow the tissues as well as models inside the laboratory and then test them. And that is how why we see that our topic for today's session is the future of animal-free research. So we can make models made up out of mammalian cells inside the lab, which can mimic the physiological and the other molecular biological parameters of the human bodies and sort of take care of the animal research. And in the drug discovery research, we know that the paradigm that is going on is that quick win fast fail. So therefore, if the drug, rather than failing it at later stages of the drug discovery process, the earlier it fails, the better. So that is why it has again such a lot of interest in the scientific community. Now, this bioprinting, even though we schematically say that it is just an extrusion process, is not always a very straightforward process, and it requires several degrees of optimization. Even today, if we see that the major limitations of the bioprinting are that we do not have really very suitable bioinks which are optimized for bioprinting. For example, when we are talking about the bioprinting, a great degree of optimization is required between the printability parameters. That means the conversion of the sol or the liquid into the solid, at what rate this solidification should take place so that it is mechanically robust also, but at the same time it is biocompatible also. The process does not cause a high degree of cell death. So this still the bioprinting community is looking for the optimization of bioinks, which will allow a much faster and smoother fabrication of constructs using this technique. Now, in this regard, what we have done is that we have taken one of the, our approach has been mostly for the tissue engineering applications, in which we have tried to reconstruct, or we have tried to develop certain in vitro constructs which can be transplanted inside the human transplanted in the human body. So we know that there is a great shortage of organs, and organ replacements are donor organs are not easily available. So if we can harvest the cells from the patient themselves, a progenitor cell or a stem cell that can be available from the patient, and then expanded into volumetric tissue inside the laboratory and then transplanted again inside the patient. So this will be helpful for patients who have burn injuries and some other organ losses. So our first work has concentrated on the skin injuries, in which just to illustrate the process, so we are designing this type of scaffolds in the Autodesk or AutoCAD softwares, and then so then we convert it into using another software. We slice it into different layers and then feed it into the bioprinter. Now, once we start bioprinting, as I said, that optimization of the bioinks is very essential. So you will be able to see that what type of structure I showed earlier is this is a very solid structure, very well defined structural components. But if I look into what I have got, is extremely difficult. It's extremely different. So why is it so? This is because of the improper optimization of the rheological parameters of the bioink. So the bioink, it is even after deposition, it is not getting immediately solidified, but then rather spreading even after it has been printed. So we did optimize. So our first work has been mostly on using the very commonly available bioink like alginate, and then we added one of the problems with alginate is that even though it has got a solidification property under the conditions of physiological conditions like temperature 37, pH of 7.4 or so, but then it is not a very active, naturally active material for promoting wound regeneration. So that is why we added honey into this alginate and then did the bioprinting experiments with it. So first of all, we try to see that the type of structures we are getting has got the faithfulness with or fidelity as per the design structure. And after doing that, we also look into the various other physiological parameters. So for example, if you see that if I keep on adding honey, the pH of the alginate honey solution is going to come down. So now I stay at a pH, okay, which is still acceptable to me. I stay at a concentration of honey and alginate, so which is going to give me mechanically robust constructs, and then finalize it for my further development. Now, then I do the several cell culture experiments and to see that these scaffolds are compatible with the cells. Mostly we have checked because here the application was the skin tissue engineered construct, so we checked the compatibility with fibroblast and keratinocytes. Then, so this was one example of ours in which we developed the skin engineered constructs or skin tissue engineered constructs made up of fibroblasts, keratinocytes, alginate, and honey as the biolinks. And we are able to see that incorporation of this honey component will accelerate the regeneration of the of the keratinocytes inside the scaffolds. Our second example was to take polyglutamic acid, which is found inside the bone. Now here also with so just alginate, which is a very popular bioink, but it suffers from the problem of less biocompatibility. We added polyglutamic acid and then used it for bone tissue engineering. And there also we saw that we will be able to optimize the properties of bioink so that it remains flowable. It solidifies after the flow, and at the same time it increases the bone tissue compatibility, as we had seen for various ALP and alizarin rate assays. Now, one of the problems that we faced when we are doing this bioprinting is that we could see that just after this extrusion pressure, there is a loss of cell viability, which is of the tune of 20%. So only 80% of the cells were surviving the bioprinting process. So now to overcome that, what we did is that we made bioprinting formulations which will contain a lot of antioxidants. Now here, what we observed was that this loss of cell viability, when I'm saying that I'm giving an extrusion pressure onto the cells and still cells are dying, that loss of cell viability we could see was also mediated by reactive oxygen species. So the shear stresses are resulting in reactive oxygen species generation, and that is implicated in the loss of cell viability. So now we thought that if we can augment the bioinks with this with antioxidants, and one of those which we tested was N-acetylcysteine, we could see that we can improve the process viability of the bioprinting process. Similarly, we have seen the various inflammation markers. Okay, after the bioprinting process, we saw that the stem cells, okay, are maintaining their integrity. We also observed a couple of other things during the bioprinting process, because we had used an extrusion bioprinter, and there you have seen that the bioprinting proceeds through extrusion of the filaments in a particular alignment. We saw that once the bioprinted cells are present in the extruded filament, there is also the matrix that they are producing, the collagen that they are depositing, okay, as a part of their growth process, are also aligned as per the direction of the bioprinting. So this higher degree of alignment was also observed in our studies, and that also shows that the applicability of bioprinting to get aligned extracellular matrix, which can have application in those type of tissues in which a high degree of alignment is present, like the skeletal muscles, neurons, and some of these examples. So there has been some other observations also. For example, if we look into our results of when we try to see that, see, in case of bioprinted, what happens is that I am putting the cell inside a three-dimensional environment. Now, what we see is that if I change the mechanical properties of the three-dimensional environment, then these mechanical properties can be sensed by the cell, and mechanotransduction pathways like the one we studied was the YAPTAS pathway. So a higher degree, so compared to let us say if I do the bioprinted with a 2% alginate solution, in which 2% alginate solution, or if I do the bioprinted with say 4% of the alginate solution, then the degree
prepare the department.
Sorry, Professor Datta, please continue. Somehow I think someone unmuted. Please continue. You are muted now.
Okay, am I audible now?
Yes, please.
Okay, sure. Thank you. So the mechanical environment, as the mechanical environment changes, so with bioprinting, with the technique of bioprinting, we can provide different mechanical environments. And I am saying that three-dimensional mechanical environments. We have already seen studies in which cells which are grown on stiffer matrices or on softer matrices will differentiate into a hard tissue like bone or a soft tissue like neuron. But all those studies, in all those studies, the cells were given only a two-dimensional, so only a monolayer on which the mechanical stiffness was changed. But here in bioprinting, we can give a three-dimensional environment and three-dimensional different mechanical environments, and that leads to different activation of mechanotransduction pathways. We also saw that when we are designing the bioprinting, it is possible to find out the rate of degradation of the bioprinted constructs. And as we know that in tissue engineering, one of the overarching objectives is to match the rate of degradation of the bioprinted constructs or the biodegradable polymers with the rate of growth of the new tissue. And in that direction also, we have been able to model the rate at which a bioprinted construct degrades, whether it degrades homogeneously or not, or there is a degree of heterogeneity. So what are the areas of bioprinted constructs which degrades faster? And now that also allows us to modulate. So for example, if you look into some of this study here, what we did is that we tried to predict what will be the properties of the bioprinted constructs, and if we can predict those properties beforehand just by knowing the properties of the bioink. So for example, if I want to develop a construct, okay, which has got the mechanical properties of the bone, which is different, which is going to be different, okay, from the mechanical properties of the skin or from the tendon or from the ligament. So now what type of bioink is going to be suitable for the bone, for the tendon or for the ligament? So to do that, what we did is that we used finite element modeling processes. We fed a lot of properties of the bioinks, like we varied the concentration of the polymer from gelma. We varied the concentration of the cells. So not all cells or not all tissues have got the similar concentrations or similar cell densities. In the physiological environment also, cell density also differs from one to another. So now what is going to be the effect of these differences onto the final bioprinted constructs, and whether those things can be predicted? Okay, that also we have tried to do. And as you can see that, okay, with these simulations, we are able to find out those areas wherein the cells are having higher stress conditions, or those areas of the bioprinted constructs where they are present, or the less stress concentrations are present. So therefore, higher stress constructs, we have seen that directly correlate with higher cell deaths in the later on stages, whereas lower stress concentration regions, okay, allow for faster development of the or better development of the cells inside the bioprinted constructs. So this has been this allows us to predict, or this allows us to predict the conditions which the cells are going to experience after the bioprinting. And at the same time, now we can design a construct in which we know that, okay, this part of the constructs are going to have higher higher stresses. So therefore, lower cell density may be given in this part of the construct, and that part of the construct may be given a higher cell density. So that is why how we can use this type of predicting algorithms for designing better constructs. At the same time, I would also like to discuss that when we are doing these experiments, or when we are trying to validate the properties of the bioinks, like what type of stresses the cells are going to experience when they are going to be subjected for the bioprinting process, we saw that most of the times what is done is that we take the cell as being part of being mechanically homogeneous. So the cell is given a material property or a mechanical property without taking into account the regional variations in the mechanical properties of the cell. So what we did is that now we developed a model in which, rather than describing the cell to be mechanically homogeneous, we divided the cell into three parts: the cytoplasm, the plasma membrane, and the nucleus, and then gave different material properties and gave different material properties to this different part of the cells. And then after using these three different material properties, the model that we are able to develop, we found that is having a higher degree of resemblance to the natural rheological properties of the cell. And this also led us to the conclusion that the degrees of the mechanical activation at different parts of the cells are different. So for example, if I have a cell which is being subjected to an extrusion pressure of say 5 kilopascal also, so then not all the components of the cells are receiving the similar mechanical forces. So the nucleus is receiving different degrees of mechanical forces, the cell membrane is receiving different degrees of mechanical forces, and the mechanotransduction pathways that are responsible, or in many cases, for cellular differentiation, cellular proliferation. So they are also going to be activated to different extent. So nuclear mechanotransduction pathways may be activated to different extent compared to the ones which are present on the surface of the surface of the cells. So this is also one of the predictive algorithms that we have developed. And also, I would like to speak about one more of our work, in which, as we discussed initially, that one of the most important limitations of the bioprinting process as of today is the absence of vascularization. So it is very difficult to create vascular channels inside the bioprinted construct. So the various fabrication mechanisms that we have are not very efficient to do that. So we also tried to improve on this, and as you can see over here, that we have been able to. So this is a gel structure inside which we have been able to create this type of vascular channels, which are having curved radius also, and these channels are remaining stable for 5 days or 6 days or so. So now these pictures has been taken when the cells were not given. So even without adding any cells, the vascular channels will remain stable inside the gel structure, and they were not collapsing. And then after that, we cultured cells inside these channels, and we have been able to see that cells are also remaining viable inside the channels. Now, this vascularization of the constructs is, I think, one of the most important challenges that we have to solve, whether it is for tissue engineering or even if it is for in vitro models for drug testing. These cells has to remain viable, and these cells can remain viable only when they have got the supply of oxygen and nutrients and the drainage of the metabolites, and that also we have tried to attempt a little bit. And we have also found that these perfused vascular channels are remaining viable for a sufficient period of time inside the channels, and that is a encouraging point to consider further. Now, just to come to my conclusion slide. So I think that from the physiological point of view, there are bioprinting process, even though most of the works that we have done is with keeping into account the tissue engineering applications, but many of these models can be easily adopted to solve some of the problems in the physiological sciences. Also, for example, we know that one of the biggest debates in physiology is that whether we should look into our physiological systems in an isolated way or in a coordinated and integrated way. So what is the feedback mechanisms between the different systems of inside of the body? Now, animal models have not really given us very clear answers for here, because human physiology is different, and bioprinting allows, I think later on in the session we have got some of the speakers who are going to speak about the microfluidic systems, the perfused bioreactor systems, in which it is possible to have a bioprinted model in which each and every there are different compartments which are in which a different type of tissues and cells can be grown, and the feedback regulation between these cells can be studied easily. So this is the way I think so. This may pave the way for our understanding of, as we said, that the philosophy of Bernard, okay, so who used to think that about the coordinated way of coordination between or a much more holistic way of treating physiology compared to our present day paradigm. So we may have certain solutions for that. At the same time, from a physiological point of view, our understanding of cognitive disorders or understanding of cognitive physiology is very limited, and we may have to really think in which physiologists have to may join hands with the bioprinting specialists, so that to try to develop such mechanisms by which we can probably find solutions to that. And let us say that last year when Papaparu and they have been given the Nobel Prize for understanding the behavior of thermosensitive and piezosensitive channels. Maybe if we have at our disposal a technique like bioprinting, much more detailed or much more detailed observations on how these ion channels function can be put up in a smaller period of time compared to what we have gained understanding so far. At the same time, even though we are also interested in some other questions of physiology, like the questions of sexual dimorphism, so we see a lot of autoimmune diseases inside the women compared to men. Then most importantly, patient-specific variations. So we can definitely harvest some cells, okay, from belonging to different communities or different groups, and then look into the behavior. But of course, now the question is going to come that when we talk about tissue engineering products, or when we talk about products for drug discovery, so already products for drug discovery have been commercialized by companies like Organovo. So so it is easier probably to address some of the ethical questions. But if we are going to ask the question that I want to develop an in vitro model to see that how this particular patient is going to respond to a particular disease target, so how the ethical concerns are going to rise, and for what type of issues are we going to get the we are not going to violate the ethical considerations. This may be some of the answers which we may discuss, and then we may discuss and then make the roadmap for the future experimentation. Finally, I would like to thank all my group members and the students, as well as the funding agencies who have supported our work. And also, once again, thank you to Dr. Falguni and Dr. Amit Asthana and Dr. Shanti Maikar and the organizers for holding the event and in the Physiological Society, such an important topic that 3D bioprinting, the future of animal-free research. But I would like to conclude with only one sentence that yes, it is not only the future of animal-free research, but the future is already here. So we can already have bioprinting, which can give us in vitro disease models, and which can be used for understanding physiology at greater extent. Thank you very much.
Yeah, thank you, Dr. Pallab Datta, for the outstanding talk, as always. Yeah, I hope I'm enjoyed to the fullest extent. I hope the audience also enjoyed the talk. So probably we are taking the questions at the end of the session, right, Dr. Kar?
Yeah, we'll take up all the questions at the end of all.
During the discussion, right, at the discussion time. So please put your questions in the QA, and also Dr. Datta, please stay connected. So probably we will have the QA at the end of all the talks. Okay. Thank you for the talk, and also being the first speaker, you have well presented put up the concept of the bioprinting, also connected the dots between how the different bioinks can be selected, how they can be used for printing, and also you have also talked on the modeling, like the stress modeling, and also the modeling that is being used for flow modeling that is being used for bioprinting, and that is very good correlation with the bioprinting process. And at last, you connected well with the physiological modeling that is very much possible with bioprinting nowadays. And you rightly said the future is here. Thank you. Thank you for the nice talk.
Thank
you. So let's move on to the next talk, that is by Dr. Bhavani Tota. So the talk is 3D bioprinting for dental materials, testing a guardian angel for animal models. So let me briefly introduce Dr. Tota. So Dr. P. Bhavani is a great combination of clinician and researcher and academician. She is chief consultant pediatric dentist and myofunctional orthodontist, founded Dhantanam, I hope I have pronounced correctly, Dental Hospital in 2016, Chennai's trusted clinic for oral health concerns. She holds the position of principal advisor at Ave Biosciences. She is visiting consultant in various prestigious hospitals like Rela Institute of Medical Center, MGM Healthcare. She lectured at national and international conferences and congresses about 3D bioprinting. She started a course 3D bioprinting in dentistry. She has, to her credit, various bioing development. The contribution in dentistry, she is the India KOL, key opinion leader for the most trusted Strauman's agent, what is this? Allegiance. Also, she is an international trainer for full mouth rehabilitation under general anesthesia. She is the national and international trainer for hospital dentistry. She is one of the few dentists who holds three Indian patents in dentistry. Dr. T. Pavani obtained her master's in pediatrics and preventive dentistry, MDS, from the prestigious Sri Ramachandra Medical College and Research Institute, Chennai, after graduating BDS from Government Dental College and Hospital, both on merit. She did PGDM from JNTU Kukatpalli. She then obtained an additional training in her super specialty, myofunctional orthodontics, making her one of the few pedodontists who holds wide expertise in braces treatment. So with this brief note, I welcome Dr. Pavani Tita for her talk. So welcome, Dr. Pavani. Please share your screen and you may please start.
Thank you, sir. Thank you for a very brief and a vast introduction for me. So I am working with Ave Biosciences. I am Dr. Pavni. I finished my MDS. And being a clinician, I correlate with research part as well, and how we can translate the research into original usage. One second. So how we can take this forward, how we can use the different materials and different experiment models to be useful in clinical part. So 3D bioprinting for dental material testing is a very, very important topic, because all the dental material testing will happen in rat models. We usually take up the rat models or kind of create an in vitro model with that, and then sacrifice a lot of animals to do dental material testing. So, and dental materials are erupting very fast, and various dental materials are coming into the market with a lot of research. There is something called silico research, in silico testing as well, that is just bioinformatics. Okay, using the laptop, you can figure out which materials you wanted to test. So once you have concluded your basic material testing, so once you know these materials are being already can be taken to human trials. So, but there is always a gap, that though in silico has given you a prompt result saying that these can be taken into human trials, but still you have a gap that whether that can be used, because there are a lot of ethical issues or organizations which want you to show you some results in vitro before proceeding with the your patent or whatever. So you know that these materials can be used in mouth, but still you can't take it directly for using in the mouth. For filling this gap, we are coming with 3D bioprint scaffolds. So this is a guardian angel for animal-free testing. Background, moving on to the content, I'm going to talk about the background, drawbacks, and our invention 3D printed oral biofilm, and other ongoing research. So usually we use Drosophila, which is low cost, rapid regeneration time, small genome, used to study functions of genes and genetic mutations, whereas Xenophus, okay, eggs are easily manipulable, and eggs are fertilized outside the animal, used to study embryonic development. And from the embryonic development, how these teeth has taken its shape. There are a lot of congenital abnormalities in the teeth. To study all those, the embryonic development, we sacrifice Xenophus or rabbit or rodents for that. And zebrafish, embryos are transparent and easy genetic engineering, used to study developmental diseases, basically for the gums. Okay, zebrafish you can use for gum studies, and rabbit you can use for teeth, gums, and most of the other. See, there are some drawbacks with the present 3D bioprinting yet, and there is a lot of research scope. And moving on to the background, mostly we use rodents, extract the teeth or periodontium to test different dental materials. This is the teeth, and the surrounding tissue is called periodontium, which is holding the tooth. So this is exactly a rodent teeth. We euthanize and sacrifice the rat. We actually take up for the ethical institution and then get the ethics and then kill the rat only to test whether the material is being viable or not, how sticky is to the teeth, or how pulp. The pulp is the nerves inside the tooth, how it is reacting with the teeth. So so what I have done is I have started experimenting with alginate, and then we printed the alginate. And first, when I have printed, the material was not constant, and then it was very kind of very thin, because I introduced calcium chloride much earlier than the print, and then it became a noodle kind of thing. And these are the flaws which I have done. But this one I printed and then introduced the calcium chloride, and it started becoming calcium alginate, and then dissolved. So how we will print, as already the previous guest lecture has speaker has told about alginate. So I'm not going into the details of the alginate. It's a polysaccharide, and then it is very good with mesenchymal stem cells, and also it is very innate with other lot of dental material testing. So it's a very good, easy material to prepare as well. So you just need to take 1% alginate, 2 grams and 5 ml of water. You have to mix it in figure of 8 to make a very homogeneous mixture without any voids. Once you prepare your alginate bio ink, then you can start printing your whatever STL file or your design code you can print. For me, I just printed a rectangular scaffold. So and then you can heat your sterilized solution calcium chloride to 40 to 50 degrees centigrade. After the printing has finished, then you can use this calcium chloride, and the cross link will happen for 15 minutes at room temperature. If you leave it for a long time, the whole structure will get distorted, which I have shown. This will melt the fresh while chemically cross linking the alginate. And what gauze you use for printing, it is gauze 0.25 I have used. So I will show you how we have done the printing. This is the Hawaii bioprinter, which is available in the market. So we use the Hawaii bioprinter for fabrication of alginate scaffolds, and a rectangular scaffold is being printed with honeycomb pattern. At the same time, we have included one more proprietary material for creating the microfluidic channels in it. So material in this, basically it has been printed, and then one more syringe, if you include, the voids can be included with either collagen, or you wanted to test with salivary models, you can take the saliva and then also include. Why do I need saliva? Because to create environment of the oral mouth, okay, I need microbiome from the mouth. So I extracted the bacterial suspension. Okay, I have selected 5 human donors, and they are not supposed to brush for 24 hours, and they should not take any food or drink for 2 hours before the saliva collection. And then stimulated saliva with paraffin chewing, and then that saliva was being kept on the ice. Saliva is diluted in glycerol to a saliva concentration of 70 degrees and stored at minus 80 degrees. So once this bacterial, we created a bioreactor as well. So the bacterial suspension prepared from the human saliva in a cultural medium, the about colony forming units are about 10 power 5. Okay, and then 30 ml per hour is being pumped into the bioreactor channel, and this is the 3.5 centimeter into 2.5 centimeter alginate with other proprietary material as a fresh we have printed. And then we have continuous suspension of the bacterial suspension is being induced. So what are these uses? Yeah, you have created a biofilm and using an alginate. Okay, alginate, why I selected alginate? Because alginate actually helps in bacterial inoculation very easily. So that is why I have selected alginate, and it is very easy to prepare and print. And because of its chelation properties also, it is my one of the favorite material. And once you prepare this model, okay, and this scaffold with continuous bacterial suspension, you can also must have used your other materials for testing into the voids. Okay, there are few voids here, okay, which are being produced during honeycomb pattern or whatever pattern you can take with the microfluidic channels created. So in that areas you can put your materials like GIC, glass ionomer cement, or composites which you are using, because we need to also know how much GIC, whether that has any antiseptic properties. What is glass ionomer cement? We use it glass ionomer cement for filling in the mouth. Okay, any fillings in a tooth, we just use glass ionomer cement. It has liquid and polymer. We mix it, and it has a setting time of 2 minutes. But if you mix very thin, in 1.8:1 ratio, it will have a more setting time. So, and then you can use this along with the scaffold in microchannels to test whether it is able to release fluoride constantly. What is fluoride? Fluoride is helpful in creating anti-cavity effect. Okay, fluoride is there in saliva usually, but still the fluoride which is available in saliva is not bioavailable for a long time, and it is not. It depends upon how much bacterial load is there and how much you are taking inside the fluoride. It all depends upon other things as well. So fluoride releasing effect of such kind of glass ionomer cements is very important to create an anti-cavity effect. And also when we are using GIC, whether that has any antibacterial effect, you can test it. So how do you test? Once you have this scaffold and done with GIC and alginate, you take up this and then do signaling pathway for biomarkers for inflammation, whether inflammation is being reduced or not. And fluoride elementary elementary studies can be done and can see whether the baseline fluoride is the same as post printing after a while. Okay, you can have your own protocol for minimum of 24 hours to 3 days, because the final setting time of a glass ionomer cement is 24 hours. For after 24 hours, how much fluoride is being released into the environment. For all these things, how we used to do before, it is we used to extract the tooth. Okay, extract the tooth of a rat, and then we make a cavity, and then we will induce the cavity, and then we will fill that cavity and see the reaction, how it is happening. The drawback of the scaffold right now, what we have prepared is, we cannot create a scaffold with the other effects like for example, if I am doing filling in the rat, okay, I will know definitely what are the CNS effects, what are the other effects of the body, but you can't create your own body as such in a 3D bioprinter. Still there is a lot of research and gap that has to be filled, but for materials like a filling material you can do. Okay, because it is not going to be taken up systematically too much, because after the setting time is done, there is very less chance of byproducts which is crossing into the blood circulation, because the sealing will be proper. So such kind of studies can be carried out, but at the same time you cannot carry out deeper studies. So mesenchymal stem cells has a good opportunity to react with alginate. So in pulp regeneration studies, when you wanted to do any pulp regeneration studies along with the mesenchymal stem cells, you can use this scaffold along with cytokines or PRP extracted from the patient old blood and then induced. So the pulp regeneration materials can go, the cytokines will help in the regeneration of the pulp. So such path breaking research can happen. That is what we are right now doing. So we are experimenting with this scaffold and placing into the pulp chamber and recreating the regeneration of the pulp in the tooth, so that there will not be need of a root canal. You need not to take out. What is a root canal? Root canal is taking out all the pulp and making it dye and then filling it with some materials. So but right now with pulp regeneration materials like these scaffolds, you can easily test in vitro, and it will speeden up like our corona vaccine, how fast it has come. The similar way, this 3D bioprinting will speeden up the process of in vitro, in vivo, and human trials in a fast pace. So we can also create a tooth-like structure. It is one of the authors who have antomically shaped tooth has been printed for periodontal regeneration by cell homing. So he has created a teeth which looks like rodent teeth. So this is like path breaking. So we can easily use this kind of tooth structure for seeing a lot of other materials. So my conclusion, 3D bioprinting for true animal-free testing is still a decade away, but with a lot of people involvement, it will become much easier. Thank you.
Yeah, thank you, Dr. Papavani,
yes,
for the nice talk on showing us the way to use 3D bioprinting for dentistry, for dental applications. So I hope the audience also enjoyed the talk. We are taking the questions at the end, so please stay connected. So we'll move to the next. Topic. Let me introduce the next speaker today. Is Dr. B Kiran Kumar. He is a scientist in CCMB. So he completed his undergraduate and postgraduate and PhD from Andhra University. Thereafter, he had his postdoctoral training in Yonsei University College of Medicine, Seoul, South Korea, where he also held the position of research professor. After returning to India, he joined CCMB, Hyderabad, where he is currently working as a senior scientist. In his accomplished research career, he has more than 35 publications in journals of higher international repute and a few book chapters, grants from prestigious funding agencies like CSR India, DBT India, and many others. He delivered several invited talks in different conferences, both at national and international level. His current research focuses on the investigation of specific microenvironments regulating stem cells, crosstalk between stem cells and their niche components against therapeutic disorders. He also focuses on investigating neural injury and its repair mechanism under different conditions by exploring different 2D and 3D cell culture platforms and animal model systems. So he will be talking on organoids as intermediate disease modeling platform. Dr. Kiran, to you. Kiran is a very good friend of mine and collaborator, so I'm very interested to hear your talk. Yeah, please.
Yeah, thank you so much, Falguni. It's been a nice introduction. Thank you, Shanti Moy, for inviting me for this wonderful session, and also Dr. Amit, who is my colleague in CCMB, who has been eventually working with me also in these 3D models. But anyways, I'll try to share some of my results, which I want to focus here now. So is my slides are visible? Hello?
Yes.
Yeah, I'm audible also, right?
Yeah, please go ahead.
Yeah, okay. Yeah, good morning, everybody. So myself, Kiran, scientist at CCMB, working on different neurodegenerative models, at the same time establishing some platforms using iPSC-based cell culture system. Right now, what you're seeing on the background is a beautiful picture of the cells that are profusely infected with SARS-CoV-2 virus. The green part indicates the viral particles, whereas the red part indicates the phalloidin, that is the intermediate membranes, and then the nucleus is represented as blue. So this is achieved with a 3D-like model system where we have done with some cell lines, which are called as Vero cell line. We made into wall-like structure. We tried to induce some infection of SARS-CoV-2 viral particles and try to see the infection at every level, how it is infecting the cells, whether it is staying in the nucleus or on the cytoplasm or on the cell membrane. So with this, I'm going to start my presentation. Basically, I'm a cell biologist, and then I try to focus more on the stem cell related aspects which can drive into the self-organization, or you can say like organoid like formation, without introducing any of these bio printing aspects in my talk, because for bioprinting I'm not an expert, but I've been collaborating with several other people who I want to introduce bioprinting in my research career also. When you look at the stem cells, it starts from a single cell embryo. When it's a 3-day embryo to 5-day embryo, it's called as the intracellular mass is considered as the source for the embryonic stem cells, which are considered as pluripotent, where it can form into all the organism. From 4 weeks to the 6 weeks embryo, they are pluripotent, where they have a restriction to form all the germ layers. They can form all the germ layers, but they cannot form all the organ systems. The sources are different from different stages of development, depending on the source of, or depending on the requirement of yours. You can isolate the cells from different types during the development process. And the cells also can be isolated from the adult and also from the infant, whereas the cord blood stem cells are considered as one of the essential things where they can preserve the cells for the future clinical abnormalities. That is a debatable thing, but the practice is still going on. At the same time, the adult stem cells, as you know, like from the adults also we can isolate the stem cells, which you can make it as pluripotent or can be remained as multipotent. And then what is the unique properties? The unique properties is something like they have a very good renewable capacity to form for longer times without losing their potency and retaining all the transcription factors. And as stem cell, they should not be specialized. They should be always unspecialized, so that whatever experiment, or you can say like the developmental cues you give, they should transform into that particular cell type. And it should not be committed. It should always be uncommitted until we give a signal to develop what kind of specialized cell you want to give it to it. When you classify the stem cells, they classify based on the source and also for the differentiation potential. The source, I already said before, it can be either from embryonic or it can be from adult. And if you talk about the differentiation potential, it ranges from the totipotency to the unipotency, depends on the state of the cell, whether it is uncommitted or committed. Yes, so I'm not going to talk more about the different aspects of the embryonic stem cells, because it is not ethically proven, or you can say like ethically approved. So people try to derive the stem cells, or you can say the pluripotent stem cells, either not from the embryonic stem cells, but from the iPSC route. So as the name itself indicates, it is induced, where you induce some pluripotent factors into the adult cells, wherein you trigger some transcriptional factors to reverse them into pluripotent in nature. Whereas this is a pioneering work from Yamanaka and his colleagues, where they have got the Nobel Prize for this work, where they have identified 4 specific or important transcription factors called SAMI, COG4, KL4, and F6, where they took the fibroblasts, which are committed cells, and then they tried to induce all these 4 transcription factors. And these 4 transcription factors could again reprogram the adult stem cells to the pluripotent in nature. So this has revolutionized the whole world of looking at manipulating these stem cells at any stage. So this is a very great work from this group. So why we can what is the purpose of having this IPS is because it can reduce the ethical concerns, where you don't need to go for abortion of the fetuses, or you can say like the living material which are not able to develop further. And at the same time, the controllable nature where it can be programmed in a way what you want to do it. So it can be controllable differentiation can be achieved using this induced pluripotent stem cells. And then the most important is you can isolate the cells from the patient, and you can make a patient-specific iPSCs, and they can be either used for drug targeting, or you can drug targeting using the genetic manipulation, or you can use it for drug screening, or you can say like you can introduce new drugs and then see the response of this patient-derived iPSCs, whether they're responding to it before going for a broad spectrum treatment to the specific. So it's kind of personalized medicine which can be achieved using this iPSC route. So as I said before, it can be directly taken from the patient, and it can be maintained indefinitely, because that's the nature of pluripotency, where it can form the colonies, or you can say the stem cell colonies for a longer time without losing their stem cell properties, and they share the similar properties of embryonic stem cells. So yeah, why the stem cells are more valuable? Because that can be used for therapy purpose. They can use for research, where they can go for genetic and molecular and biological control of tissue growth. And also you can able to understand the functions of the genes or the proteins that have been responsible for the pathologies or abnormalities that causes due to this due to the aberrations in the genetic makeup. And you can go for a new drug testing, where you can check for the efficacy and toxicity screening methods for new chemical development. So from the pluripotent stem cells, you can isolate either hematopoietic, endothelial. It's all different, depends on the differentiation from what germ layer you're looking at it. Yes, and coming into this this topic, we have the pluripotent stem cells, wherein you have all these transcriptional factors that have been induced inside. And this is the adult fibroblast. When you reprogram these cells, they form into an iPSCs, which could be either in the form of colonies. And from these colonies, where you have an uncommitted or undifferentiated cells, and you have all the three germ layers at your disposal, and you can able to derive a specific organ type, depending on what kind of research question you're trying to address it. And from these organoids, from this iPSCs, the organoids can be generated. This is a purely self-reorganization of the cells by providing different specific growth factors at specific time of differentiation. And as of now, there are different organoids that has been created through iPSC route. It is OptiCup. OptiCup has been created using the organoid route. The pituitary gland has been created, intestine, thymus, liver, and several other factors are also in. This is a brain organoid, where you can see the organization of the hypothalamus, or you can see like the hippocampal region and subventricular adjoint region. And so this is the generation of the organoids, or you can say like how the organoids has been like taken its course until 2022. 20. So this is what we consider as the dissociation of reorganization of experiment done in amphibian proembryos, and then the differentiation of these embryos into embryoid bodies in early 60s, and they started making into epithelial-like cells using extracellular matrix extracts, and they could able to differentiate into alveolar type 2 cells, which represents the airway organs. And right from there's a lot of leap that happened from 2006. People have kind of taken into several controls on the specific differentiation stages and tried to grow different different organ types in this decade. So they started growing this gut organoids from adult intestinal stem cells. They grow. We have grown this the retinal organoids, the prostate, lung organoids, gastrolites, mammary glands, fallopian tubes. So So majority of the important organs are now can be seen as an organoid model, so without using any bioprinting techniques, with the core property of self-reorganization, which happens when there is a control at different stages of differentiation. So recently, they have grown a snake venom gland organoids, wherein they could able to produce the venom also in that organoids. Yeah, coming into how this is different in vitro and in the body organization. You start with the monolayer cultures, and you, at the same time in the body organization, you have subcellular control of all the parameters, and then from the monolayer cultures, you try to make spheroids, whereas from subcellular form, they make into cells, and cells into simple tissues, and then the tissues have been layered with different tissue types, and they form into organ and organ system. That's how the body organization happens in vivo. But when you try to mimic the same condition in vitro, you start with your monolayer culture, and then you try to aggregate these cells to form a spheroid-like structure, wherein you can have a cell-to-cell interaction and ECM to cell contacts, wherein they can form and they induce a factor called a self-reorganization, depending on the signals that has been given to them, and they form into organoids, and then they can use a tissue explants, and this can be modeled as a multiplex model chips. So this has all been controlled by biochemical tools and also the complexity of the culture, and this is how it looks. And here is the place where we introduce all these methods like bioprinting or bioreactors, wherein you try to establish more cell-to-cell contacts, and then the transition of the small molecules from one cell to one cell type, and then try to make into a assembloid-like model. So we discuss this. We'll discuss this assembloids later. This okay, and then you try to check this applications, so how we are going to use these things, and this is system scale, and then you try to make at subcellular level, like you try to control at subcellular level, at genetic level, epigenetic level, and when you talk about the stem cell level, you control the niche that actually governs the cells, the cell types to form into a particular organ type, depending on the growth factors what you're providing, or depending on the ECM factors what you're providing to get into one kind of cell tissue type. And when you talk about this control here, and there is a tissue compartment, and then you start controlling the cell organization called as self-reorganization at the tissue compartment. And when you come into the organ type, you kind of control the function of the cells. So you control the cell organization at some stage, and then you try to control the structure and function of at the cells at that other later stage. And this is how the bioengineering strategies that has been implemented, or you can say like introduced, to make it more robust, trying to integrate the bioengineering techniques and the self-organization capacity of the stem cells to get into more relevant clinical applications, right from the organogenesis, and then to study organogenesis, and then to study pathogenesis, to try new drug entities for drug discovery models and toxicity assessments, and also the patient, I'm saying like about the personalized medicine treatments, and also the in vivo disease modeling, and they can also be tried for kind of organ transplantation. As of now, it's a far away process, but still the existence lies in future, wherein you kind of make an organ surrogates. You culture this tissues outside and try to introduce in the patients, and you have a built-in response wherein there should not be any organ regeneration. You try to manipulate the immune system in such a way it should be host compatible, and the therapeutic transplants and the repaired autologous organs. So this kind of vast future is available when we talk about the bioengineering strategies with complemented with complemented with the regulation at cell and subcellular levels. So okay, now coming into the difference between what is the 2D and the 3D cultures strategy is. And then because you already know they cannot purely or they cannot overlay the simplistic and cannot accurately reflect the in vivo complexity, whereas it also cannot have a major contribution for drug testing also. It can be a frontline, but it cannot be the ultimate thing for the drug testing platforms. At the same time, when you talk about the morphology, the 3D systems resemble, or you can say like have a close relation to more natural form. And one thing that has a activity in generating model is it takes small, I mean like very less days to make a model with 2D, but for making an organoid, it takes weeks to months. And there will be more in vivo expression patterns at the genome and proteome levels when you talk about the 3D, which may not be so relevant with the 2D model system. And coming into the culture systems, you have only a uniform culture system in 2D, but when you talk about the 3D, you should have a perfusion model system wherein you the whole small molecules or whatever growth factors that have been supplied to the organoids could reach the core of the body. Sensitivity, it has an improved productivity when related to 3D system, and compatibility for workflow. Both have some kind of drawbacks that can be discussed later. And assays are more, it's not, the assays is also not very much close to the in vivo system, but the 3D could be a reach out model when compared, and complexity is more challenging in 3D, and then the cost is high in the 3D culture systems. Yeah, some of the common methods that normally used for spheroid and organoid models, people use suspension cultures wherein they try to make as a concave surface and then try to aggregate the cells in the center. At the same time, there is a centrifugation method wherein you have a non-adhesive coating and you try to centrifuge and then get into ball-like structure at the end of the culture disc flask. And you have a stirring method wherein you concentrate the cells at one specific time, and then you try to concentrate the cells at one particular space. At the same time, the roller method is also available. So apart from this, there are several methods available, but mostly this is the process that normally they do for spheroid cultures, not the organoid, but spheroid cultures. So the why we need this platform. So this is an emphasis on the high throughput and also emphasis on the validation. So because we talk about animal-free testing, we emphasis on sacrificing more animals for one particular drug. Instead of that, if you have a high throughput screening using an organoid models, you can screen a large number of drugs, or you can create large number of mutations in a single plate, and it can be used as both throughput and high validation platform. And right from here, for the drug discovery, you can have a target selection. You can have from the target, you can actually make a lead from the target, and then you can optimize it, and you can go for preclinical, and you can go for clinical, which can be used as a regulatory studies here. So this is a routine in vitro screening, and it is where they use the mice, or you can say mice or humans for the models. But we try to reduce this use of animals. We try to use the organoid models for more in vivo predictivity, and then capacity, or you can say like the emphasis on the validation. So these are the different organoids that has been generated during the course, and we have a good several organoid models, optic cup models, tongue organoids, stomach organoids, colon, intestinal, pancreatic, liver, lung. So major organs are already been taken its shape to resemble an in vivo environment. So when we talk about how this organoids looks like, and what is the microarchitecture of the organoid, it's very essential to see where exactly the growth factors and where will be the percentage and everything. So if you look at these organoids here, and organoid is a kind of a spherical ball wherein you have a zonation right from periphery to the innermost, where the innermost is the necrotic zone, necrotic area. This is the place where there is a they need an intervention of all the biochemical strategies to have robust cultures of perfusion system, where the gaseous, or you can say like the growth factor, should reach inside the core region. And and you have within outer layer, which is used for the drug penetration, or we can say like the excretory stuff, or you can say the oxygen nutrition growth factors and cell lithium contacts. And then this is a quiescent viable zone, and this is the proliferating zone, and this is outer core, and then the middle core, and then the inner core. So the inner core is the most the area which is compromised of all these things, because some of the things will not actually reach to the maximum level. When you talk about the HEP2 cells, so you have a proliferative zone here, and then you have a quiescent zone, you have a necrotic zone, and then the oxygen nutrients, or also the carbon metabolites, will reach up to this stage. So this is a challenging zone wherein we need to have more intervention, or you can say like more methods to develop to get this the CO2, or you can the oxygen nutrients inside this area. Yes, so this is about the general aspect of the organoids, and then how the organoids are being generated from different organs, and how it is been how it can be modulated at every stage, and how it can be derived to a specific organ type. Right now, I want to share my lab protocols, wherein we try to generate the several organoids, or you can say like the brain organoids, using a iPSC route, and how we have characterized those cells, and then how this can be used for future applications. So the first figure where you see the HIPSS colonies, and then we try to make an embryoid body here, and which seems to have all the three germ layers representing the ecto, endo, and mesoderm, and then we try to induce the ectodermal differentiation called as neuroepithelium differentiation, and we try to expand this neuroepithelial cells to form into neural-like tissue, and then we go for a neural maturation, and then this is how your the neural organoid, you can say the brain organoid, looks like, with the wavy natures, which could resemble the gyre and sulci. And then how we have used these cells to establish the cell types that could resemble the actual brain in a 4-month embryo. So this is the first slide. This is the picture which we show that these organoids are being subjected for the expression studies, to see whether all these three different germ layers are there in this organoid or not. So we have used the ectodermal, endodermal, and mesodermal endodermal, mesodermal, and ectodermal markers to see this organoid, or you can say like the embryoid, could able to represent all the three germ layers. That indicated like these are pure cells which have been derived from HIPSA root. And this is how the immunocytochemistry pictures looks like. You have an alpha fetoprotein, which could represent an endodermal lineage, and then you have a smooth muscle actin, that is a SMA, which represents the mesodermal, and then beta tubulin, which could represent the ectodermal origin. So, so we have a Z-sectioning of this organoid, and then try to see how they are placed in the embryoid body, how these cells are being organized in this specific what you can say like the embryoid body. So you can see the cells are evenly distributed. You can see like the haphazardly distributed, which could represent all the three germ layers. And we try to see whether after the induction, after the embryoid body, when I showed the second slide, we tried to differentiate them into neural ectodermal lineage. So we try to represent the neural ectodermal lineage, and we made a full organoid. So whatever the picture I'm showing here is a full-grown brain organoid model, and how you're going to check whether this has all the cell types, or you can say the majority of the cell types which a brain tissue looks like. So we try to section each and every cell type in this tissue type. So we have done some confocal imaging, and then we try to see whether we could able to say the mature neurons and the radioglial cells. So beautifully, we could able to see the neurons which are labeled green, and then the glial cells which are labeled in red are having a co-localization, saying that yes, the neurons or the radioglial cells which are richly present in the brain is also been reflected in the organoids which we have generated. So this has been published in recently in stem cell research paper, research journal, and we try to see how these cells have been placed inside, and you can see a beautiful co-localization of these radial glial cells which are red, and then immature neurons in blue in this organoid tissue model. At the same time, we try to see whether we could able to see in the any organization like the hippocampal region or the choroid plexus where the CSF has been generated. So we want to see whether this could able to, this tissue structure could able to form these layers of the brain. So we try to again section it, and then beautifully you will see that there is a very good channel that has been representing the choroid plexus, which could be the spaces for the CSF generation. And also the thinking region, which we consider the hippocampal region, is also been beautifully marked at this region, which is represented in green in color. So the choroid plexus in red in color, which shows the region for the CSF production, and also the hippocampal region, which are labeled green for the thinking region. So we try to label this also, and then we saw that these cells beautifully show. I don't know why it is not playing, but this is how this has been labeled to represent the choroid plexus for the CSF junction, and also the hippocampal region for the thinking region of the brain. And then this is the one. So you can see this this TR1, and then you have seen a beautiful choroid plexus coming here. And we try to see whether we could able to see the mature neurons which are kind of differentiated, and also the astrocytes, which are the which you can say like stepping stone, or you can say like the platform for the neurons to form. Yes, we could able to trace those cell types also in this brain organoid. We can see the mature neurons are present everywhere. That is obvious in the brain. At the same time, the astrocytes are also been overlaid on the mature neurons, because the astrocytes will be kind of a layer wherein the mature neurons will be propagated. So you can see the blue, the green here, which represents the mature neurons, and then the astrocytes, which are representing the green area, the red area. So yeah, this is how the co-localization looks like when you G-section this brain section at 10 nanometers in size.
I'm sorry, Kiran, if you can wrap up quickly.
Yeah. So I'm done with it now. So we have the intermediate progenitor cells and also deep layer neurons also been identified in this cell types. And then we have the cholinergic neurons and then GABAergic neurons, also different cell types of the neurons that have been generated in this brain. At the same time, the synaptophysin and then the forebrain, that is the FOXA2, also been generated in this. And also the gamma and then gangliocytes, and then the HSBB expression has also been traced here. So right now my summary will be like we have generated a new line of iPSCs in-house, and then we have submitted to stem cell repository. And this is a very good iPSC cells where we could able to derive all the 3 specific germ layers, and then we generated both brain and the hepatic organoids, and we have seen several neuronal cell types. And we try to see whether if we induce different disease-specific mutations, can we able to see the neuronal abnormalities during the development. And the other thing is like the integration of one organ to another organ called as assembloids, because assembloids, introducing assembloids will be a good realistic form of integrating different organ systems. So research, or you can say like the efforts are also should be in the focus of developing the organs on a chip rather than organ on a chip. So with this, I thank all the organizers and the committee for giving an opportunity to show my lab data and also some of the views on the organoids which we are using right now. Thank you so much. Sorry,
yeah,
I consumed time.
Thank you, Kiran, for the outstanding talk describing the importance of organoids, the development starting from the iPSCs to, and thanks also for showing your interesting work. I hope the audience also enjoyed their talk. So please stay connected. So we'll take the questions at the end. Okay, we have one more talk, then we'll take the questions.
Yes. Thank you.
Thank you. So let me welcome the next speaker for the symposium, Dr. Prajakta Dandekar Jain from ICT Mumbai. So Dr. Dandekar Jain is a UGC assistant professor at the Department of Pharmaceutical Sciences and Technology, Institute of Chemical Technology, Mumbai, India. She completed her PhD in bioprocess technology from the ICT Mumbai in 2010, following which she was the first woman researcher to receive Respire long-term fellowship from the European Respiratory Cow Society, ERS Marie Curie Co-Fund, to conduct research in siRNA delivery systems for alleviating lung infections. She conducted this research at the Helmholtz Institute of Pharmaceutical Sciences, Saarland, Germany for 2 years. Currently, her research work is focused in the areas of nanofibers for tissue engineering, 3D cell culture technology, microfluidic technology for organ development, 3D bioprinting, and green technology. She has guided 6 PhD students and over 30 master students. She has authored over 90 publications in international journals, 2 books, 9 book chapters, and has been awarded 4 design patents during her research career of 15 years. She has received various awards, including NR Kamath Book Award in 2014 and 2020, M. V. Deshpande and Department of Atomic Energy Young Scientist Award, Young Associate of Maharashtra Academy of Sciences, etc. She has also been instrumental in initiating the Department of Biotechnology, Government of India sponsored MTech in Pharmaceutical Biotechnology course at DPST ICT Mumbai, and is currently the coordinator of the course. With this brief introduction, I welcome Dr. Jain to present her talk on optimizing biopolymer-based bioink for 3D printing of microfluidic skin cell culture. Over to you, Dr. Jain. Thank you.
Just one point to Professor Pati's point, that as Dr. Jain is currently in traveling, so we'll show her recorded presentation. So I'm sharing that.
Okay, please. Yeah. So Dr. Jain has shared a recorded talk with us, so we'll be playing that. Thank you, please.
Hello, everyone. I'm Dr. Prajakta Dandekar Jain from Department of Pharmaceutical Science.
Is it visible and audible now? Can anyone confirm?
Yes, it is.
In Technology, Institute of Chemical Technology in Mumbai. And today I will be talking about optimization of a biopolymerase bioink, which was developed in our laboratory for 3D printing of microfluidic skin cell culture. To talk a little bit of our institute, it was set up in 1933 to help the Indian chemical industry and we impart programs in chemical engineering and 7 different branches of chemical technology. In fact, we have some of the best programs in chemical engineering, pharmaceutical sciences, and biotechnology at our institute. The impact of our alumni members on the Indian Scientific Society has been phenomenal, and we have more than 500 plus first-generation chemical entrepreneurs. Our alumni have served as scientific advisors to the Indian government, as well as have headed the national research laboratories. These are some of the alumni members from our institute, and it is typically said that you cannot spend a day in India without using some product or the other which has been developed by an ICT. Similarly, the impact on the International Scientific Society is also clearly seen through this slide, where our alumni members have been elected as fellows of various societies, international societies. They have received highest civilian honors instituted by the government of India. So having been trained in scientifically stimulating environment, when I decided to work on an area of industrial importance, it came as a natural choice. Now, to talk about the importance of tissue engineering, the WHO's Global Observatory on Donation and Transplantation states that over 150,000 solid organ transplants are being performed worldwide, which is only less than 10% of the global requirement of the organ transplants. Moreover, the Ministry of Health in India states that only 15,000 organs are available for transplantation in over 220,000 patients who are on the waiting list. And as a result, about 5 lakh people die annually waiting for the organs for transplantation. In fact, India has the lowest ratio of the available transplants, which is 0.5% donors per 10 lakh individuals. Thus, tissue engineering is an emerging and promising solution to this acute organ shortage. This branch of medicine, it deals with development of methods to repair, regrow, or replace damaged cells, tissues, and even organs. And this particular branch of medicine has a huge potential to revolutionize the medical sciences by offering innumerable possibilities of replacing cells, tissues, and organs to combat injuries, disease, as well as aging. Now, most of the tissue engineering scaffolds, they are based on three-dimensional frameworks or scaffolds which are preferably made using biopolymers. And these scaffolds, they serve as temporary or permanent artificial matrix to accommodate cells and to promote tissue regeneration. Now, the extracellular matrix is a dynamic, highly dynamic structural network which is present inside the body that undergoes continuous remodeling, and this is essential to regulate the cellular responses and the cellular functions in the body. Therefore, the scaffolds serve the purpose of mimicking this naturally occurring ECM. That appropriate selection of materials is thus crucial so as to successfully trigger cellular growth and successfully engineer artificial organ constructs. So these are some of the ideal properties of a scaffold material: biocompatibility of the material which is utilized, then porosity so as to promote cellular proliferation throughout the structure of the scaffold, cost effectiveness so that if the artificial organ construct is to be transplanted, the cost is affording to the patients, especially since deep wounds require a long time for regeneration. Then ease of fabrication of the scaffold by technologies that are easily scalable, non-toxicity, bearing peptides or certain sequences that promote cellular adherence, cellular attachment, and proliferation, which ultimately governs the cellular functions and tissue regeneration, mechanical strength so as to support cell growth and proliferation, as well as suitable functional groups that can be modified by functionalities that will promote cellular adherence and cellular proliferation. So now, engineering of artificial organs and tissues has been attempted by two main approaches. The traditional top-down approach, it involves seeding of cells into fully porous scaffolds to form tissue constructs. However, this approach poses many limitations, such as low degree of vascularization, diffusional limitations, low cellular density, because the pore size, if it is not suitable for the cells to proliferate through, it could result in low cellular density. And this also results in non-uniform cellular distribution throughout the scaffold. However, in contrast, the bottoms-up approach, as you can see here, it is also known as the modular approach. It involves non-diffusion limited cell-laden modules such as aggregates, cell sheets, etc., or inks, which comprise of biopolymers along with the cells, for forming larger structures that have the potential to eliminate all the drawbacks that we saw just now with the traditional top-down approach. So 3D bioprinting is basically a bottoms-up approach. It consists of 3 steps, namely the pre-printing or pre-processing. This includes selection of cells and bioink materials, mixing of the ingredients, and creation of the 3D design with the help of CAD softwares. The bioprinting, that is the actual process which utilizes one or more of these technologies, that is either laser-assisted, multi-material inkjet, or most commonly extrusion, which is being utilized in our work also. We will be focusing on extrusion-based printer. And then post-processing phase, which includes cultivation of the tissue constructs for scale-up in larger bioreactors, the mechanical testing of the 3D constructs, and basically the cellular differentiation and making them suitable for actual intended application within the body. Now, the 3D bioprinting process involves deposition of the bioink by a layer-by-layer approach, where each layer has a thickness of about 0.5 millimeters or less. Several different bioprinting techniques have been developed, although the most commonly used bioprinters at the industrial level is the extrusion-based printing. The bioinks which are utilized so as to incorporate the cells during the process of bioprinting, they are responsible for governing cellular responses, etc. However, there are no standard guidelines which are currently available for the bioinks. So due to the lack of these standard procedures, only a limited number of bioinks are available in the market. And more importantly, there is not a single bioink which is available for clinical use at a commercial level. So amongst all the different parameters related to the bioink, the rheology plays a very important role in depicting the printability of the bioink or assessing the printability of the bioink. Now, there are 2 important parameters, the storage modulus and the loss modulus, both of which they play an essential role which is required for filament formation, the structural integrity of the filaments that are formed, and the mechanical strength that is able to support cell growth and proliferation. Another important parameter concerning the bioinks is their bloom strength, which can provide a numerical value to the bioinks and help in comparison of different materials based on this particular parameter. So in our study, the bloom strength was basically correlated with the oscillatory shear test so as to predict the printability of the indigenously developed bioink. Now, we chose to develop a bioink based on gelatin because it is a thermoresponsive polymer that readily dissolves in hot water, and when cooled down, it forms a gel. Now, this material is extracted by the partial hydrolysis of collagen, which is a natural ECM component inside our body. So, gelatin comprises of RGD peptides, which are made up of arginine, glycine, and aspartic acid residues, and these promote cellular adhesion, cellular proliferation, etc. And basically they help to establish the functions of the tissue in case of the regenerated tissue. So due to these reasons, gelatin has already been used by other researchers for development of a variety of tissues like skin, like cartilages, like nervous tissue, etc. However, because of the limitations in mechanical strength, we chose to combine this polymer with another biopolymer, that is vargam, which is a carbohydrate polymer rich in galactose and mannose units. And because of this reason, this polymer is also soluble in cold water. We cross-link the bioink so as to give it elasticity, more viscosity. Now, because hydrogels, which are typically utilized in bioinks, are inherently soluble in water, they are also cross-linked to render them some degree of insolubility so as to give them the strength to help to support the cells, promote their proliferation, etc. The cross-linkers which we used in this study was glutaraldehyde to cross-link gelatin and borax to cross-link vargam. The bioink formulations were prepared with varying concentrations of vargam ranging from 0.1 to 10% and 10% of gelatin, which was kept as constant. Now, the concentration of these components had a significant impact on the mixing, wherein higher speed was found to be better at homogeneously dispersing both the components. And after this, the formulation was kept stabilized for 6 hours at about 24 degrees Celsius to promote its gelation. Now, as the concentration of guar was increased, we found that the bioink became more and more viscous, and also it became less transparent, as you can clearly see here in this figure. So the formulation containing 0.1 guar had the lowest viscosity, and it's quite transparent as against the formulation containing 10% guar. Now, like I said, rheology is an important parameter to assess the deformation and flow behavior of the materials. It plays a very important role in characterization, and during the process of these investigations, we evaluated two parameters, namely the storage modulus, which refers to the elasticity of the material, and the loss modulus, which refers to its viscosity. Now, in amplitude sweep method, the frequency was kept constant and the strain was incrementally increased to observe the point of breakdown of structure, which is known as the critical strain. The critical strain also defines the linear viscoelastic region, or the LVE region, as I would be referring to during the rest of my presentation. And this region indicates the range in which the test can be carried out without destroying the sample structure. Now, the amplitude sweep can be determined in two modes, with the constant shear stress mode, that is the CSS mode, and constant shear deformation mode, that is the CSD mode. So we carried out the sweep studies both in CSS and CSD modes. And however, the amplitude sweep analysis in CSD mode, it has not been performed till date in case of soft materials such as bioink. However, determination of it is essential to conduct the study in both these modes so as to determine the concrete linear viscoelastic region. Yeah. So as you can see here, it was found that in the constant shear stress mode, the strain was kept constant at 10, as you can clearly see here. So it was found that as the concentration of the guar in the formulation was increased, the G dash and G double dash values also increased, and in the formulation containing 3% GG and 10% of gelatin, that is 3% of guar and 10% of gelatin, the G dash and G double dash values were above 1000 pascals and 100 pascals respectively, which is related to the minimum requirement that is necessary for filament formation, and that is important for bioprinting. So it was finalized that the concentration containing 3% guar and 10% gelatin would be suitable for during the bioprinting application. The same was also observed in the CSD mode. The strain is kept constant, and as the guar was increased, the G dash value slowly went on increasing, and once again it was observed that the concentration containing 3% GG and 10% G had a G dash value which was greater than 1000 pascals, which is the minimum that is required to form a uniform, to result in a uniform filament formation. So basically, when we compared the G dash values obtained using both the studies, that is constant shear stress mode and constant deformation mode, we found that the values obtained from both the methods were nearly identical. Thereafter, the frequency sweep of the material was determined at 1% shear strain, which falls under the LVE region of the material, that is the linear viscoelastic region of the material. The storage modulus and the loss modulus of the material of different formulations were they were studied against angular frequency, as you can see in these various graphs here. And as you can see in this graph on the extreme left side, in case of the formulation containing 10% GE, it had a G dash value of approximately 10 pascals, which means that the network formed by gelatin is highly elastic in nature rather than viscous, and therefore gelatin alone cannot be utilized for bioprinting. However, therefore, we decided to incorporate guargum in the formulation, and as the guargum percentage was increased, the G dash and the G double dash values also increased. And what we observed is that in the formulation containing 3% Gg and 10% Ge, the G dash and G double dash values were above 100 pascals and 1000 pascals once again, which is the minimum requirement that is necessary for uniform fiber formation. So the formulation containing a blend of 3% weight by volume of guargum and 10% weight by volume of gelatin, it exhibited a G dash value of 180 and G double dash value of 1000, as you can clearly see from the chart here. So the G double dash value, sorry, the G dash value is 180 and G double dash value is 1000, which is optimum for extruding a uniform filament at a relatively low pressure. And therefore, it was recommended that the storage and the loss modulus of the bioink formulation should be somewhere around these values so as to result in successful bioprinting. Thereafter, we determined the tan delta value. The tan delta is a measure of the initial friction of the material. It is the ratio. It is defined as the ratio of G dash to G double dash. And the tan delta, the materials having the tan delta value below 0.15, they form droplets, and therefore they form droplets at the tip of the needle during the process of bioprinting, and they are not able to form a uniform filament. It is necessary that bioinks should have the tan delta value anywhere between 0.151 to 0.209 so as to exhibit a uniform filament formation. And this was observed in case of bioinks containing 3% Gg, 3% guargum and 10% Ge. As you can see from the graph here, the tan delta value lies within the appropriate range, that is in between 0.151 to 0.20209, and hence this particular formulation was considered to be favorable for bioprinting, which was to be carried on later. The flow wear of fluids is also described by a simple linear relationship between the viscosity and the shear rate, as you can see here. And if the fluid is a Newtonian fluid, then the bioink will experience a very high shear because the viscosity here is independent of the shear rate. And if the bioink experiences a high shear, the cells will die. However, when the bioink is a non-Newtonian fluid, then a high shear will lower its viscosity. As you can clearly see in the graph here, the viscosity was found to reduce with the shear rate. So this showed that all the concentrations that were tested in our study with different combinations of guargum and gelatin, they were found to exhibit shear thinning behavior, which will reduce the amount of shear that is faced by the cells during the process of bioprinting, and it was found to be a suitable property. This material combination was found to give a suitable shear thinning behavior that is necessary for bioprinting. Later on, we carried out the bloom test to measure the strength of the bioink. The bioink must have enough strength to form a uniform filament, which will govern the structural integrity of the printed structures once the ink is bioprinted. It was found that the gel strength slowly increased as the concentration of the guargum was increased in the formulation, and it was found that all the bloom strength values from 480 onwards, they corresponded to tan delta value, which was between 0.5 to 0.2, as we have discussed on the previous slide, and this was found to be an ideal range for the bioprinting applications. Then we also consist the analysis of the extrusion pattern. Now there are basically 3 different patterns which are possible. In case the viscosity is very low, it would result in droplet formation. In case of ideal tan delta value, it will result in a uniform filament formation. Whereas in case of larger values, it would result in coil-like formulation, an aggregate-like formulation, which I will show to you in the subsequent slides. So when we analyze the filament formation with different concentrations, we found that the low viscosity formulations containing lower concentrations of guargum, they resulted in droplet formation, whereas between 2 to 10%, it resulted in It resulted in uniform fiber formation where 3% GG and 10% GE formulation was selected because it has an appropriate tan delta value, and it also resulted in good filament formation. Now 15% of guargum, the gel too viscous and in oil formation. Positive formulations, you can see the droplet that is being formed, the uniform filament structure which is formed in case of optimum tan delta value, a structure that is formed at very high concentration of the polymer, as you can see.
Can you please?
We also conducted analysis of the minimum pressure that is required for extrusion. The pressure that was required for extrusion increased as the concentration. So this was conducted with needles having different gauges, and the pressure that was required for extrusion, it gradually increased as the concentration of guargum was increased. In this case, the maximum pressure that is generally tolerated by the mammalian cells is around 300 kilopascals, whereas we found that all our formulations containing guargum, and especially the formulation which we are optimized containing 3% guargum, it had a much lower extrusion pressure than the maximum tolerated pressure by the cells, and therefore it was found to be suitable for loading with the cells for further bioprinting applications. So this was a final optimized bioink formulation. Amplitude sweep studies were performed at 10% strain. Tan delta value was between 0.17. Bloom was 750. Minimum pressure of extrusion was 86.52 kilopascals. And minimum force of extrusion was 36. Later on, we also optimized the different bioprinting parameters. Bioprinting trials were performed at pressures of 86 kilopascals using a needle of 26 gauge, and it yielded the thinnest filament having the diameter of about 0.26 millimeter, which was suitable for preparation of grids. So as you can see, we also optimized different parameters such as the printing speed, the nozzle diameter, the layer height, the infill density. Now, infill density is basically, it is a measure of the percentage volume of the printed structure that is occupied by the bioink. Now, when we conducted the optimization studies with these different parameters, we found that at the printing speed was evaluated at 3 different values, that is 5 millimeters per second, 7 millimeters per second, and 9 millimeters per second. So when the printing speed was at 5 millimeters per second, it did not result in any grid formation at all, while there was no excessive deposition that was observed when the printing speed was increased to 7 millimeters per second and 9 millimeters per second. However, there were slight deformities in the printed structure. However, when the speed was increased between 10 millimeter to 14 millimeter per second, it resulted in uniform filament formation, and it resulted in formation of structures with good fidelity. However, when the printing speed was increased beyond 15 millimeters per second, it resulted in irregular deposition of fibers, and because it resulted in a kind of jerk movement on the print head, and therefore above 15 millimeter per second was not discontinued for further experimentation. So the printing speed, so the final optimized values of the bioprinting process were as follows. The needle used for first 26 gauge, the printing speed was 10 millimeters per second, infill density was 10%. The pattern which was observed was a grid pattern. I will also show you a short video of the printing process here. This is the final grid structure which is obtained after the process of printing. So the printed structures were immediately cross-linked so as to enhance their structural integrity, which was done with the help of borax, which was used for guar gum. And successful cross-linking was confirmed due to the appearance of a broad peak at 6.287 ppm in the B11 NMR, which was in the range of standard borax complex, whereas a small peak was also observed for unreacted borax, which did not cross-link the hydrogen. Now, the bioprinting of microfluidic skin was performed using two different cell types, that is dermal fibroblasts and keratinocytes. Bioprinting was utilized over the traditional manual seeding because it allows for a better spatio-temporal control for the development of the 3D skin model. And we used a microfluidic device to do the printing so as to provide the flow conditions that are very similar to the tissue microenvironment which is faced in the native tissue. So the device was designed with the help of PDMS, and as you can see, the cell culture chamber is fitted with a lid so as to control the pressure. Initially, we conducted the studies with static culture of the cells, and your HDF cells, that is human dermal fibroblasts and keratinocytes, were initially utilized, and the toxicity was assessed by live dead cell assay, where the calcium AM is a fluorescent dye, is a colorless dye which is converted into a brightly fluorescent dye by the enzymes which are present only inside the live cells, whereas ethidium homodimer is permeable only in the dead cells. So it was found that the percent viability was as high as 94% in case of the keratinocytes, and it was 77.1% in case of HDF, which was suitable to take on further. We also conducted the culture of the cells with bioprinting in HDF using the HDF cells in the 3D format. And your microfluidic conditions were followed, the microfluidic flow conditions were followed, where the flow rate of the medium was maintained at 5 microliters per minute. And after 3 days, the devices were disconnected and the membrane was taken out for imaging, and we found that a good cell viability of about 80% is seen when the cells were cultivated within the microfluidic device after the process of bioprinting. So to conclude, biopolymeric bioink was studied and evaluated for its printability, storage modulus, loss modulus, tan delta value, and bloom strength. The bloom strength, it can be used as a primary method to determine the printability of the bioink. Oscillatory shear tests and bloom strength tests can thus be utilized as a standard framework for the development of bioinks in future. Further, we utilize the process of bioprinting for 3D printing of the skin cells within the microfluidic device, where the cells were found to be viable. They had a very good viability of over 80%. So a potential combination of these technologies, if they're used in combination with each other, they have the potential to result in reproducible production of skin-on-chip models for preclinical and biological investigations. These are the outcomes of the work that has been conducted so far. And I'm very thankful to the funding agencies that have made all this work possible. And finally, of course, my students without whom this work is not possible. And thank you all for giving me a very patient ear. And I shall be happy to answer any questions that you may have. Thank you so much.
Yeah. Thank you, Dr. Jain, for sharing the recorded talk. And now the floor is open for question and answer. If anyone has questions, please type in the QA or also in the chat box. I also welcome all the speakers to live. And so there are already a few questions. I can see in the QA, there is a question: how far has the development gone in terms of generation of midbrain organoids and its modeling for studying Parkinson's disease? So anyone? Kiran, if you want to answer.
Yeah. I think the question is for me.
Yes.
Yeah. Can I see the question?
In the QA, it is there.
It's there. Okay, just a minute. how far has the development gone in terms of generation of midbrain organoids and its modeling for studying Parkinson's? Yes. Yeah. So all the three different the midbrain, hindbrain, the forebrain has been generated as organoids, and there are certain studies going on to introduce the mutations that may cause, or you can say like some factors that can have a resemblance to the Parkinson's disease. Yes, the active research is going on now, and then not only for Parkinson, there is a model that has been generated for Huntington's also, and also Alzheimer's. And also there is the model has been created for Zika virus research also. So there are a lot of interesting models that have been created using the neural organoids, or you can say like brain organoids as a model system.
Yeah, thanks, Kiran. There is another question for you probably. Can you highlight your insights on screening models of Alzheimer using organoids? Models of Alzheimer using organoids.
Yeah, so yeah, that's what. So Alzheimer's model can be induced while you are compromising on the number of neurons, or you can say like the degenerating neurons during the development. The efforts are going on to control the neuronal growth at every stage of its development, and try to influence some genetic factors which could lead to Alzheimer-like disease model. So as of now, I can say like my lab doesn't work on this Alzheimer's, but we try to induce some mutations that may lead to some neurodegenerative diseases. But organoids, the Alzheimer's model creation using the organoids are already in the way of research now.
Yeah. Okay. Thank you. Thank you, Kiran. So another question asked by Ashutosh Pandey, whether or what viscosity or range of viscosities is ideal for good formulation of self nano emulsion. Anyone in the panel, if you want to answer that question, Dr. Pallab or Dr. Pavani?
I think self-emulsified nano-emulsion, there has been, if you are talking about 3D printing, so 1 or 2 of them have been used. The viscosity 100 to 150 centipars should be good for that. Yeah.
I see. Okay. Thank you, Pallab. Is that answer your question, Ashutosh Pandey? If you are live, you can also. Others, if you have any questions, please type your questions in the chat box or also in the QA if you have any more questions. Yeah, okay. Yeah, you are welcome, Ashutosh.
Speakers and chairpersons of the session, Dr. Falguni Pati and Dr. Shanti Moikar. Sir, so shall we end the session or like any more questions?
No,
yeah, I cannot see any more questions in the chat box. Probably, yeah. Okay. Anyway, the sessions, all the talks were outstanding. I thank all the speakers for their enlightening and elaborate talk on the respective topics. And I also thank the organizers, especially Dr. Amit Astana and Dr. Santimai, for giving me the opportunity to chair this session. Over to you, Dr. Santimai. Yeah.
Yes, so there is one more question I can see now in Q&A that.
Okay, yeah, I missed that. Sorry. Please.
What could be the possible reasons behind the male are more susceptible to PD while mortality rates is higher in females? I'm not sure that whether this question belongs to this session, but anyone would like to comment?
This is PD Parkinson's disease probably, or?
Yes, susceptible to PD means okay. I believe it's Parkinson's.
Kiran, do you know, like you want to answer that? Why males are more susceptible to PD than females? That's the question. I don't know whether if you want to answer that, that's okay.
But they say like the mortality rate is higher in females. But males are more susceptible, but the mortality rate is higher in females. So I may not correlate between these two, but maybe the hormones is the main factor which may play a role for this Parkinson's disease activity.
Okay, yeah.
I don't have any specific thing, but if it is having a disparity in the gender, then probably the hormones is the main thing that plays a key role in controlling the pathology.
Okay, thank you.
I'll just take another 2 minutes to thank the speakers and the session chair. So I would like to thank the NIPER Hyderabad team and Fips organizing team with Dr. Sasivala Singh Madam for her constant guidance to conduct this symposium. And like throughout this session, we have learned by initially the first speaker, Professor Pallab Datta, set the stage with a clear overview of 3D bioprinting, and then we saw the case-specific study with oral dentistry applications, followed by Professor Kiran's iPSC means the stem cells, organoids for brain development studies, and later we have seen physiochemical characteristic studies from Professor Jan. So it covered all 4 different verticals, starting from basics to the applications to a particular physiological domain. And so I really thank for your time and appreciate your presentation. So thank you very much. And I would like to thank the speaker, Professor Pati, for conducting this, chair, Professor Pati, for conducting this session so beautifully, though we're ahead by another 10 minutes. So I'd like to conclude here. Now, over to you, the Fips organizing team.
Thank you very much.
So can we leave now?
Yes, sir. Sure, sir. Thank you all the speakers and again chairpersons Dr. Falguni Pati and Dr. Shanti Moikar. We'll begin our next session in 5 to 10 minutes.
Thank you so much.
Thank you. Yeah, we're leaving there.
Thank you very much.
एक बार ना सर को बोले ये वो रख दो तो नाइपर वाला वो जो है, मैं हट जाऊंगी यहाँ से। I request chairpersons Professor Vinay K. Khanna and Dr. K.P. Mishra, sir. Sir, can we start the symposium 5?
Yes, I think we should start. Mary, yeah.
Okay.
Hello.
Okay.
I'll just take a minute, ma'am. Dr. Mishra, I'll just take a minute because still we have 10 more minutes to start because to begin with.
Okay, fine, sir. So shall we wait?
Can we wait? Dr. Mishra, can we wait for 2 to 3 minutes?
Yes, sir. Sure, sir.
Okay.
Because see that the timing is from 2 p.m. to 4 p.m. I think that we should.
Okay, fine. Fine, sir. Fine, sir. Not an issue. Fine, sir. Thank you, sir.
But definitely we'll start at 1:55.
Okay, fine, sir. Sure, sir.
Thank you, ma'am.
Namaste, sir.
Namaskar, ma'am. Namaskar.
Hello, sir. Good afternoon, Mishra, sir.
Dr. Kalpana.
जी मैम.
Sir.
Good afternoon, ma'am. Dr. Kalpana, namaskar.
Namaste, sir. Namaste, sir. How are you, sir?
बहुत बढ़िया मैम. आप हमारे सेशन की ओपनर हैं मैम.
जी सर, जी सर, जी सर.
मैडम आपको 15 मिनट में खत्म करना है.
सर, I'll try my best.
पक्का जी पक्का.
जी.
बस अभी शुरू करते हैं मैम. 3-4 मिनट पे विल स्टार्ट. हाँ.
Sir, good afternoon. Namaskar, sir. Welcome.
Namaskar, Dr. Khatri sahab. Namaskar.
I think we all are here. We can start, sir, with your permission.
Not a problem. We can start now.
Dr. K.P. Mishra, sir.
ऐसा मैंने उनसे कहा था. वो 2-3 मिनट में आ रहे हैं अभी.
Yes. Yes, we can start.
Okay. Anika, we can start. Please proceed with the official.
Okay, sir. Fine, sir. Welcome all. Now we'll begin our symposium 5, Nutrition, Brain Aging and Neurodegeneration. Now I request chairpersons, Professor Vinay K. Khanna and Dr. K.P. Mishra to take over.
Thank you, ma'am. On behalf of my co-chair, Dr. Kirti Mishra, and on my own, I take this opportunity to thank the organizers for giving us this opportunity to be the part of this symposium, and moreover the fact we really enjoyed the delicacies which were served during the past 3 days, and continuing with the scientific program, as Anika, you have rightly mentioned that this is another symposium 5 on nutrition, brain aging, and neurodegeneration. So without wasting any time, because we have now 5 speakers, one of the speaker will not be able to be with us due to some reasons. So what I will do that before we start the program, I like to share that because the other programs are also lined up just after this symposium. So what we'll do that we'll we'll appreciate if all the speakers, if they are able to complete their talk within 20 minutes, and see that if you are able to complete your talk within 10 minutes, probably the organizers will send you a special gift. This is again in the lighter vein. But I will definitely request all of you to be on time. And in this session, we have the first speaker is Dr. Kalpana Barwan, and she is one of the very fine researcher in the area of cell physiology, stress physiology, and also her specific interest is in the area of molecular signaling in hypoxia. And she is an accomplished researcher in the area, and her work is widely cited. And in the interest of time, I'll not go into the details, because if I go through her contributions, this will take a lot of time. Dr. Kalpana, right now she is a faculty at, and she's working as associate professor at Department of Physiology at AIIMS, Bhubaneswar. And with this short introduction, I'll request Dr. Kalpana to share her findings. Over to Dr. Kalpana, please.
Thank you so much, sir, for your nice introduction. I will ask the organizers if they can see my slides. Are the slides visible?
Yes, we can see that.
Okay, and are the slides moving?
But can you keep it on the slide mode, please?
Okay.
The slides are not on. Yes, now I think that now you can move on the slides first.
Are they moving?
Not now. Yes, it is moving now. Yes, Dr.
It is moving, sir.
Right now.
Okay. Okay, sir. Okay, shall I start, sir, now with your permission?
Sure, ma'am.
Okay. So a very good afternoon to all the panelists, the chairpersons, and all the attendees also who are attending this session. At the outset, I would like to first thank the organizers of Federation of Human Physiological Societies 2022 for giving me this opportunity to present my research work. So basically, this research work was carried out in my previous organization, that was Defense Institute of High Altitude Research. And I will be sharing a part of that research work in this forum. And the topic of today's presentation is alterations in myelination induces neurodegeneration during the hypoxic stress. Okay, so first of all, I would like to give a brief introduction about the hyperbaric hypoxia. So basically, hyperbaric hypoxia is a condition wherein there is a decrease in the barometric pressure. And because of this decrease in the barometric pressure, there is decrease in the oxygen availability to the tissues, which is seen at the high altitude. Now, the soldiers who are basically deployed at high altitude because of their operational need, they are the ones who get affected because of this hyperbaric hypoxic stress. It's not only the climatic condition, but also this particular stress that affects them. And therefore, it affects not only just one physiological response, but there are several alterations that occur, ranging from autonomic alterations, sleep disturbances, and cognitive deficits. My area of research basically is on the cognitive deficits specifically. And before our team took into this particular research area, the research was, the pioneering research was carried out in 1989 by Hornbeam, when he showed that there are impaired learning abilities. And after him, there were several other papers that came up indicating that there are reductions in the psychomotor performance, there's mood disorders, impairment in...
Dr. Kalpana, your slides are not in the slide mode, man. Can you put it on the
Okay, they're not moving, sir. Are they not moving?
Now the slide is, you have the third slide right now.
Sir, are they moving now?
I think that, yes, now it is on the slide mode. Now please continue. Please continue.
Okay, sir. So this was about the brief introduction about the hypobaric hypoxia, and I was talking about the cognitive deficit research that has been done in the area of hypoxic stress. So studies have shown that there are impairment in the learning abilities, there's reduction in the psychomotor performance, mood disorders, impairment in spatial working memory, and cognitive deficit. So the benchmark study was given by Shukit Hale in 1994, after which several other, lot of research groups, they got activated and they started working on this particular area. And of all the regions in our body, brain is the region which gets affected the most because of the higher oxygen utilization. And we were concerned with an area called the hippocampus, because this area is responsible for the memory formation and the memory processing, and therefore could lead to the cognitive deficits that we see at the hypobaric hypoxic stress. Now, our research group looked into several mechanisms that could play a role in mediating this cognitive impairment. And we found that there could be alterations in the ions, specifically the calcium, neurotransmitter alterations, particularly glutamate, and so on and so forth. I will not take much time into this. And this we have published in in several papers and several journals as well. However, what I was interested in this particular work was on a specific brain function at high altitude, which is the executive functions. Now, the soldiers who are deployed at high altitude, because of their operational need, they need to be highly attentive. They should be highly alert. They should have, they should be very quick in their decision making. They should have memory and recall. They should have low response time. However, because they are exposed to this hypoxic stress, they usually suffer from impaired attention, delay in the P300 latency, which is a kind of event-related potential indicating impairment in the neurotransmission. There could be episodes of anxiety, depression, or both as well in them. And this has been proved by several researchers all across the globe. And the benchmark study was done by Dr. Shashi Bala Singh in 2004, who happens to be my guide, my mentor, and she happens to be the president of the society as well. So she did a path-breaking study in 2004, wherein her research group studied that there is an altitude-dependent increase in the P300 latency, indicating that there could be a delay in the neurotransmission, or there could be impaired conduction velocity. And after that, several other groups, related groups, they gave similar findings. And besides this, there were impairment in the other cognitive domains as well, such as delayed recall, procedural memory, and visual spatial executive. However, I was majorly interested into this, the reason why there is a increase in the P300 latency, and what could be the reason for this impaired neurotransmission, or this impaired conduction velocity that we see at high altitude, which could lead to cognitive impairment. So basically, whenever we talk of neurotransmission, we always talk about myelin, which is responsible for wrapping of the neurons in a myelin sheath. And we know that the myelin sheath is required for an efficient neurotransmission because it causes a quick conduction velocity. The electrical impulses are quick with the myelination. Now, this myelin sheath, it consists of both lipids and the proteins. Basically, the proteins are your myelin basic protein and the protolipid protein, and several other lipids include cholesterol, glycolipids, and phospholipids, which could play a role in myelin compaction as well as myelin synthesis. Now, since myelination is important for effective neurotransmission, we wanted to look into the cells which are responsible for this myelin sheath synthesis. We know that oligodendrocytes are the cells which are responsible for wrapping of the neurons in the central nervous system, and Schwann cells usually wrap in the peripheral nervous system. However, these oligodendrocytes, they are highly susceptible to the oxidative damage during the hypoxic stress. And this is mainly because of their high metabolic rate, because of their low antioxidant enzymes which are present inside them. Therefore, they are highly susceptible. Now, besides these oligodendrocytes, there was an interesting paper which was published in 1987, long back, by Comolli et al. And therein they had shown that an enzyme called carbonic anhydrase, they are the ones which could be a pre-indicator for demyelination during hypoxic stress. So this gave us a lead So we wanted to look into this subclass of the enzymes as well. So amongst these, it's a big family of enzyme carbonic anhydrases. We wanted to focus on two important enzyme isoforms, which were carbonic anhydrase 2 and carbonic anhydrase 4. Carbonic anhydrase 2 is a cytosolic enzyme, and it is known to be playing a role in signal processing, long-term synaptic transformation, and gating of the memory storage through modulation of receptors, while carbonic anhydrase 4 isosome is present in the membrane, in the lipid portion, and it plays a compensatory role for the myelin synthesis and compaction. So there was a study by Bryan in 1994, and they showed that when they used carbonic anhydrase 2 deficient mice, they found that carbonic anhydrase 4 was playing a compensatory role, that is playing the role of CA2. It was basically, compensating the role of CA2 in causing myelin synthesis and compaction. So with this literature, we wanted to go ahead with the study with the following objectives. The first thing which we wanted to do, which we wanted to look into, was whether the global hypoxia is causing any effect on the myelination of the neurons. Now, though several other research has been done in the area, wherein they have seen the effect of this myelination in prenatal hypoxia, fetal hypoxia, and other ischemic conditions, focal ischemic conditions. However, the literature on the global hypoxia, effect of global hypoxia on myelination is very, very sparse. So this we wanted to look into. Then we also wanted to look into what is happening to the myelin architecture if it is getting affected, and what could be the mechanisms that could lead to these alterations. So this was the experimental design that we followed. So there were two sets. What was the hypoxic group of animals, wherein we exposed the animals in a specially fabricated chamber, which is the hyperbaric hypoxic chamber. We simulated the animals for different durations of hypoxia, and we simulated them at approximately 25,000 feet of the hyperbaric hypoxia. We maintained the pressure at 282 mmHg, and we exposed them for different durations. There was a similar group of animals which we housed under normoxic conditions, and they served as normoxic for controls. Besides these two groups, we had other sets of animals as well, wherein we give inhibitors to see or look into the mechanisms, the underlying mechanisms, which I will discuss during my results section. So coming on to the findings of this particular study. So the first experiment that we wanted to do was to look into the myelination, whether it is getting affected or not. So the first basic study we did was the Luxol fast blue staining. So the upper panel that you see, it shows the CA3 region of the hippocampus, and you can see the lower panels, they show that there is a increase in the Luxol fast blue stained cell, which indicates that there is an increase in the percentage of the demyelinated cells in the CA3 region. And we found, interestingly, a biphasic response. So in the acute exposure, there was demyelination. However, during the chronic exposures of the hypoxia, we found that there was a remyelination that was happening. This was really very interesting, and we wanted to look more into this. So we also confirmed this particular experiment by doing electron microscopy, and we found similar result. As you can see here, at 7 days and 14 days of hypoxia, there was alterations in the myelin architecture, which you see here. However, on 21 days and 28 days of the hypoxia, there was again, remyelination was seen. We looked into the G ratio, which is actually a measure of the inner axon diameter and overall diameter of the axon, and we found that there was an increase in the G ratio, particularly at the 14 days. And when we correlated the G ratio and the axon diameter, we found, interestingly, that the neurons which are having a high axon diameter, or they were thick, they were undergoing demyelination the most. So this is one thing which we wanted to emphasize here, though we didn't look much further into this. Maybe some other study can, or future studies can be taken up to look why it is like that. But this was an interesting finding, so that's why I'm mentioning it here. Now, after that, once we saw that demyelination was occurring and it was associated with the exposure to the hyperbaric hypoxia, we wanted to look into the expression of the cells that lead to this myelination. So I told you, oligodendrocytes are the ones which are responsible for the ensheathment of the neurons by this myelin. So we wanted to look in both the populations, the mature oligodendrocytes as well as the precursor of the oligodendrocytes. So basically, we stained the oligodendrocytes, the mature oligodendrocytes with GST-PI, and co-labeled them with caspase 3 to look into their apoptosis, their death mechanism. And we stained the precursor cells, the oligodendrocyte precursor cells, the OPCs, with the PDGFR alpha, and co-label them again with the caspase 2. What we found was that there was an increase in the death of the both the oligodendrocytes as well as the precursor cells with the increase in the hypoxic stress durations, and it was very, very high on the 14 days of exposure of the hypoxia. However, we saw that there was a decrease in the death of these oligodendrocytes, mainly during the chronic exposures of hypoxia. However, as you see here, there was no recovery for the precursor cells. So all these precursor cells, they were undergoing death, no matter the duration of exposure of this hypoxia. This may be because of they were unable to mature into the oligodendrocytes because of this continued stress. Now, after knowing about the cells, we wanted to look into the myelin sheath per se. And myelin sheath, since it is composed of both proteins and lipids, so we wanted to look into the expression levels of these proteins as well as lipids. And we found that there was a reduction in the myelin basic protein as well as in the protolipid protein on exposure to the hyperbaric hypoxia, which was very, very significant on the 7 and the 14 days. And we again found a remyelinative phase. As you can see here, there was again, a recovery of this MBP proteins as well as PLP protein on the chronic exposure to the hyperbaric hypoxia. This was about the proteins. Then we wanted to see the lipid profile. When we did the lipid profiling in the myelin fraction, we found that evidently 2 important lipids that were showing a reduction were the cerebrosides and the phosphatidylcholine. These 2 lipids were mainly reduced in the 14 days hypoxic exposure, and we found a significant, very slight but significant improvement on the acute exposures as well. The lipid protein ratio was also showing a similar response as we saw in the demyelination studies also. Now, since both lipids and the proteins, they were getting affected because of this hyperbaric hypoxic stress, we wanted to look into the mechanisms, what is leading to, these lipid alterations or these protein alterations. So we all know that lipid peroxidation is one of the mechanisms that can lead, to the alterations that we see in the lipid portion. So we look into the H&E addicts, which are the hydroxynonenal addicts. It is an important parameter to see in case of the lipid peroxidation. And we found that there was an increase in the H&E addicts on exposure to the hyperbaric hypoxia. And there was a fall in these H&E addicts in the 21 days and the 28 days of hypoxia. And we were wondering why this is happening. But we found when we did the nitration experiments. So when we did the protein nitration, we wanted to look into the protein part, what is happening to them. We did the nitrotyrosine assay, and we saw that there was an increase in the nitration of the proteins as well, which was progressively high. Rather, it was high and it continued to be high even on the chronic durations as well. So mainly the nitration of the proteins was responsible, the protein nitration or the nitrosated stress was responsible for the reduction in the lipids and the proteins that we were seeing on exposure to the hyperbaric hypoxic stress. Now, this nitration of the proteins, mainly the cause for the nitration, the first thing that comes to our mind is the nitric oxide. We looked into this concentration of the nitric oxide, and we found that there was an increase in the nitric oxide, and the expression level of the INOS, which is inducible nitric oxide synthase, it remains the same, indicating that it is being expressed and it is resulting in the formation of this nitric oxide radical, leading to the nitrosated stress, leading to the nitration of the proteins, which is responsible for the demyelination. Now, going further, then we wanted to look into the expression levels of the carbonic anhydrases enzymes. As I told you, we were interested in carbonic anhydrase 2 and 4 because literature was available and they showed that they could play a role in demyelination. It could be a pre-indicator of demyelination. So we wanted to look into the expression levels of these 2 enzymes as well. So when we look into the mRNA levels of these enzymes, we found that there was no change in the CHO enzyme, which is a cytosolic enzyme. However, there was an increase in the CA4 isoform of the carbonic anhydrase on the chronic exposure on the 21 and the 28 days. And when we looked into the protein expression of these enzymes, we found that though there was no change in the mRNA levels, we found that the protein, that it means that, the translated protein was getting affected. So there was a loss of this protein on the chronic exposure to the hypoxia, and the compensatory role was played by this carbonic anhydrase 4 isozyme. So in our study as well, we found similar studies as by the Brian, and we found that CFO could be, compensating the role of carbonic anhydrase 2 on the chronic hypoxic stress. We also did immunoprecipitation of immunoprecipitation study. We immunoprecipitated the carbonic anhydrase 2, and we wanted to see the nitration, what is happening, whether it is getting nitrated, how it is, there's a reduction in this particular enzyme. So we found that there was a nitration occurring. It was evident here by the immunoprecipitation study, as you can see here. There was an increase in the nitration of this particular enzyme, which concluded that yes, the nitration was occurring, because of which there was a reduction in the carbonic anhydrase 2 protein in the cytosol. And the similar we found for carbonic anhydrase 4, we found that there was no change. There was we couldn't find any change. There was an increase on this carbonic anhydrase 4, indicating its compensatory role. Now, to go further, we wanted to look what could be the reason for this compensatory role of the why is it happening? Why this carbonic anhydrase 4 that switching, class switching is occurring for carbonic anhydrase 2 to 4. So we went deeper into the mechanism, and we could spot a transcription factor which was PPR alpha. So it's an important transcription factor which was playing a regulatory role in causing this class switching. So we looked into the expression levels of this PPR alpha, and here we supplemented inhibitor. So we gave an inhibitor called the 1400W, which is a selective INOS inhibitor. So we inhibited the inducible nitric oxide synthase. So there was no nitric oxide radical production. So they should be ideally in the presence of the inhibitor. We shouldn't be having, any this PPR alpha. As you can see, there was no expression was seen in the PPR alpha group. However, in the absence of this particular inhibitor, we found that there was the expression of the CA2 was still there, indicating the role of the PPR alpha in mediating the nitration of the CA2. Now, next, we wanted to look into the morphology of the mature oligodendrocytes and the carbonic anhydrase 4. And we gave, in this case, we gave inhibitor, PPR alpha inhibitor, we gave, which is MK886. Now, in the presence of this inhibitor, we wanted to see whether the mature oligodendrocytes, what is happening to them. As you can see on the right-hand side panel here, on the increased exposure or to the chronic exposure of the hyperbaric hypoxia, you can see the percentage of the oligodendrocyte, mature oligodendrocytes seems to be, it seems to be high. And in the presence of the inhibitor, when we gave the inhibitor, in this case, as you can see here, these the lower boxes, the gray colored boxes, they are with the inhibitors. You can see when we inhibited, then the expression levels of the CA4 was going down, meaning that the CA4 was actually playing a role in preventing the neurons from getting demyelinated. Now, we wanted to also look at the contribution of the carbonic anhydrase enzyme. So we blocked this particular enzyme carbonic anhydrase by a selective carbonic anhydrase inhibitor, which is the acetazolamide. This is a common drug which is being used clinically, and we wanted to look into the mechanism of action of this particular enzyme, and we saw that when we gave this acetazolamide, therein we didn't find any change in the diameter of the exons, which could be found in the previous study as well. However, when we gave when we looked at the G ratio of these neurons, when we gave these inhibitor as well as the acetazolamide, of course to the separate groups, we found that there was an increase in the G ratio as compared to the group which was without the acetazolamide, indicating the contribution of this particular enzyme in mediating the remyelination. Now, to summarize my study of all the studies that we have, which I've just shown. So basically, under the acute hypoxic stress and the chronic hypoxic stress, the inducible nitric oxide synthase mechanism, it gets activated. Now, since inos it gets activated, it leads to the formation of these radicals, which are the carbonyl radicals and the nitric oxide radicals, both during the acute conditions as well as in the chronic conditions, which could contribute to the oxidative damage as well as the nitrosative damage. So oxidative damage mainly of the lipids and nitrosative damage of the proteins. And this could lead to the reduction in the carbonic anhydrase 2 enzyme in the cytosol. This could lead to demyelination in case of the acute stress. Now, during the chronic hypobaric hypoxic stress, though there was demyelination, there was a decrease in the demyelination. However, because of the modulatory action of the PPR alpha transcription factor, there was an increase in the transcription activity of this particular factor, which could lead to the activation of the carbonic anhydrase 4. There was a class switching of carbonic anhydrase 2 to anhydrase 4, leading to remyelination of the neurons on the chronic hypobaric hypoxic stress. So to conclude, we may say that during the hypobaric hypoxic stress, there's an upregulation of the inducible neurons. It leads to the formation of these radicals and lipid peroxidation, which leads to demyelination. On the acute exposure, the peroxynitrite mediated oxidative damage mechanism actually causes nitrosative and oxidative stress, leading to a decrease in the expression of the carbonic anhydrase 2 isozyme in the cytosol. However, on chronic exposure to hypoxia, there's an upregulation or class switching. There's an upregulation of carbonic anhydrase 4 via this transcription factor PPR alpha, which is mediated by the nitric oxide radical. The lower susceptibility of the carbonic anhydrase 4 to the nitrosative damage, it promotes the remyelination and improves the myelin compaction as myelin synthesis in the hippocampus. And the findings of this particular study, it indicates towards the potential role of demyelination, which could lead, play a role in the cognitive impairment that we observe. Also, it provides a preliminary evidence for the inhibition of remyelination of the CE3 hippocampal neurons on administration of acetazolamide, and this can be taken further. This can be taken further for the clinical based studies, wherein we can further look into the mechanisms of the acetazolamide that are leading to the remyelination. This can be further looked into. So with this, I would like to conclude my talk, and I would like to acknowledge Dr. Sunil Kumar Hota, Dr. O.P. Chaurasia, and Dr. K.C. Nag, and Dr. Surya Biswal, who were there with me during this particular study. And with this, I would like to thank for your patient hearing, and thank you so much. On to the organizers.
Thank you, Dr. Kalpana, for your lucid talk. It was indeed a very important aspect of study, that how the hypobaric hypoxia causes the demyelination of the neurons, and you have very methodically presented the mechanism aspect. Now I invite my fellow, the chairperson, Professor Vinay Kumar Khanna, for his presentation on the topic potential of quercetin to modulate signaling targets in neurodegenerative diseases. Dr. Khanna is presently chief scientist at Indian Institute of Toxicology Research. His area of interest is the neurotoxicology and the neuroprotection, development of biomarkers for central nervous system diseases. He has published more than 130 articles, reviewed various books and chapters. He is a coordinator for the System Toxicology and Health Risk Assessment Group, Food, Drug and Chemical Toxicology Group. He is the fellow of the National Academy of Sciences, India, and Indian Academy of Neurosciences, Society of Toxicology, India, and Academy of Environmental Biology. So with this, may I request Professor Khanna to please present his lecture.
Thank you, Dr. Mishra. Am I audible? Am I audible?
Yes, sir, you are audible.
My slides are there on the screen now?
Your slide, sir, not in slide presentation mode. Please make it in slide presentation mode.
Just a moment. I'll just take a minute now. The slides are moving, sir? Dr. Mishra?
No, sir. Your slides are not moving. Can you make it in full slide presentation mode? Actually, we are just looking at the.
I will try it again now. Is it now there on the screen as well in the presentation mode?
No, sir, it is not in the presentation mode.
Is it moving now? Slides are moving, sir?
No, sir. Yes, now it is in a presentation mode. And can you click on second slide, sir? Yes, it is moving.
Fine, sir. So thanks, Dr. Mishra, for the nice words. And before I start, I once again thank the organizers and especially to Dr. Shashibala Singh for asking me to be the part of this August audience and also to be the part of this conference. It has been really a learning to attend this international e-conference and hearing some of the exciting talks. And you see that if you have a good speaker before you, it becomes difficult to meet the expectation of the audience. So what I have done, I have again segregated my talk into 3 components. I'll not go into the detail, but will give you a touch as far as the title of the talk is there, potential of quercetin to modulate signaling targets in neurodegenerative diseases. So you see that the quercetin is a bioflavonoid which has natural occurrence in vegetables, fruits, alcoholic and non-alcoholic beverages, and is extensively present. And because of the radical scavenging activity and moreover the antioxidant activity, which are very well characterized because of its uniqueness in the structure, and the metal chelating property, which are also well established as far as the quercetin is concerned. These properties have been very well attributed to its pharmacological spectrum, which further enhance its anti-inflammatory, anti-apoptotic, anti-carcinogenic, and other activities. And moreover, the fact that it has been found to have very high bioavailability, and the evidences which have been very well characterized that quercetin crosses the blood-brain barrier has further fascinated the neuropharmacologists and neuroscientists to explore its potential in neurodegeneration, especially to understand its protective effect in CNS diseases. A number of preclinical studies have been carried out using different models, especially in the Parkinson's models, where it has been found that quercetin is quite effective to correct the dopamine levels in animal model of Parkinson's disease, where people have used different kind of models like 6-OSDA, and even they have used the MPTP-induced model. And it was also found that quercetin was found to protect the alpha-synuclein levels. In case of AD models, it was found to correct the senile plaques and also to enhance the learning and memory, and also was found to have quite interesting results in the case of the SD model, where people have used the 3-nitropropanic acid, where it was found to effect on to modulate the GABA levels and to reduce the mitochondrial distress. Even it was found effective in the case of ALS model and also in the depression, where it was found to modulate the NMD receptors and NOS. These studies and the leads have provided an interesting evidence to move forward, and a number of animal models have been developed to further understand and to assess whether it is effective in the case of animal model of cognitive deficits, where a number of studies have been carried out using different preclinical models involving the streptozotocin induced model of memory impairment using both mice and rats, where it was found to modulate the oxidative stress and was found to correct the enhanced oxidative stress and also the acetylcholine stress levels. Therefore, the level of the acetylcholine at the synapse was found to be corrected, and it was found to be associated with the learning and memory impairment. In case of amyloid beta-induced Alzheimer's disease model, it was further found to modulate the oxidative stress, which has been very well reported, and also it affected the BDNF, which are known as the trophic factors, especially the BDNF, and also found to modulate the ERK signaling and also the TREB, which is one of the downstream target, and this is one of the important contributor while we talk about the learning and memory. In case of scopolamine-induced memory impairment, where rat model was developed, it was further found to correct the beta amyloid levels and also to modulate the oxidative stress. In one of the interesting studies, it was found that because quercetin, as I had mentioned in the beginning, it has been found to have the antioxidant effects, where people have found that this is the NRF2-ARE signaling pathway is very well modulated by quercetin. And while we talk about the NRF2-ARE signaling, this is one of the key redox sensitive transcription factor that regulates multiple antioxidants and binds to ARE, that is antioxidant response element, and induces the expression of pro-survival proteins and cytoprotective enzymes. And it also, the levels of the ARE has, because where the three important candidates are involved, one is the NRF2, HO1, and the key one, which is one of the excluder protein, which is one of the repressor protein. And the decreased expression of ARE signaling, these targets has been reported, very well reported in Parkinson's disease, Alzheimer's disease, and in other neurodegenerative diseases. It attenuates the oxidative stress and enhances the antioxidant capacity and modulates the transcription factor KEAP1. So this NRF2-ARE signaling was found to be modulated, which was further these changes were very well attributed to control the neuronal cell apoptosis. So with this background, I'd like to share that our interest was to see whether the quercetin has any protective effect in animal model of cadmium-induced cognitive deficits, for which we have been carrying out these studies for last many years in our laboratory. So we already have a very well developed model of cadmium-induced cognitive deficits, where we wanted to see what is the impact of quercetin in an attempt to understand its protective potential, and also to understand whether it has the potential to modulate those targets which are altered by cadmium and they are involved in the cognitive dysfunctions or not. So this is one of the interesting studies that we carried out in one frame, where we found that cadmium disrupts the cholinergic receptor signaling involving the M1 and M2 receptors, and this signaling was found to be disrupted because of the alteration in the presynaptic terminals, especially in the levels of the chat and ACHE, because of which the acetylcholine levels were found to be increased, which disrupted the functioning of the M1 and M2 receptor. And this alteration in the cholinergic receptors was disrupted the PKC beta 1, which is one of the important mediator and involved and associated to modulate the cognitive functions, was also found to be disrupted in the case of on exposure to cadmium. And these changes in the cholinergic pathways, especially, were found to be altered because of the cadmium-induced changes in the mitochondrial functions, wherein we found that this is the JNK pathway, which was found to be altered and was found to be, and this alteration was found to be associated with changes in the cadmium-induced cholinergic alterations. And what we found interesting was that simultaneous treatment with quercetin in cadmium-exposed rats was found to protect these cadmium-induced cholinergic alterations. This was the first evidence provided by us that quercetin has the ability to protect cadmium-induced cognitive deficits, which are normally associated by the cholinergic alterations. So, continuing with the leads, we further wanted to see whether this is only the cholinergic alterations which are involved in the cadmium or the NMDA receptor, because NMDA receptor, if we talk about, this is one of the ionotropic in nature, and it is gated by glutamate and glycine, and it's a heteromeric complex of 4 subunits which are differentially distributed in the brain, and especially in the hippocampus, all the 3 subunits are present, and it has a lot of potential to modulate the learning and memory associated with LTP and LTD. I will not go into those details, but here we wanted to see whether this is only the cholinergic alteration or whether this NMDA receptor is also involved, and in case it is involved, what are the subunits which are altered on exposure to cadmium? And based on that, we further wanted, our interest was to see the effect on the postsynaptic signaling. So what we find here, that exposure of rats to cadmium disrupted the function of the NR1 and NR2A subunits, and this alteration in the NR1 and NR2 subunits of NMDA receptor disrupted the postsynaptic signaling associated with this is PSD95, which anchors the NMDA receptor. Here the integrity of the PSD95 was also found to be disrupted, which further altered the CAMK2A signaling, and ultimately the level of the TAP, as I had mentioned in the beginning. Because of the time constraints, I will not go into the details. And further, our interest was to see whether this ARE signaling is also involved, because cadmium enhances the free radical generation and also impairs the antioxidant defense. Therefore, enhanced oxidative stress is considered to be one of the prominent mechanisms in the cadmium-induced neurotoxicity. Here we wanted to see what is the fate of the ARE signaling. So what we found here, that this is the NRF2 and also the HO1 signaling was found to be altered, and the key one, as I had mentioned, this is one of the important mediator, was also found to be increased because it anchors all these effects. And based on that, we could provide an interesting evidence that cadmium-induced alteration is the NMDA receptor associated with the postsynaptic signaling are due to changes in the ARE signaling. And simultaneous treatment with quercetin was found to protect by modulating the ARE signaling, where we could find that the cadmium-induced cognitive deficits were very well protected on simultaneous treatment with quercetin. And these changes further, we also found that quercetin was found to upregulate the levels of the BDNF, which is associated with the TRK beta, where it was found to modulate the BDNF TRK beta signaling also. So once we found that cadmium disrupts the NMDA receptor signaling, our further interest was to understand whether cadmium also affects the step, which is considered to be one of the important enzymes and modulates in the modulation of the cognitive pathway. And alteration in the NMDA receptor modulates step through a calcineurin PP1 alpha pathway. So here we further wanted to see whether this is the effect only on the NMDA receptors or is through the step signaling, where we found that step 61 was found to be upregulated, which is also very well reported in many of the neurodegenerative diseases. And in case of cadmium-induced on exposure to cadmium, we found that an upregulation of the step 61, which was further found to be modulated, and these changes are well protected by quercetin. This is another facet of the study where because in case of cadmium exposure, tonic exposure to cadmium also causes the movement disorders, those kind of changes which are normally visible during the movement disorder. And there are a lot of reports where people have shown that cadmium also causes the Parkinson-like symptoms. So where we wanted to, and our interest was to further understand whether this is the nigrostriatal pathway is also affected on exposure to cadmium. And so based on that hypothesis, we further developed another model where we expose the animal to cadmium, and we wanted to see whether disruption in the cadmium-induced dopaminergic signaling, what are the candidates which are modulated and the targets which are normally associated in the cadmium-induced dopaminergic dysfunctions, and if at all these changes are there, whether quercetin has the potential to protect that or not. So what we found here, that the cadmium disrupts the presynaptic signaling, where the tyrosine hydroxylase, which is one of the important enzyme involved in the synthesis of dopamine, and this also was found to disrupt the DA-D2 receptor type, and there was no change in the DA-D1 receptor. And this DA-D2 receptor mediated changes were found to affect both the canonical and the non-canonical signaling, and these changes were very well associated with the cadmium-induced motor deficits in preclinical models. Especially, we carried out these studies in the rats, and simultaneous treatment with quercetin was found to protect these cadmium-induced alterations in the dopaminergic signal. I've come across with an important study where it has been very well reported that quercetin modulates the ERK, CREP, and BDNF signaling. It also alters the NMDA receptor and AMPK signaling, NRF2 ARE signaling, and it also modulates the GNK and DA signaling by involving important targets therein. So based on these clinical studies, there are a lot of clinical trials are being carried out in the case of AD patients and also in the case of Parkinson's disease, where the early stage AD patients, this is one of the studies that I have come across while I was searching the data also, where cadmium has been found to improve memory recall in case of AD, and also it has been found effective in the case of PD patients. In spite of the fact we have a lot of, the quercetin has a lot of potential to modulate the activity, but there are certain challenges because the poor solubility is still one of the important factor, and it has also a short biological half-life, and it has low bioavailability and poor permeability and chemical instability. And for this, we are using the nanoquercetin now in order to compare and to further understand whether we are able to get through or not. And one of the other challenge is also there that we are working on it. We have been able to get the protective changes, and also we have been able to demonstrate that these are the targets which are very well modulated and regulated by quercetin. But whether quercetin will be effective as a therapeutic agent or not, still we are working on it. We have got good leads by carrying out the in vitro studies, but still we have to prove it now. Because of the constraints of the time, I have not shown the data as such. I have shown the pathway that has been deciphered by my colleagues, especially these, because I have learned a lot from my own students, and the work that I have shared here, especially on the cholinergic signaling and also on the NMDA receptor, Anugya is working there, and the dopaminergic signaling, the work was carried out by Richa. Richa is again a faculty at NIMS University, and Anima is working to understand the therapeutic potential of quercetin. With these words, I thank you all for patient hearing. Over to Dr. K.P. Mishra.
Thank you so much, sir, for enlightening us how the cadmium causes the neurodegeneration or causes the effect on our neurons, and how the quercetin is able to protect us from the cadmium toxicity. With this, thank you so much, sir. May I now request you to please call upon another speaker?
Okay. I think the next speaker is Dr. Hemant Kumar. Dr. Hemant Kumar.
Dr. Saurabh Srivastava.
I think Dr. Saurabh will not be with us. That's what I have come to know, that he is not keeping well. So probably he will not be with us for this symposium.
Okay, so with your permission, may I introduce Mr. Hemant, Dr.
Sure, sir.
Hemant Kumar?
Sure, sir. Please go ahead. Please go.
Okay, sir. So Dr. Hemant is going to deliver a talk on spinal cord injury, animal model, pathophysiology, and treatment approaches. So Dr. Hemant Kumar, may I request you to please upload your slides?
Yes, I will share my slides now. Okay, so is it visible now?
Yes, please make it in presentation
Yeah,
mode.
yeah, sure. Yeah. Is it okay now?
Yeah.
So
It's okay.
good afternoon, everyone. Yeah.
Well, just to introduce you to begin with, Dr. Hemant Kumar is presently assistant professor at the Department of Pharmacology, Naipur, Ahmedabad. Dr. Kumar did B.Pharm and B.Pharm from Manipal University and M.Pharm from BIT Mesra. He has number of publications, more than 50 publications he has published, and he has total citations 2,616, and H index is of 26. And he has been awarded with the various awards like Korea Research Fellowship for postdoctoral research, Brain Korea Award, and Best Paper Award from Biospine Asia Pacific. So with this brief introduction, and now I request Dr. Hemant Kumar to please deliver your lecture.
Thank you so much, Professor Mishra. And am I audible?
Yes.
Okay.
Go
okay.
ahead. You are very much audible.
Yeah, thank you so much. So today I am going to present a brief overview about spinal cord injury and what are the present animal model and treatment approaches. I will not share many of my data because that will become too long. So I just briefly discuss about the pathophysiology and what is spinal cord injury. So spinal cord injury and why it is relevant, because if you see most of the motor accident or vehicle accidents had some kind of injury, it may lead to spinal cord injury. It can be complete spinal cord injury or incomplete spinal cord injury. So depending on the location of the spine, we can have quadriplegia, paraplegia, hemiplegia. So most of the spinal cord injury occurs because of the accidents. Apart from accidents, sometimes the sports injury or like normal falls, mountain fall, or sometimes fight with someone can also lead to spinal cord injury. So the coming to the brief pathophysiology, so you can see a small kind of spinal cord here, which I illustrated here. So basically what happened during the acute phase of spinal cord injury, there is a infiltration of neutrophils at the injured area, and this infiltration will cause lot of inflammation by the release of cytokines and chemokines, which will further cause disruption of blood spinal cord barrier, that is BSCB. So blood spinal cord barrier is disrupted, which will further cause more inflammation, more recruitment of cytokine. And finally, what will happen, if you see here, the red phase is inflammatory phase, which will be subside by 7 to 10 days. And after the inflammatory phase is gone, there will be recruitment of microglia or astroglia, which will be causing a kind of scar formation. Scar will be formed at the injured area. So basically, scar is a mainly a dual sword. Sometimes it acts as a protective, but majorly it inhibit the neuronal transmission. So basically, the injured area and below injured area, you can see all kind of paralysis, which is directly linked with the glial scar. So currently, the one of the important aspect which clinicians do, they used to do decompression and stabilization of the spinal cord immediately after the injury. One of the drug, so we have to focus here that most important aspect is only one drug is FDA approved, that is methylprednisolone. We don't have any further approved drug for this particular treatment, and methylprednisolone should be given within 3 hours to after injury, and it should not be delayed after 8 hours. So the window frame is very low. So if the patient had any kind of accident, a road accident, it should be brought to the hospital within 3 to 8 hours. And after that, methylprednisolone does not work. Second problem with the methylprednisolone is that it have lot of serious side effects. Apart from drug approaches, there are certain approaches which is used in clinical setting. One is that hypothermia. So patient is kept under hypothermia for 2 days, so that the inflammatory phase is subside. Apart from hypothermia approach, there are certain other approaches which are done in hospitals, which include the hospital care by taking the bladder control, because the spinal cord injury cannot have normal bladder, so they cannot go to urine by themselves. So it is taken care by the hospital. Then there are some kind of rehabilitation therapy and some other kind of medication like painkiller, because they will have lot of pain. So there are a lot of challenges with the treatment approach in spinal cord injury. Currently, there is no treatment for spinal cord injury. There are a lot of research which is going with the stem cells, and there are some other newer technologies are coming to the picture. When I say newer technology, it is mostly the electrical stimulation of spinal cord. So this is currently undergoing in different places. Now coming to different animal model of spinal cord injury, we have mainly 4 animal model we can broadly classify. So the first and second, contusion and compression, they are more reliable model because they directly can be correlated in the clinical setting. Whenever someone had any kind of injury by the road accident, it will have some kind of contusion or compression of the spinal cord. So these are more clinically relevant model, contusion and compression. So what we used to do in this kind of model, we open the spinal cord at a specific area, either at cervical, thoracic, depend on the where we want to create the animal model. So we open the spinal cord, we open the lamina, we do laminectomy, and then finally we had to use some kind of impactor. So depending on the weight of the impactor, we can use 5 gram, 10 gram, 20 gram. We can create injury graded like severe, mild, moderate kind of injury in the animal model. The third one, which is transaction or hemisection, is usually used when we want to study some kind of biomaterial. We want to have some kind of implantation. We want to have some kind of scaffold into the spinal cord. So for these kind of testing therapeutic, we need to have some kind of animal model which are either transaction, complete transaction, or hemisection. So after we remove certain part of the spinal cord, we can implant the biomaterial on that particular area. We can also load some cells into the implant, and we can confirm whether this particular approach is working or not. Apart from this model, one of the another model is chemical induced. So this is not used a lot these days. Here what we do, we can inject some kind of glutamate or other chemicals which can cause excessive toxicity and finally cause spinal cord injury. So the contusion and compression are most reliable animal model in clinics. Now I just want to show one small picture here. So what you are seeing here is a normal animal, and after spinal cord injury, we can see that there is a drop of tail position. There is no motor coordination. There is no body support. If you see the C figure, so we can do some kind of clinical scoring in animal. And apart from that, we can do scoring. The normal scoring we can do is starting from 21, and we can reduce the number of scores at depending on the severity of injury. Also, we can do different kind of footprinting. We can mark the foot with different ink. And if you see on this side, there is sham animal. So if you see that when we keep the sham animal, it will walk and it will follow certain motor coordination, where the front limb and hind limb will be, you can easily correlate, you can correlate the stride length. But when there is a kind of injury, you have to see the paraplegia or there is a dragging of the limbs. So this is one of the another method which we use for confirming the spinal cord injury in case of animal models. So BBB scoring, footprinting, grid walk, these are the things which we can do. Apart from that, we can also perform catwalk analysis, which is a instrument which is automated instrument which will be doing all kind of activity and providing all data in the ready to use format. So till now what we have discussed is what is spinal cord injury, what are the animal model. Now what we can do about it. So first thing is fix what we have. So whatever the spinal cord which is remained after the trauma or injury, we have to prevent further damage. So we can prevent the further cell death. We can promote axon regrowth. Although this is very challenging and there is no wonderful drug, or currently there is no wonder drug which is present which can promote regeneration of the CNS neuron, but some drug can provide neuroprotection. And also we can remove the blockages. This is what we can do with the current setup. Second approach is we can use tissue engineering or biomaterial. What we can do, we can build a kind of interface at the injured area and we can provide a kind of we can include some type of cell. We can do cell therapy. We can build around that particular injured area, and we can make the continuation of the spinal cord. So there are some progress toward neuronal repair and regeneration. So important aspect is, in the case of acute injury, our main focus is we need to reduce the edema formation, and we need to control the inflammation. So anti-inflammatory and edema is one of the important aspect when we are dealing with the acute kind of setting. So that's why methylprednisolone is the only approved drug which is a strong anti-inflammatory agent, which is approved by FDA. Now, second thing is, we can rescue the further damage of the neuronal cell by preventing the apoptosis, by restoring the trophic support, which like BDNF, NT3, NT4, these kind of neurotrophic factors. We can also remove any kind of barrier which can prevent the axonal regeneration. So now when I say barrier, the barrier are mostly the fibrotic scar or the glial scar which formed after the injured area. So we can remove the barriers, which are mostly done by some kind of enzymatic method. So chondroitinase ABC is one of the important enzyme which can help in the removing the barriers. We can give a guidance, because when we were planning to make a new kind of neurons, we should make sure that newly formed neurons should have follow certain pathway, some pattern. So there is one term called neuronal patterning. So we can also do that. Apart from that, there is a kind of gene therapy. We can do transplantation, as I mentioned. So these all things can be done by stem cell or cell-based therapy. So this is very famous kind of approach which is used from last 20 years. From last 20 years, people have been trying different kind of stem cell. They have used different kind of Schwann cell, neuronal stem cell, oligodendrocyte cell, different kind of cell they have used. But the main problem is that they have some kind of in clinical setting, they are not currently have any kind of safety profile. Or second thing is, most of the cell which are present in the initial phase, they will die because of the inflammation. So this is one of the challenge with the stem cell therapy. So basically, preclinical level suggests that they may provide neuroprotection, they may provide immunomodulation, they may also promote regeneration in preclinical setting, and also help in myelin regeneration. But in actual clinical setting, the main problem is majority of the injected cell, they wash out, and also there is a strong inflammation which is present at the injured area, so they cannot survive for longer period of time. Third problem is the immune responses. There are chances of immune rejection. Sometimes we have to use the immunosuppressant, so which finally weaken the immune system. So also, as I mentioned, the after a spinal cord injury, the microenvironment is very dynamic. It changes very fast from acute inflammation to subacute, then chronic phase. It will have lot of component like inflammation, gliosis, microgliosis, astrogliosis. Then you have glial scar, fibrotic scar, secondary damage. So this is one of the challenges with the current spinal cord cell transplantation therapy. So one of the recent approach which people are trying is cell-free regenerative medicine. So what they are doing, they are taking the exosomes which contain all positive things from stem cell, but they are not having any kind of problem like immune rejection. So what we can do, we can take the all major DNA, RNA, microRNA from the parent cell, and we can make them into the spinal cord, inject them into the spinal cord. So this is one of the paper which we have published in ACS Nano, having impact factor of around 12. So what we did here, we used a magnet guided approach. We have done spinal cord injury in rat mouse model, and what we did, we have also used a magnet, the small magnet what you are seeing here, and then we have injected the exosomes. So we keep this magnet for 24 hours. After 24 hours, we remove this magnet. The initial 24 hours serve as a like a guidance. So this particular kind of magnet help in the taking of the like iron oxide nanoparticle. So this we have a kind of setup where we have embedded the iron oxide nanoparticle, which were having like this kind of all mesenchymal stem cells, so exosomes. So when we injected, the magnet guided them, and most of them were present at the like injured area. So this is one of the finding which we have published in ACS Nano. Apart from that, currently what my major research involved is I am trying to focus on the microtubule dynamics and neuron endothelial cell crosstalk, and also I am focusing on regeneration. So we have shown different kind of approaches in recent last 3 to 4 years, and we have found out some role of some ion channel like TRPV4, which is like main ion channel, which is mainly linked with the osmo sensor as well as mechanical sensor. So whenever there is a kind of mechanical injury, as we see in spinal cord injury, this particular ion channel like TRPV4 is upregulated. So we have already published this in Journal of Neuroscience, so one of the good journal in neuroscience field. So we have shown that TRPV4 is also upregulated after spinal cord injury, and it was mainly localized in the endothelial cells and glial cell network. Now also what we are doing is we are trying to do some kind of targeting approach, where we are using some kind of labeled probe. After any kind of injury, what we are doing, we are injecting them into the mouse, and we can confirm whether after injury, how much is the damage, how much is the damage in the endothelial cell, how much is the damage in the other neuronal cell, or is there any kind of inflammatory changes which is having in the real-time manner. So we can confirm in our setup these kind of approaches. So what I have discussed here is mostly the main clinical aspect of this injury. So with this, I just want to thank all of you to listen my talk and providing me the opportunity to present my talks. And these are my students of PhD student and MS student. And also I thank the SERB India for providing the funding and NIPER Ahmedabad. So thank you everyone for my for the patience.
Thank you, Dr. Hemant, for being in time. And it's a nice presentation on the spinal cord injury, and the methodology which you have used shown us is a great importance. And we will take questions at later when all the speakers will finish their lectures. So may I now request Dr. Avaneesh Misra to please upload your presentation. Dr. Avaneesh Misra, are you there?
Yes, sir. My voice is audible, sir?
Yes, you are audible. Please go.
Can you upload your presentation?
Yes, sir. I'm just uploading it, sir.
So, meanwhile, I am just introducing the speaker, Dr. Misra, Avaneesh Misra. Dr. Avaneesh Misra has earned his master's pharmacy degree with the specialization in pharmacology from Guru Nanak Dev University, Amritsar in year 2008. He joined Faculty of Medicine, Department of Pharmaceutical Sciences and Drug Research, Punjab University, Patiala, and earned his PhD degree. Thereafter, he served as assistant professor in Department of Pharmacology and Toxicology at NIPER, Raebareli. Now he is currently working as assistant professor in Department of Pharmacology and Toxicology at NIPER, Guwahati. His major areas of interest are neuroscience, nanomedicines, and nanotoxicology. He is focused on the understanding the molecular mechanism and development of neurological diseases like epilepsy, Alzheimer's, and Parkinson's disease, and associated psychiatric comorbidities. He has added interest in how groundwater pollutants like arsenic, iron, copper, fluoride, and insecticides, especially organophosphates, are causing neurotoxicity, and how natural bioactive molecules are useful in mitigating the same. In addition, he is also working on utilizing nanomedicine and neurological disorders and evaluation of toxicological aspects of nanomedicines. He has contributed more than 45 publications, 10 book chapters, and filed 3 patents. Now, may I request Dr. Avanish Misra to please share your presentation?
Yes, sir. I'm facing a little difficulty in sharing the screen. Will it be possible if I send my presentation to the organizing committee?
Yes, sir. भेज दीजिए, sir. Please.
Okay.
डॉक्टर खत्री मुझे भेज दीजिए, मैं करूँगा.
आप भेज दीजिए सर. अभी तो रसा टाइम पे है हम लोग, मिश्रा जी.
Sir, we are in the allotted time.
Right, sir.
Slot.
जी जी. तो जैसे ही प्रेजेंटेशन भेजते हैं ना सब तो देख लेते हैं, because we like to end up the session by 3:45 also.
Yes, sir. Once I receive, I'll update you, sir. I'll share from you.
Dr. Hemal, it was a nice presentation. I'm really impressed the way you presented your findings.
Thank you, sir. Thank you so much.
Please keep it up.
Thank you, sir.
May I request delegates to send your questions in chat box if you have any query?
Dr. Khatri, I have sent you. Please check.
I'll just do it.
Dr. Khatri, have you received the presentation?
Yes, I received. I'm just opening, sir.
Fine, sir. Not a problem.
I think there is some question in the chat box. Will you take it?
Oh, yes, thank you. Thank you, Dr. Khatri. Shall I start?
Please go ahead.
Okay, thank you. First of all, I would like to thank the FIPS team for organizing such a wonderful event where we can showcase our findings. And I would like to thank chairs who have given the wonderful introduction for myself. And my talk of today will be emerging role of flavonoids in the neurodegenerative diseases and what are the challenges and opportunities are there prevailing that we'll be discussing now. So just to start with the brief introduction of flavonoids. Okay, so these flavonoids are derived from the Greek term flavus, which means yellow color. And basically they are polyphenolic compounds and secondary metabolites from the plants. And they're having diverse chemical structures. And till date, more than 4,000 flavonoids has been identified. And if we see around they are ubiquitously distributed throughout the plants. Many of the plant parts will be available with these flavonoids, and they are also important. They are very important because of their intramedical values, diversified from respiratory, cardiovascular, neurological disorders are all one of the emerging areas where they have shown the wide variety of therapeutic importance. If next slide, please. If we see the chemistry of these flavonoids, basically they are C6, C3, and C6 moiety, and basically six categories of these flavonoids has been classified, which is flavonol, flavontriol, anthocyanins, flavonols, flavonones, isoflavones. So basically, these are different categories, and based upon different plant products, we will be getting these. And please next. Basically, if you see the chemistry here, the one splitted ring is there, which is on the C side. We can see that two separations are there for the basic flavonoids. They will be giving us two different domains in the UV visible spectrometry. So the first benzoyl band will be appeared at 258, and at 370 nanometer, we will be identifying the cinnamyl group. Just to take an example, just previous one, please. Just to take an example, I will be just going to give a brief of chrysin, which is one of the major constituent of honey, propolis. And if you see the chemistry of this molecule, they are having basic antioxidant and anti-inflammatory properties. These hydroxyl and this ketone group and diene group is very important for their anti-inflammatory and antioxidant properties. A part of it, most of these biological activities are going to be important by these. And they're also one of the important in case of chelating heavy metals by these groups. Next, please. Once we take an example of chrysin, how they are being useful in the case of Parkinson disease, a part of antioxidant, there is anti-inflammatory properties of chrysin has been derived. And this study is representing the zebrafish model where 6-OSDA is going to induce dopaminergic neurotoxicity. And if we see here on the left side, we are just going to see the chrysin, different concentration has been exposed, and they are upregulating the NRF2 expression in the cytosol and nucleus domain. So here we can see that their expression is elevated, and the downstream pathway is getting affected by the HO1 heme oxygenase pathways. So in nutshell, if we see around a part of this antioxidant, this anti-inflammatory role is very important. So here in the one graph, we can see that how the representation of chrysin is there, and how they are going to reduce the free radicals, reactive oxygen species, reactive nitrogen species, and their activation of NRF2 pathway, as well as they are promoting the NRF2 pathway, and they are inhibiting the NF-kappa B pathway, which is going to inhibit the release of these pro-inflammatory cytokines, which is going to produce the further cytokine storm and worsen the neurodegeneration cases. And in these effective pathways, we can see that HO1 is one of the pathway which is going to help additionally by producing the biliverdin and the carbon monoxides, and they are going to add one additive antioxidant properties, and they are going to tackle up the NF-kappa B pathway, which further inhibits the neuroinflammation pathway. So the next and in these cases, if we summarize what the chrysin is there and how they are affecting the different neurological conditions, I'll just start from the down. They have been identified from the like honey, and when we see that the GABA receptor at downward, we have seen that these flavonoids, basically, they are going to activate the GABA receptors, and they're enhancing the GABA release and chloride ion conductance across the membrane, and that helps in the repolarization of calcium channels as well as NMDA receptors. So that in turns inhibit the excitotoxicity, and that prevents the seizures. A part of it, this excitotoxicity leads to the neuronal degeneration, which will be going to worsen the neurological conditions. A part of it, one of the intermediate steps, activation of apoptosomes and inflammasomes are the key steps, which is going to be inhibited by the chrysin, and they are going to inhibit the development of Alzheimer's and Parkinson's disease. This is the nut and shell. Various literature we are going to identify in the literature, but again, the major problem is no clinical study is there to support these findings that why these chrysin is not effective in the clinical conditions. So the next slide, please. So we have identified that there are major challenges in the clinical translations. That could be the first one is please click next. So the first one is the solubility term, solubility parameter. Why the solubility? Because these just one by one. Because these as our chairperson, Dr. Khanna, has also mentioned that these molecules are having a very less solubility in the aqueous solution. So that's one of the major limitations. We can see that here it has been summarized that rutin, isoquercetin, quercetin, chrysin, they are having different solubility status. So that's one of the major problem that they are not entering into the body system. So once we are just going to consume with the oral route, they are undergoing the different pathways, the intestine, the liver first pass effect, and they're being cleared through the kidney, and they're being metabolized by the liver, and they're eliminated ultimately from the GI tract. So once we just see that what are the metabolism of this chrysin, please click next. So the major metabolic pathways are with the glucuronosyl transferase enzyme and sulfotransferase enzymes. They are going to give the glucuronide conjugates and sulfate conjugates of chrysin. So these molecules are metabolizing so fast that what we can say that their plasma levels are difficult to identify, and just for the brain it will be difficult for analysis. So majorly we are being dependent on these two analysis of these two glucuronide conjugates and sulfate conjugates. And a part of this, what we can say, this hydroxylated chrysin is also being appeared in the systemic circulations of, after the metabolism with the CYP3A4 and CYP2D6, and they will be giving the different base for the glucuronide conjugates. So overall, if we see, please click next. Overall, if we see the next, that even after the, please go back, yes, thank you. If we see that how after even after absorption, or why the results are sometimes inconsistent with the chrysin, that they are also being a substrate for the efflux proteins. Here we are discussing about only two proteins, multi-drug resistant proteins and breast cancer resistance protein. These resistant proteins are basically going to efflux the molecules from the cell, and they are effluxing them outside. On the first top corner, left top corner, we can see that intestinal epithelial cells are there, where these glucuronide conjugates and sulfate conjugates are being effluxed by the MRP2 and BCRP proteins, and they are just excreted into the lumen, and which will be just going to be washed away from the stool. And if we see in the kidney proximal tubules, here also from the blood, these glucuronide conjugates enter into the proximal tubule cells, basically the epithelial cells, and with the help of MRP2 and MRP4, they will be excreted outside in the urine. And if we see in the hepatocytes, again, here MRP2 and BCRP, they are playing major role in the biliary secretion of these glucuronide and sulfate conjugates of chrysin. Further importantly, if we see around the blood-brain barrier, here also, whatever the metabolites have reached to the brain, they will be also getting excreted by the breast cancer resistant protein and this multi-drug resistant protein 4. Especially here, we can see that this MRP4 is being expressed in the brain, and where it will be going to help in the efflux of this. So majorly a part of being absorption, one is the elimination, and there also being a participation in the efflux. And upon long-term exposure of this chrysin, they are also going to induce the levels of these glucuronosyl transferase enzyme, which induces their own metabolism. So this tolerance will be also getting developed with the continuous administration. So overall bioavailability, if we see about the chrysin, here it is ranging from 0.003 to 0.02%. That is very less. And even if you see the brain bioavailability, that is very poor. And if you see about the plasma binding status, that is more than 99% of the this chrysin is binding to the plasma proteins. So that is why the distribution is majorly limited to the plasma, not to the brain. Next, please. Yes, please next. Click on next, it will be just coming up. So overall, if you see that whatever the limitations are there with the chrysin is the lower solubility, first pass effect, and higher metabolic rate, poor blood-brain barrier permeability, and efflux proteins. So let us see that what would be the strategies we can explore further for the overcoming these effects. So the first of all, the synthetic modifications of these could be possible. While protecting these hydroxyl group, we can put some structural activity relationship and combinatorial chemistry. We can develop some new drugs, and we can also propose development of some prodrugs which can protect these functional groups, and they can deliver towards the brain targeted. A part of it, these will be just going to protect from the first of few of the effects, like first pass effect, higher metabolism. These could be going to be protected by the synthetic modification and pro drug approach, while the nanoformulation approach could be beneficial, and that could be going to be like in the next domain, will be coming up with the nano applications of these flavonoids. Please next. So while expecting the nano formulations, we will be just going to see around what nano formulations we can think of and what the advantages they will be giving us. So the first limitation is the poor solubility. By the help of nanoformulations, we can go ahead with the improving the solubility index, and by increasing the permeation, by the use of these liposomes and different carriers, or different targeted nanoformulations we can develop, which can enhance this. And while protecting them in the outer covering of these nano carriers, we can protect them from first pass effect and the liver metabolism as well. And they can be targeted, they can be directly given into the they can be directly administered into the systemic circulation, and through this it could be reaching to the blood brain barrier, and they will be also having the advantages to cross blood brain barrier easily. So in this regard, we can see that liposomes, polymeric micelles, polymeric nanoparticles, nanogels, dendrimers, nanoemulsions, emulsion complex, phytosomes, and solid lipid nanoparticles, and there are various aspects which has been seen. So the current trend of research is moving towards the development of nanoformulation of these. So I would like to quote one example here, that how the 6-hydroxy dopamine induced neurotoxicity, especially the dopaminergic neurotoxicity, is being overcome by application of nanocrystals of quercetin. So here the nanocrystals of quercetin, you can expect that at the here the vehicle treatment is there, and here we can see that the quercetin, here we can see that the promising findings is observed at the 10 milligram per kg dose is observed, and which is equivalent, which is more than the curcumin bulk. So here some of the slight changes we can see, and we can limited observations are there still. Some of the nano formulations are being developed these days. So I would like to quote one example, please. Next, one example where we have done with the polyphenol nanoformulations, and we have seen that how effectively they are going for the management of the neurotoxicity. Here we have gone for the polyphenol, especially the curcumin, and we have developed the formulations with the PLJ nanoparticle. Here the PLJ nanoparticle was 50/50 was used, and here we have found that there are different batches were there, and peak size we have identified as 114.9, and please next. With the loading efficiency and PDI, these values were there. And after the in vitro release studies, we have evaluated them into the monoprotophos-induced neurotoxicity. Monoprotophos is also one of the important organophosphate, which is causing the neurotoxicity. So very much similar to the Parkinson's disease. And we have identified that lower dose of these monoprotophos is also going to produce the long-term changes, even below the NOAEL doses. So here we have exposed the animals with the monoprotophos, and we have seen that the curcumin bulk and curcumin nanoformulation, we have identified that how they are effectively going to reduce. So we have taken up here the curcumin 15 milligram dose, which is quite lesser than their therapeutic doses. Therapeutic doses has been identified at 50, 100, 200 milligram per kg dose, which has been identified. So the first image is indicating the fall of time from the rotor rod test, and that is indicating that this curcumin nanoformulation is having a better effect in the mitigating the motor impairment effect induced by the monoprotophos. And escape latency was the parameter to evaluate the passive shock avoidance test, which is indicated the contextual fear memory. So even the contextual fear memory was being improved in the case of curcumin nanoformulation treatment. And further, we have analyzed the plasma acetylcholinesterase level and brain acetylcholinesterase level, and we have found the significant improvement with the curcumin nanoparticle formulations. And further, a part of it, we have identified the antioxidant status, which was significantly improved by the curcumin nanoformulations. Please, yes. In the next one, we have seen the intracellular calcium level. It was very peculiar to identify that what are the ROS status, and how is the mitochondrial health is there. So in this case, we have identified the mitochondrial stress with the Fura 2M, and this calcium level, it was significantly reduced in the case of curcumin nanoformulations. So that was advantageous to us, that this nanoformulation was not only going to show the behavioral findings, but they were going to give us better values in the plasma, brain, and intracellular calcium levels as well. So the next, please. Please next. So with these, just please be back. So yes. So here still we have found that different studies are there, but coordination of these different studies are lacking. Some of the formulation people are working, but again they are lacking the preclinical studies. And hope we will be just developing these nanoformulations and effective investigations preclinical. We'll be just opening the new horizons for the these formulations to develop into the clinics. And we expect that further advancements or further future prospects could be also developed in this regard. And in this case, we are also developing some nanoformulations which are specifically designed for the nose to brain delivery, which will be bypassing all the systemic effects, as well as they will be just going to deliver the drug directly into the intranasal route, and that will be going to directly entering into the brain. So that will be minimizing the complications. But again, apart of these developments, we should be also thinking that these nanoformulations are also going to show different safety aspects. And in these regards, the size is also mattering. Size is also quite a considerable fact. If the size is lesser than 10 nanometer, then it will be quite problematic for the genotoxicity. So these safety aspects should be also going to be considered importantly, and that will be the kind of new horizons in this regard. So with these, I will be summarizing my presentation. I would like to acknowledge the support provided by the Department of Pharmaceuticals and Ministry of Chemicals and Fertilizers. And I would like to thank my director, Dr. U.S.N. Murthy, for giving me opportunity to work with him, and my students who have supported me all the way to perform these studies. So thank you very much.
Much, sir, for being on time. May I request Professor Khanna to please proceed for the next presentation?
Sure. Thank you, Dr. Mishra. And we have another gem from Naipur, Hyderabad. He is Dr. Dharmendra Khatri. And Dr. Dharmendra Khatri is the work, as far as his work is concerned, he is known by many of the people. He needs no introduction. But for the sake of formality, I'll just mention about his contributions in the area of Parkinson disease. That is his basic research of interest. And he has been the recipient of many of the awards. To mention few of them, he has been designated as Indian Ambassador on behalf of International Society of Neurochemistry for the year 2018 to 19. He has been the recipient of Lalita Ravindranath International Travel Award by Indian Academy of Neurosciences in the year 2019. And he also has been the recipient of Next Generation Young Neuroscientist for Basic Science Summer School that was held at Osaka University, Japan in 2018. He has extensively contributed in the area and has published extensively. And he also has 7 patents to his credit, and also extensively contributed in various societies where he has been the member. With this brief introduction, I'll request Dr. Dharmendra Khatri to share his presentation now. Dr. Khatri, please. Dr. Khatri?
Okay, sir, can you hear me?
Yes.
Thank you very much. Thank you very much, Dr. Khanna and the KP Mishra, sir, the senior most pioneer in the field of neurodegeneration. And I always salute you guys because we are following your footpath to be on the stage where you are in the future. Thank you very much. And I'll be again thanking the the Fips conference for giving this platform to share some of my research, which we are doing right now at Hyderabad. And I will give you some of the brief overview about the Parkinson disease, and later on I'll share some of my recent research. Okay, guys. Okay, so various models which we do studies in case of any neurodegenerative diseases majorly fall under the in vitro and in vivo capacity. If you look at the in vivo types, we do have several options to choose from the in vivo, where we have C. elegans, zebrafish, we have rodents also, and we have higher vertebrates also. The most preferred choice remains for the preclinical research is rodent, and the reason why not others and why the rodents, we knew that the whole genome has been sequenced for the rats, which has been identified more than 26,000 genes, and they are being shared amongst the humans about 70% of their genomes, and that has become the choice for the experimenters as a rodents model for the preclinical research. So what has given the advantages of the rodents to consider in the biomedical research, because of their being most preferred, among them again the mice having the better molecular techniques, and we can be able to manipulate their genomes easily. In case of rats, yes, they can be extensively studied for the various pharmacological areas, including cardiovascular, tox, and behavioral studies. Being more social and mimicking the behavior of the human, the rats are being more preferred in case of biomedical research. As far as the higher vertebrates is concerned, the chimpanzee is sharing nearly about 98% of their genomes, while the mice and rats shares more than 90% of human genome. So this has clearly been studied that the genome is matching with the human, and they're being perfect. So what purpose we are doing those animals, especially in neurobehavioral disorder, in order to understand the brain-behavioral relationship and the underlying mechanism which controls the normal and abnormal behavior. And eventually we go for the translations of these preclinical study to find out in the clinical settings of the new targets, new pathways, mechanisms underlying the neuroprotectives, anti-degenerative or rehabilitating or supporting those studies, mental health promoting, and various other areas, especially in safety, teratology, and neurotox studies being done on these animals. So there are few factors which actually, influence, I would say, that in animal experiments, especially in neurobehaviorals. This belongs to trait factors and technical factors. So trait factors belongs to the experience that the experimenter has, the time at which we perform those experiments, because chronopharmacology has a great influence on those behavior parameters. The strain, gender, and the age of the animals is greatly influenced the behavioral parameters, and the developmental varying, housing, social effects is also contributing. While at the other hand, technical factors include the state and the health of those animals, the test conditions, the previous test experience, the level of measurements, what we do, whether it's normal, ordinal, internal ratios, and the level of automations, because nowadays, whatever the experiments that we do in case of the neurobehavioral studies, we have a very advanced techniques to monitor those parameters. So up to what level we use those in order to avoid the error in those experiments. So these are the few comparative statements where you can see that in case of all the models which we do in case of neurobehaviorals, what are the factors that will influence sex, strain, age, housing, handling, environment, and confounding behavior. In case of transgenic model, of course, the gene construct, the penetrance, the constitutive and inducible, whether the choice of promoter is right or not, what kind of pathological phenotype they will produce, also the behaviors, and what kind of mutation they will going to produce. And one of the very interesting, the techniques which I do have a lysine model in case, what is the surgical skill that person or the experimenter has, and what kind of toxin which we are choosing, and what rate we are delivering those toxins, and up to what extent they will going to cause the damage to those specific area that we are targeting. And what would be the recovery and the post-operative cares, the infections and post-operative care anesthetic which we are using. So I'll be just focusing on lesion models, how the surgical skills and the is influencing, because as said by one of the person, that the hand who performs the surgical procedure have the magic in their hand. So everybody cannot be a surgeon. So you require a great skill in order to perform those kind of surgical procedures. The choice of toxin is also again depends on the what is your end point of your study, because each toxin which is being used for, especially in Parkinson's disease of MPTP, we have rotenone, we have paraquats, we have 6-OHDA. All of them are affecting at the different mechanisms. So it depends upon the endpoints, and it depends upon the aim and objective of the study which you are going to undertake. So we have to choose the toxin accordingly. That what is your area of affecting. So the dose and speed is again the very important factors, because in case of the PD model of the I would say that ICV injections, in which you have to limit the rate of the drug at the very, I would say 4 microliter per minute. So it is quite difficult. So you have to choose accordingly what is the drug which you are using, whether it is of high molecular weight or low molecular weight, and accordingly we have to choose the speed or the dose of the drugs to be delivered into the brain, especially in case of ICV injections, and then how these toxins is going to damage your target populations of the cells, whether you wanted to target the acute or you want to target the progressive dopamine cell loss. So it depends upon the endpoints and the objectives that we set in order to induce the model. And then we have a recovery environment, infections, and post-operative anesthetics. Is all in order to have your the models to stay alive at least to send it, because the surgery involves a very chronic procedure. So you need to acclimatize them. You have to give the overnight fasting, and then you give the very specific amount of the anesthesia, and then you have to take care after the post-operative care. You have to give the painkillers, and then you have to give them the normal drinking water, food pellets. These are all things just to be taken care while performing or doing those. So talking about the more widely used PD models includes toxins and transgenic, which is very well established so far, including 6-OHDA with the toxins, MPTP, rotenone. In case of transgenic, we have alpha-synuclein, LARC, Parkin, DJ, and PINK2, which has been discovered so far, very well established in case of Parkinson's disease, and they do topographic depletions and sometimes mimic the pathology. So while we knew that the PD is nothing but the loss of dopamine concentration in the specific population of the brain called as substantia nigra from the dopaminergic cells, and then eventually they cause the decrease in the concentration of the synaptic cleft, and it results in the the disturbance in the smooth coordination of the movements, and that eventually leads to cause the Parkinson's disease. So the history is all known to us, and it is not very new, but it is already been given into the AD 175 as a shaking palsy, but there has been many advancement, and the new information has been added since then. And then now, if you look at the 2020, after the 2020, there has been a great interest has been devoted from the scientists in order to find out the mitochondrial DNA and genetics effects, and I would be focusing on here what we are doing right now on this. So we all knew that this Parkinson's is affecting the basal ganglia, specific part of the substantia nigra, and we look at what pathophysiology belongs to. In case of normal functioning, the cerebral cortex send the send the signals to the dorsal horn of your spinal cords, and from where it is going to control your normal movement of the skeletal muscles. But what happened in case of the Parkinson's disease, the outcome which is going from the basal ganglia to thalamus is not happening in case of Parkinson's disease, and you don't have a normal movement pattern. So this is where the D2 receptor, which is arising from the SNPC area, if you don't have enough amount of the dopamine, then this pathway will going to affect, and eventually you don't have inhibitory effect. So this is how it will bring about the abnormal movements. In case of Parkinson's, there are several factors which contribute in the neurodegenerations, not limited to one factor. There are several factors which contribute and which are well established so far that leads to the degeneration of those neurons. And these are the few symptoms, I would say the cardinal features of tremor rigidity, in which we all knew that. So looking at the assessment of the PD model in case of neuroprotective strategy, we should, or one should do a behavioral impairment study, a neurochemical deficit, and a pathological disease. These are the three aspects that one has to look in order to say that yes, this drug is showing the neuroprotective strategy. As I mentioned earlier, that there are different compounds are there which is being used, especially in order to induce the Parkinson's disease, and they have their own specific mechanisms to induce the Parkinson's disease via damaging the mitochondrial complex or proteasome inhibitions or the stress. In case of in vivo, we have a unilateral injections, we have systemic injections of those chemicals. We have terminal oxoterminal antagonists, we have reserpine antagonists. We can do the unilateral with the 6-OHDA. We have systemic injections of rotenone as well. In case of in vitro model, we have different cell cultures which we can be used in order to induce the Parkinson model. And I will just share the protocol of how to induce the rotenone, especially of IP, which is the very well standard model in our labs, where we acclimatize the animal initially, and then we consider 0 day as a baseline value, and we perform certain behavioral parameters with respect to Parkinson's disease, and then we inject the rotenone at the 0 day, and then it goes for 21 days. If it is of therapeutic approach, then you give your drug initially started with the 21 days and goes for the, it depends upon the, your duration of study, and then eventually you again compare your behavior parameter from the 0 day to 21 days, and then you depends upon the what kind of behavioral parameters you do, whether the motor coordination, skeletal muscle strength, and learning memory remains the the mainstay for the Parkinson's disease. And then you may sacrifice the animal, you can process for the biochemical and immunocytochemistry. In the another approach, if your drug is tend to be a prophylactic approach, then you can do the pre-drug treatment, and then you can, depending upon your bioavailability and solubility of the drug, you can do whether the 7 days pre-treatment, and then you can perform the another model which is called as ICV injections of MPTP and 6-OHDA, a single injections, and then you can continue your post-drug treatment for again another 14 days or 21 days. So this is another protocol that we can follow in case of Parkinson's disease, and eventually you can do both baseline value of your behavior and also the 21 days behavior. And eventually you can sacrifice them for the biochemical and anesthetic. So this is the typical stereotactic surgery protocol which I am expert in performing, which is what my lab is doing right now. And the coordinates that we set from either from the Bregman lambda, which you can follow from the Watson infections, the atlas, and then you can find out which area you wanted to inject your drug, and then accordingly you can set your AP, lateral, and the dorsoventrals. This is the one of the most commonly used tests called as open field test, in which we keep the animal into the central arena, and then we try to find out the behavior of the animal, especially the exploratory behavior, and which can be captured into the the video camera and the images. So I'm showing you the few of the recent the real-time experiments. If you see the normal animals, which remains very well explored after giving the acclimatization time, and he will try to go to the every zones, and we have captured for at least 5 minutes. And then if I show you the rotenone-induced IP model, the animal it's not much exploratory because he tend to keep into the corners. I'll show you the trajectory pattern of the complete 5 minutes. He became freeze now. If you can see that this is called as freezing behavior. So I'll show you the treatment, which is again of X drug. I want to share. This is of 1 dose, and you see this negative controls. It's positive control, rotenone freezing behavior. And then if you see the another dose level, that the animal has shown a good amount of improvement in their locomotion, I would say that. So what we do observe in case of in addition to the total time spent and exploratory behavior, we do check the rearing, we do check the grooming, and the duration of immobility. So this is the very beautiful trajectory that we observe in case of the IP model of rotenone, where you see the comparative statement of comparative trajectory of the disease control. The animal has remained freezed and did not show a good amount of movement comparing with the normal. But the treatment has shown a significant improvement, but not as much of the normal. The another one which we have the Rotarod, very well established model, which is used for the motor coordinations of skeletal muscles by keeping the animal on the revolving road at a specific speed, and we keep the cutoff time of depends on 120 seconds, and then we see the time at the animal fall down. So this is the typical model of the normal animals, and they are being trained actually, and they will remain on the revolving road for more than 120 seconds. And comparing with this is the rotenone induced, the mice model. So they're not able to remain on the revolving board. And then we have a, you see this normal one, and then we see the different dose. They could be able to remain on the revolving board for at least improvement, a significant improvement as compared to the negative control. Quite interesting. And we have another model called as Morris water maze, which is being discovered by the Richard Morris in the late 1980s, in which we have visual cues, and then we have a hidden platform, and then we have a water tank in which we keep the animals, and then we have we try to find out the time at which the animal find out the initially for the visible platform, and then we do the probe test. So these are the few technical parameters that has to be taken care while performing the Morris water test. So we do the 3 stages: visible target, the hidden targets, and the probe targets. So initially we give training for the visible targets, and then we remove these visible targets, and then we do the probe test. Talking about the recent research which is going on with respect to the therapeutics in case of clinical trial pipelines, if we could see here, just a minute. If you look at the simvastatin, which is right now we are doing with the drug development, where we have developed the in situ nanogel nose to brain delivery of simvastatin, and also one of our students is working right now on the cannabidiol, and since it is approval is still pending, but we are working on these 2 drugs recently. And this is what right now we are doing in our lab. This is what our area of research is focusing on. We knew that whenever there is a complex 1 inhibition that leads to cause the neural damage via the increase in mitochondrial permeability or damage in the mitochondrial DNA with respect to activation of astrocytes, which eventually cause the increase in NF-kappa B and leads to the development of NLRP3 inflammasome. So this inflammasome helps in the development of the pro-inflammatory mediators into the inflammatory mediators. Eventually they cause the damage to the mitochondria, and then they cause the damage to neurons. So what our interventions right now is going on in our labs, we are giving the chronic exercise to the animals with few natural bioactive compounds and targeting the molecular targets and few of the repurposed molecules. We see the molecular mechanism of activation of AMPK dependent PGC1 alpha and KEAP1, where we try to see the NRF2 activations, PPRs, and FOXO3 has become again our group of interest, and try to see that how it can produce the anti-inflammatory neuroprotective effects and also the mitochondrial biogenesis. So this is one of our recent article which is published in another model, where we try to see that how the NLP3 inflammasomes is augmenting the pathways that is what our lab is focusing on, KEAP and NRF2 activations. So in which we have used the a neuropathic pain model, where we've seen the extensive improvement in the mitochondrial quality control mechanisms. And then another recent research going on right now with respect to mitophagy and mitochondrial dysfunctions. Again, on one of our recent articles, we have tried to see that how the pink parking pathways could be able to improve the by using certain molecules, myoinositols. If you look at the the mitophagic proteins, pink and parking, they will significantly improve as compared to the negative controls, high glucose groups in case of the neural cells, and also in case of the animal as well. With this, I would like to take you through the NIPER Hyderabad. We have 11 departments, and then we are offering the M-Form in all the departments and PhD in certain departments, and we all are equipped and having expert faculties are working on different areas of molecular drug discovery, natural products and synthetic molecules, molecular modeling, high throughput screening. We have a formulation and development experts. We have a complete setup of the in vitro safety and efficacy evaluations. We have tech transfer people as well, and pharmaceutical market analysis plus pharmacovigilance people are also there. I will take you through the few of our institute, or especially in my department, we have a very good instruments called as Seahorse flux analyzer. We have flow cytometry, we have a stereotaxics, we have a cryotome, we have a ultracentrifuge, we have confocal microscopes, and we have a UV, and we have a TRF and alpha screen as well. So we are coupled with the all complete advanced instrumentations, and we welcome all anybody who wish to have some collaborative works. And finally, I would like to thank the NIPER Hyderabad for giving this opportunity, and the admissions are open right now for the NIPER J 2022. Please keep watching NIPER Hyderabad for the upcoming NIPER J 2022. Thank you everyone for patience listening, and thank you chairpersons.
I thank Dr. Dharmendra for a nice presentation, and also for the virtual tour of NIPER Hyderabad for taking us, because we really enjoyed. And moreover, the fact I must appreciate the interest the director NIPER Hyderabad has taken to establish all these facilities. And I still feel that this is not the platform to thank her, but I feel that you all are well protected and that you are in safe hands, so that you have been able to create these kind of facilities to carry out an excellent work. And I would also like to thank Dr. Dharmendra for helping Dr. Avanish for running the slides. So this is again a good help to run the session. We still have 5 to 6 minutes before I request my co-chair for the specific comments on the session. I'll just tell you that there were 5 presentations. 4 presentations were really very good. I cannot assess the 5th presentation because that was delivered by me. I do not know where I am assessed by other speakers and other participants. But definitely I must appreciate Dr. Kalpana for setting the tone of the symposium. And before I request Dr. KP Mishra for the comments on this session, I have a small question from Dr. Avanish Mishra. So if you are given the choice to choose between curcumin and quercetin, Dr. Avanish, you are here? Doctor, I think that is not connected. Fine. Dr. Dharmendra, can you tell us, like the rotenone, what dose did you use to produce?
I think it is a very well standard model, which we have used 1 mg per kg, sir.
Very nice.
IP.
Did you find any pathological changes?
Pathological changes, sir, we have tried to increase the dose from 1 to 1.5, and then we have see a great mortality. Yes, it is. If you go higher than the 1, it is showing higher mortality, sir.
So if you are given the choice to choose an animal model for Parkinson's disease among the 3, like the MPTP, 6-OHD or rotenone?
Sir, very good question. I think from the topographic depletion point of view and from the pathological, mimicking the pathology of Parkinson's disease, it all depends on the, since the Parkinson's disease is progressive disorder. So if you use 6-OHD and MPTP, they immediately cause destructive of your dopaminergic cell. So and it does not mimic your topographic depletion. So I would prefer that if we give rotenone over the period of time, that will cause the a slowly and progressive manner degeneration, which mimics the Parkinson's disease. So it depends upon what is your aim and what would be your objectives to find out. Accordingly, we can choose this model.
And now I'll request Dr. KP Mishra to please give his comments, and also to take up any question if there is any question from the participants.
Thank you so much, sir. Dr. Dharmendra has presented in a very nice way his presentations. He first shown us how the models can be used for the scientific studies, like he compared the genome like rodents and humans and chimpanzees and other invertebrates and invertebrate animals. And subsequently, he took up us to Parkinson's disease and shown us how the different types of the effects can be observed and can be treated for the purpose of the saving the life of the Parkinson's disease people. And as you know, the symposium is on nutrition, neurodegeneration and brain aging, and we have touched upon all the aspects of the quercetin is one of the flavonoids which could be important nutraceuticals. The neurodegeneration, which you have shown us, Professor Khanna itself given the talk on the cadmium-induced neurotoxicity. Dr. Kalpana has given a very good talk on how the hypobaric hypoxia induces the neurodegeneration, particularly demyelination. And the Dr. Hemant has also presented a very well presentation on the spinal cord injury, how the spinal cord injury is created. I think all the aspects of the symposium has been touched upon and very lucidly presented by all the speakers. And I think there are some questions in the chat box. With your permission, sir?
Yes, sir.
If you permit, I can read that question.
Please.
One of the delegate asked, what chemicals are used to induce the SCI in animal model, that is spinal cord injury in animal model? I think this question is posed to Dr. Hemant Kumar.
Yes.
Dr. Hemant, can you answer this question?
So basically, if you want to be specific, we can use any kind of excitotoxicity agent like glutamate kind of things. If you inject any kind of agent which can cause excitotoxicity, it can be used as an animal model of spinal cord injury. But usually these kind of chemical model are not preferred because they are not well controlled. So we prefer a kind of dynamic environment like contusion or compression, where we can control the injury setting. So coming to the question, like we can use glutamate or any kind of excitotoxicity agent to induce these kind of chemical and like on any mode of spinal cord injury. Yeah.
Thank you so much. The second question is in the chat box. In nanopolyherbal formulation, how can do bioavailability in rat? I think this is the question. How we can do the bioavailability in rat in nanopolyherbal formulations if not separate any chemical from polyherbs? I think who can answer this question?
I think this question was
Dr. Avnish.
Dr. Avanish, are you there?
English.
Dr. Dharmendra.
Sir.
You are running your slides. Now this is your turn to respond to this question, but this is in a lighter vein. I'm not not a problem. See that we'll forward this question to Dr. Avnish.
Yes,
I also had one question from him. So if given the choice to choose between the curcumin and quercetin, what is his opinion as far as the pharmacological effects and the spectrum of both the compounds? Because both have the very promising effects. Suppose one has to move forward as far as the nanoparticles are concerned. So what will be his choice? So that will definitely help us to decide to move forward. Anyway, if there are no, if Dr. Avnish, due to some reasons, is not connected here, we'll forward this
question.
Sorry, sir.
Connected. He's having some network issues, I guess.
Not a problem. Well, Dr. Dharmendra, you can forward these two questions to him so that.
I think he's joining, sir. I think he's joining. Let's see.
Because see that probably at 4 o'clock there is another.
We have a GB meeting, sir. Yes, sir.
We have another 4 minutes.
Let's see if he joins, then we will wind up, sir.
If he doesn't join, then he should be penalized to arrange the pizzas for all of us.
सेशन उनके लिए उनका टीचर है, वो कहते हमारे सर थे, इसलिए हम कुछ नहीं बोल रहे हैं।
Sir, namaskar. Namaskar. But see, this all the presentations very good. But in the interest of time, what we'll do, I'll request Dr. Dharmendra to forward these two questions to Dr. Avnish, and then we can later on.
Yes. Sorry for the interruption. I'm here. Yes, Dr. Khatri, my voice is audible.
Yes, you are audible, sir.
Yes, sir. Yes, sir. You have a few questions from the audience. I think chairpersons can again.
Oh, sorry for the inconvenience.
Dr. Mishra?
Can I read question?
Please.
Yes,
nanopolyherbal formulations. How can one do the bioavailability in rat using polyherbal formulation?
Definitely, this is a good question. First of all, the problem with the polyherbal formulation is that there is a multiple compounds. First of all, and the next problem is we are facing problem with one polyphenol, which is difficult to identify the major active metabolites, and they're very less valuable in the brain, so their exact amount is very difficult to trace. And if we talk about the polyherbal formulations, it will be difficult to trace out them. So really, and in this regard, we can use LCMS techniques, and considering their metabolites could be giving us a better result. Like I have talked about the chrysin metabolite, especially the glucuronides and sulfate. They can be quantified to determine the one particular flavonoid. So by using these metabolite and parent molecule, we can identify the level of different flavonoids, and even in the case of polyherbal formulations as well.
Thank you so much. I think one question is from Professor Khanna himself. Sir, can you ask your question directly?
Dr. Avnish, if you are given the choice to choose between curcumin and quercetin, what will be your choice?
Sir, first of all, my choice will be curcumin, because
without any bias.
Yes. Because I'll tell you that because I have just worked on the curcumin, so that's why my response will be a little bit biased towards curcumin, because I have found that there is a lot more potential. Even we have reduced the dose up to 1/10th level of whatever the therapeutic level observed in the preclinical stage. We have achieved with the nano formulations, and the safety aspects was quite promising with these molecules. So that is why we were very much promising with the curcumin.
Fine. Otherwise, see that both the compounds like the quercetin and also the curcumin, they are very strong antioxidants.
They're very strong, very strong. Yes.
But curcumin definitely has more intensity and more intense potential.
Yes.
With these words, I will once again request Dr. KP Mishra just to conclude the session.
With this, we have come to the end of the session, that is on the nutrition, aging, and brain aging, and the neurodegeneration. I would like to thank the organizers for giving me this opportunity to be the part of this session. And with this, we are closing this session, this symposium number 5, I think. And now we should move ahead for the general body meeting. Thank you all. Thank you for your patience hearing, listening, and
thank you.
Thank you, sir. Good evening, ma'am.
Good evening. Good evening.
Thank you, Professor Khanna and KP Mishra, sir, for chairing the symposium. Now we'll start with the GB meeting.
Everyone who has joined us online and offline for this GB meeting, which is part of 9th International Conference of Fips. I can see many people. There are about 97 participants who are there online with us. And Professor Chandra, you are there?
Yes, madam. Good afternoon.
Okay. Good afternoon, Professor Rajendra sir is there. Then Professor Pal, I can see. And there are many more, like EC member. I know Dr. Selvamurthy is traveling. I sort of personally even today rang up and passed a message about the GBM. So there are many people. Dr. Usha, are you there?
Yes, I'm here.
Okay,
I'm there.
Good. Okay. So I think we can start GB meeting, right?
Yes, madam. Whether our treasurer is present, treasurer, if it requires treasurer.
डाउन साहब है इसमें है इज इधर देखना
Madam, all the 97 members are participants and members of this association, or you said 97 members are there?
Sir.
You said 97 members are there.
Yeah, I can see the list 97 which is being projected.
All 97 are the members of the Fips, or they are just simply participants?
No, it seems actually they were attending the program,
yeah,
and they continue to be here. In fact,
they cannot be members for the general body meeting.
Okay.
Yeah, we need to have only the members of this association.
Sure.
5 sub associations are there, and they should be there. If they are not there, we don't have a quorum for that.
Okay, right. So I would request all the participants, attendees who are not member of SCAPE, CEPI, APPI and PSI can kindly log off for half an hour. My request to all the participants and attendees who are there online, kindly log off if you are not a member of APPI, PSI, SCAPE and CEPI.
Then we will know exactly the
Yeah, my humble request to each one of you, kindly log off if you are not a member of PSI, CEPI, APPI and SCAPE.
It is good, madam.
Okay.
Yeah, Dr. Dang has joined. Professor Dang is here.
Oh, good.
Dr. Sasiwala Singh, you can unmute. We are not hearing you. You can unmute yourself.
We are writing in the chat box also. It's my humble request. Since it's a general body meeting for the people who are part of the consortium of Fips, that is 4 associations, PSI, APPI, SCAPE and CEPI. We are writing in the chat box also. I think people are people are going, I can see the number reducing now. Yeah. I mean, give us some direction. Professor Chandra साहब बोलिए क्या करना है?
Madam, as per notice, there are a number of agendas.
नहीं, I'm talking of the number. Please, agenda तो आ जाएगा. पहले ये बताइए क्योंकि one of the concern is people who are not member of these 4 societies should not be part of this GBM.
Yes, madam, you are okay. And if anybody want to say something,
Sir, Professor Rajendran sir, now our number has reduced to 74. Can we start?
What is the total number of the members of the?
Sir, member is very high, sir. APPI itself has more than 1,000.
Okay.
PSI will have a number will run into 3,000-4,000, sir.
Oh, okay.
No, there may be members actually. They may be members of either of the organization. So that is why they are there. We can start the meeting.
Yeah, I think we can, and I do not know it. I mean, 1 or 2 if they are there. After all, we have told them, requested them. We can still go ahead with us. This is a small issue. It should not be a hurdle in conducting GBM, to my mind.
Okay.
Madam, anybody other than the 4 constituent societies join, then he will be asked his identity. If anybody asks any question, you should ask whether he is the member
Sir,
of
you
any
please
of the
as a general secretary, I will request you to please ask if someone is asking.
Okay.
Please ask. Okay.
You have done right thing. You have done right thing, and it is the compulsion that no other members other than the 4 constituent societies at this moment will not participate. Please proceed, madam. So the first.
Sir, बोलिए, General Secretary साहब, please start the meeting.
Madam, first agenda is President's welcome address.
So I welcome each one of you, especially the executive members who are there online, General Secretary, Treasurer, and all the esteemed members of the executive body, and also all the members from different society who are there with us online. A very warm welcome to the online GBM meeting, which is being organized while we are conducting 9th Fips conference, which started on day before yesterday, that is 25th of March, and today we will be concluding this conference by doing the valedictory program at 7 p.m. So very warm welcome to each one of you. And now you can go ahead, Professor Chandra. You are muted, Professor Chandra.
Yes, madam. Now it is your part. That is, you will deliver the welcome address.
Sir, welcome address is over, sir. We have just half an hour.
Okay.
There is another session pending. Let us not make it 1 hour.
Welcome address हो गया.
हाँ. Acha. Then the confirmation of the minutes of EC meeting held on 24th March 2022 in online mode at Naipar, Hyderabad. So, madam, I will go through it. I will go through it. Achha. I am discussing about the minutes that was held on 23rd March 2022 at 5 p.m. In this meeting, the following members are present. That is Dr. Shoshivala Singh, President Fips, Dr. W. Rajendra, Vice President, Professor G.K. Pal, Vice President, Dr. Usha Panjwani, Executive Committee Member, Dr. Silva Murthy, Immediate Past President Fips, Dr. Ajoy K. Dang, Treasurer Fips, myself, and Dr. S.K. Reddy, Executive Members. And Dr. K.K. Deepak was not present as he was outstation. The meeting was chaired by Dr. Shoshivala Singh, President Fips. At the outset, the President welcomed all the Executive Committee members and office bearer of Fips. After that, following agenda points were discussed.
Professor Chandra, just a request. I think this was already circulated and it was approved in the EC meeting, and maybe this can be approved. Instead of reading the whole thing, it was already approved. It was circulated and approved in the EC meeting.
Okay. Then I shall not discuss.
With the permission of President, I think we can go ahead.
Okay. Thank you. If nobody is interested, then I will not read out the writings or the proceedings or the minutes of the last EC meeting. Okay, no problem. So we can go to the next agenda. Approval of the minutes of Fips General Body held in ESIC.
Dr. General Secretary Sahab, we have to tell the GBM what about the decisions in EC meeting. So please let the GBM know, the other member know what was the decision taken. And one of the important decisions which was taken in this EC meeting was inclusion of
inclusion of association.
Yeah, please, you tell the important decision taken, and we have to get them ratified by GBM.
Madam, just I am reading.
Dr. Pal, we have to sort of tell them in nutshell and get it ratified by GBM.
Yes. No. I agree with that. The just important points can be highlighted. Yes. Not to read out everything.
Yeah.
Just I am reading the headlines. Agenda 1, confirmation of the minutes of EC meeting held on 27 January 2022. Agenda 2, confirmation of the resolution of the General Body meeting of Fips held in ESIC Medical College, Faridabad 2019. Number 2. Number 3, financial status of Fips, audited statement of Fips, and possible measure for enhancement of funds in Fips. Number 3, completed. Number 4, inclusion of Association of Physiologists of India as a new constituent society of Fips. Terms and conditions. It has already been completed. Next, number 5.
Please just hold on, General Secretary Saab. Inclusion of Association of Physiologists in India, the EC has sort of agreed to include Association of Physiology of India as a new constitutive society of Fips. And it was also decided as per the bylaws that there will be a charge of 10 rupees per member, which the society has to pay. So this was the decision. We would like the GBM, some of the members to ratify this decision.
Okay. Next one.
Sir, let some members say.
Yeah, let this is over. So we all agree for that. Okay.
Any member?
Yeah, if no objection is there, we can accept it.
There is no objection.
Yeah.
Okay.
Achha, next one is venue of the 10th Fips. It has been decided that Dr. G.K. Pal proposed to conduct the 10th Fips Congress 2024. Next one, miscellaneous. We have already discussed regarding this, wherein Dr. Dharmendra Khatri has discussed that how many keynote address, plenary lectures, symposium, oral presentation, and so on. So we have agreed on this resolution. We have no objection regarding this. Okay, please, I am going to the next agenda. President's report.
ये तो बहुत आगे पहुंच गए.
Number 4. Achha, and number 3 is approval of the minutes. Number 3, approval of the minutes of Fifth General Body held at ESIC Medical College, Hyderabad 2019. Whether there is a necessity to read out it. If you are agreed, then I can go through it.
Yeah.
No, Professor Chandra, it has already been mentioned in the EC meeting that it has been approved. And
Paul, this is a general body meeting. Even though EC members know, but general body people should have all the approval of the meetings in the EC. Then we have to convey.
I don't say that. No, you don't read out the details. But what I say, already you have read out, no? So it was there as part of this thing, no, EC minutes. So that is why if somebody asks, actually, we can go ahead. Otherwise, it is approved.
No, sir. We are the executive members are well aware regarding this, as Professor Rao has clearly mentioned. But the other members are not well aware about it.
Yeah, that's correct.
It will take 5 minutes only.
Please, Dr. Amar Chandraji, in nutshell, just tell in the abstract form. Don't read line to line. Just tell us so that people know what were the minute of that meeting.
Achha. Okay. The meeting was held in the event of 8th Congress of Fips, that is on 28 September 2019 at Hyderabad. And in that meeting, the meeting was presided by Professor G.K. Pal. And next, in that meeting, the financial position of Fips has been discussed clearly. After that, there were some proposal that G.K. Pal has raised the issue that Fips meeting has to be conducted twice in a year for better interaction and improvement of the society. This suggestion was supported by Professor K.K. Deepak and the other eminent physiologists who were present. Next one, Fips fellowship should be introduced as early as possible. Next one, it was discussed that all Fips constituent societies should organize local workshop seminar under the aegis of Fips by Fips itself. This is popularized Fips and will reach the physiologists of the country. Next one, Professor S.B. Deshpande asked about the inclusion criteria of the other physiological societies. We have discussed regarding this, that in the constitution there is provision. We have discussed and we have discussed about it. Next one, it was decided that the venue of the next Fips conference discussed. And the last one is that the 9th Fips conference, it was mentioned that one proposal from NIMHANS has been received but due to COVID situation, it has not been possible. And for that purpose, on the basis of the minutes of the executive committee meeting, we have decided to organize online meeting at NIPAR, Hyderabad, under the leadership and the director of NIPAR, President Dr. Sushibala Singh. The meeting ended with vote of thanks to the chair. So I have covered almost all the important points that has been discussed in the general body meeting held on 20th September 2019 at Hyderabad. If anybody has any objection, he can raise. Okay, there's
it's okay. It's okay.
We have no objections.
Okay, now President's report. Madam, you are requested to deliver your presidential address. Now your part, madam.
Yeah. So good afternoon and namaskar. It is my pleasure to be part of this Fips, and as you know that we have been trying to sort of organize different activities under Fips banner and trying to do our best. And in that series, the last conference was organized by ESI Faridabad in 2019. Because of the pandemic, we could not hold many activities, and also there were a lot of communication gap for many reasons, and even in terms of the Fips, the way the financial status and other things we need to improve. But this conference has, to my mind, has rejuvenated Fips and also rejuvenated all the societies. My humble request to all the society is to be very proactive and to be part of Fips, so that we are able to take it at a higher level. And I will also request that we, as a president, we have been able to organize this conference at NIPAR Hyderabad with the support of all the societies. I would also request that I would like this position to be sort of given to someone else, because I have served enough, and I'm sure there are much better people who will be able to do a better job as a Fips president. And I would like to sort of request all of you, and let us see the person. The way I am looking at this is, the person should be active, he or she should be in office, and general secretary and treasurer should be in the same city, so that we have a good functioning. Otherwise, I've seen it becomes a one-man show or one-woman show only. And I would like this Fips should to be handed over to in some safer hands, which can take it forward. This is what I would like to do. So next conference, and maybe before that, I would urge the EC member in a very systematic way, that we can form a committee or whatever and ask for nomination, so that we are able to give this consortium of 4 society of Fips to another committee, which will be doing much better job than what we have done. With this, I want to thank each one of you for all the support which I have received personally from every member. My sincere thanks to Professor Rajendra, Dr. Satyavelu Reddy, Dr. Amar Chandra, Dr. G.K. Paul, and many other, Dr. Kushal Das, who was very sincerely, helping us in organizing this conference. And all the EC members, Dr. Selvamurthy, Dr. Usha Panjwani, it was great, and not the least, Mr. Manmeet, who is always at the back, but he has helped us a lot in organizing this conference. And with this, I want to thank each one of you, and looking forward for a new committee. Thank you.
Madam, I like to mention, just next agenda is the General Secretary's report. So in this context, I like to say that in this body, there were 10 members, as Dr. Sushibala Singh as President, Dr. Rajendra, Dr. G.K. Palwar as Vice President, K.K. Deepak from APPI, Dr. Usha Panjwani from PSI, Dr. Silva Murthy, Past President, S.K. Reddy from ISCAP, Dr. Ajoy Jang from Treasurer Fips, and myself. During the COVID situation, we have conducted total 5 meetings. Of the 5 meetings, 3 were the basic meetings with the physiological societies, and other 2 meetings were with the conference arrangement. So the first agenda we have already discussed, that is confirmation of the resolution of the last EC held at ESIC Medical College, Hyderabad in September 2019. These are the common things. Number 2, current financial status. Current financial status in this respect, as Dr. Dang has mentioned, that we have completed the audit up to 2019-2020, and after that, due to COVID situation, no further action has been taken, and during the period we have no income, rather expenditure. Next agenda was that formation of new EC committee. It has been discussed. It was discussed in the first EC meeting that is held in August 2021. The proposal came that what will be the fate of this body. It has been discussed after successful organization of the Fips online meeting at Naipar under the leadership of Professor Dr. Sushibala Singh. The executive body will discuss on this point and take the decision accordingly. Next agenda was that that Fips will organize workshops, seminar, conferences, and public awareness program. The meeting was ended with, and the last one was that some discussion has been made, as we have already discussed, that if a new society, registered society, likely to include themselves in Fips, then there are certain rules and regulations as per our bylaws. This has been discussed. This was the activities of our first meeting. In the second meeting, most of the members were present, and it is one important thing that 10th member was Dr. Mohan Kumar. He was not present in any of the meetings. In that meeting, in the second meeting, that was held in 28 December 2021, wherein we have discussed about the organizational thing, that almost all the 4 member societies should active part. That is APPI, PSI, ISCAP, and SAPI should take active part. And then it has been decided about the financial position. It has been discussed about the financial status of Fips. Financial status of Fips. And in this context, it is mentioned that FOPS has claimed a huge amount of money, and we have discussed about it. And we will try to pay the payment, but the fund is not so strong by which we can do it. It was the resolution. And in addition, we have discussed that after 2 to 3 months, a public meeting will be organized. And in addition to that, Fips will start to publish its newsletter. And the respective charges for the newsletter has been given to Dr. Ajoy K. Dang, present treasurer of the society, and Dr. Usha Panjwali. Accordingly, in this context, I like to mention that the first issue of the newsletter is almost ready. It will going to publish immediately. And the last EC meeting held on 27th January 2022, wherein 6 members were present, like our president, Dr. Silvamurthy, K.K. Deepak, Usha Panjwani, G.K. Paul, and Amurthy Chandru. Wherein certain discussion has been taken. And in this context, I'd like to mention that considering the financial position or the betterment of the society, Dr. Silvamurthy has paid 1 lakh rupees as a donation or a subscription or like that in the fund. Their money has already been deposited. And mostly our president madam has requested all the respective society to advise their member to take active part. In addition to that, madam has also suggested to organize symposium to each and every society. And lastly, a 500 rupees registration fees has been charged, and most of the executive committee members and the important members or interested members paid all these things. And in this connection, I like to mention that in the meeting of the 28th February, it has been discussed that in the mid-February, a meeting will be organized under the leadership of Madam Sushivala Singh. We have organized it and we have taken the decision how nicely we will organize the same. Dr. Usha Panjani also mentioned that he is also ready to organize parallel discussion like NIPER on public awareness issue. So with this, these are the activities. That is, in summary, I can say that during the COVID period, we are not silent. We organize occasional meetings, and at least we started the meeting. First meeting started in August 2021. In the meantime, we have organized 5 meetings. 3 of the discussion of 3 of the meetings has already been narrated, and the last 2 meetings we have discussed about the how we will organize the 9th Fips conference successfully in NIPER, Hyderabad. So it is the activities of the General Secretary during the Fips conference. Next one, madam. So it is okay. If it is okay, then I am going to the next agenda.
Any comments?
Dr. Chandra.
Yes, please.
A little correction I want to make here, that we have presented the audited statement balance sheet as on 31st March 2021. So in your this address you told that 2020, actually it is 2021. Okay.
22 का तो अभी होगा बाद में.
22 will be after this 31st March.
So it is okay. So if it is completed up to 2021, then it is okay. And during the COVID period, you are not able to do any audit or account.
No, this is during the COVID period only. 2021 was also COVID.
Okay. But
this year, by 31st March, we will do it.
Okay, no problem. Okay. No problem. It is the agenda. So I will include. It will be mentioned, and I will suggest, because the write-up has already been communicated to Dr. Usha Panjwani. Usha Panjwani is also present. Dr. Usha Panjwani is also present in the meeting, and he will correct the thing. She will correct the thing. Okay. Next, the financial status of Fips audited statement. I will get the opportunity to say these things under the point 6. So discuss on this point, whatever you like. Financial status of Fips, audited statement of Fips, and possible measure for enhancement of fund of Fips account. So please discuss. I request Dr. Dung to comment on this agenda.
As I had already informed in the last EC meeting on 24th that at present we have 1,40,991 rupees in our current balance, and about 2 lakh FD we have fixed deposit. So total amount we have is 3,40,991. This is our total amount we have in Fips. And every year we are paying a professional fees of 14,000 to the CA and 3,000 we are paying for the website. So this is the total amount Fips has at present. And also Dr. Selva Murthy, he donated rupees 1 lakh, and this is including of that 1 lakh also. Okay.
Okay, if it is approved, then I can go to the next agenda.
Yeah, okay.
Okay.
Approved.
Okay.
Then the 7th agenda is the most important agenda of the Fips, because after 3 decades, a new society, that is Association of Physiologists of India, as a new constituent society of Fips, its terms and conditions. So we have discussed many times regarding the inclusion of the societies in our EC meeting, and it has been resolved unanimously in the EC meeting that this society under the presidency of Professor G.K. Pal will be included. So it is the point. So if the general body members approve it, then there is no problem for its inclusion. So any general body member has any question regarding this, they can discuss. Otherwise, it has already been included, and we have taken, we have considered it on principle that the Fifth Society of the Federation of Indian Physiological Societies will be Association of Physiologists of India. Any comments?
It's okay, sir. Continue.
Okay, thank you. It is a great inclusion after 3 decades after the inception of Fips. Next, agenda 8, venue and possible time for the 10th Fips conference. So, on principle, we have decided as Association of Physiologists of India has been included as a new society. Its president as well as the vice president of the Fips, Professor G.K. Pal, was present, and he has committed that he will organize the 10th Fips in Pondicherry. So we like to hear something from Professor G.K. Pal.
Good evening, all of you, and it is a pleasure to speak here. I would like to thank profusely our president, Professor Sasiwala Singh, our General Secretary, Professor Amar K. Chandra, and all the executive body members for agreeing to include SOP, Association of Physiologists of India, as a constituent of Federation of Indian Physiological Society. I hope that SOP will contribute in the best way possible for the growth and development of Fips at the national and international level. We have 583 members, and I'll tell you, it is actually just few years back we started the organization, but these 583 members are very active and vibrant members of the association, and we have a system of participating all the members in all the activities. That is how I know actually how our members are very active, and I'm sure this will add more dynamism to Fips. On behalf of SOP, I had proposed in the executive committee meeting, which was held 2 days before, and also in the general body. I proposed that the next Fips Congress, the 10th Congress of Fips, will be organized by me. I will be organizing it. Maybe not at Jipmer Puducherry. It could be at EMS Patna. I'll be intimating that, and I'll talk to all the executive body members, president, secretary, and by consensus, we'll find out the suitable date, time, the month and the dates of the meeting. And very soon I'll come up with the details of, not the details, a skeleton of the program. We'll make it international, we'll make it physical meeting, and I'll take everybody into confidence while finalizing the details of the program. And I welcome all of you, and I look forward the best participation from all of you in the 10th Fips Congress. Thank you all.
Thank you, Professor Pal. And according to Professor Pal, he will intimate the date and a formal skeleton of the next Fips conference likely to be held in the EMS Patna, according to him. So it is almost the last 8th numbered agenda, and 9th agenda was other business. And for the other business, if any member like to raise, he has to submit a letter 10 days earlier. As we have not, President and myself has not received any letter from any member, so there is no agenda and the meeting is ended. I am handing over the microphone to our President, Dr. Sasiwala Singh, to conclude, or like that.
Yeah. No, just one point, madam. On behalf of all of us, I would like to express the heartfelt gratitude from all the members of Fips to Professor Sasiwala Singh. As she has mentioned, the only woman power. The woman power is the best power actually. Woman power is more powerful than the manpower that you have shown, madam. You are the real example of the woman power of the country and the woman power of the physiologists of the India. And I wanted to just express my sense of heartfelt gratitude for organizing such a beautiful conference. And I wish this is minuted that this was one of the best conference that we have done so far. I have seen actually all the 3 days, so wonderful. Everything is well done. In fact, it is online meeting. Otherwise, I would have requested everybody to give a standing ovation to Professor Sasiwala Singh for conducting such a beautiful program, such an excellent program. and outstanding, I'll say. So this is just I want it to be minuted.
We can give her a big hand.
Thank you. At the end, I thank you so much.
Madam, just 1 minute. Madam, we would also like to thank the organizing secretary.
I wanted to say this.
Dr. Dharmendra Khatri and his team for a very effective, successful, and productive conference.
Right,
It was
I agree.
splendid event. So please convey our congratulations to your organizing secretary and the team members also.
Sure. So at the end, I have to say a few things. One is thank you for imposing trust in me, and thank you for strengthening my team, that we have done an excellent job. It gives us a lot of enthusiasm and a lot of motivation for us to work further. It's a really great hearing all of you, but I just want to put on record, it's a teamwork. I am the last person in the queue. Other people have taken a front seat and they have worked much harder than me. And the main champion of this conference who have organized is Dr. Dharmender, who was there almost for 2 months. They have started working 24/7, requesting speakers, requesting the various societies for proposal, inviting proposal for symposiums, for his oral presentations, for at PhD level, postdoc, and at faculty level. Beside Dr. Dharmender, who is the assistant professor in pharmacology and toxicology, we were also supported by Dr. Madan, who was also there as a faculty member. However, we have a good number of students. I can tell you, my hats off to each one of them, and they have done an excellent job. Believe it, I'm the last one. My efforts were the least. Whatever you have seen in last 3 days is the hard work of Dr. Dharmender, who has been studying this, along with all the students who are at PG and PhD. If I would like to read name of each one of them, that will give me some consolation.
Please, ma'am.
So I can read in order of what I've got. Dr. Valencia, Anika, Rahul, Manjuri, Sabia, Vaishnavi, Pushpa, Nusrat, Ankita Devi, Anuradha, Gurpreet, Rohit, Sushmita, Shruti, Deepak, and of course, above all, Khatri sir, Dharmender Khatri sir, who was the champion. I'm grateful to all of them. Believe it, I have not seen such a team before, and they have done a wonderful job, and I have no words to thank them. I have no words to express my gratitude to each one of them. I feel elated when I see all of them. So thank you so much. Thank you all my dear students and my colleague, Dr. Dharmender and Dr. Madan. And one thing I just want to tell you, because the GB and some of the members may not know, they have been able to register our 300 registrations we got. They organized 6 symposiums in different categories. We have 73 speakers in symposium, drawn not only at the national level, but at the international level. Hats off to all the team members who have done this job. And we also had an oral presentation everyday parallel session, where we had more than 250 speakers. We wanted to encourage the young researchers. So we organized this session in 3 categories. One was the young faculty, postdoc, and who are doing PhD. And we have a jury which was again constituted not only from NIPER, but drawn from other organizations also. And we will be announcing the results, the best 3 presentation in all the 3 categories during the valedictory session. Please join us in the valedictory session, which is going to be at 7 p.m. And Dr. Rao, who is Vice Chancellor, Mizoram University, has consented to be part of this session. I would request all of you to please continue logging in so that we don't lose you at 7 p.m. Please join us. That will motivate my team, and that will also motivate me to work further and work much harder. With this, I want to thank you, each one of you. Thank you. Jai Hind.
And I like to say one word about Dr. Khatri, that during the time when I contacted him, he immediately responded me. And when I contacted through email or WhatsApp, he gave response within 5 minutes. He is a very sincere person. He is very sincere faculty. Very sincere faculty.
Oh, thank you. Thank you so much.
Madam, on behalf of Tirupati people, We are congratulating you and your team. Really, you have done excellent job, man.
So he's Harminder Khatri.
We know him. We have seen him earlier. He's a very
Yes, he
young.
was delivering his talk also. Lecture. We heard
right,
his lecture.
Right.
खत्री साहब या माना आप जब किया है ना वो इतना तो देखा नहीं खूब कम देखा young you are so active very much you have well done.
Good, well done.
Well done.
You've done a great job, Dr. Khatri. Great job. Yeah.
Thank you, madam. We have done a great
Namaskar,
job.
sir. Thank you, Professor Rajendra, sir. You have been really a great support to me, I know.
No,
In rough weather, you were there.
we have not done anything, but we have done many things.
Yeah.
Thank you very much, and hope the things will be better.
Namaskar.
Thank you, madam.
Namaskar.
Okay, madam. Thank you.
Sir. అదే సరేలేండి ఆయన అవసరం లేదు అవసరం
33956814.
Oh, ये कौन है? हाँ, ये तो भैया गुस्ताव साहब।
So we will come back at 5. We will come back at 5 then, right?
Please, yes, please.
entered there.
I am there now.
You entered.
I'm inside.
Because I was not there because I'm in the different chairing the session of the young scientists.
Okay.
I'll be joining as soon as finished. I'll be joining.
Okay. Thank you. Thank you for calling. I am inside.
Good evening, Dr. Gustavo. How are you?
Hello.
I'm Shashi from Naiper,
Hello,
Hyderabad. Seeing you after a long, long time.
Yes, my dear Dr. Shasha. It is so wonderful to see you again. It
Same
feels
here, same here. How are your daughters doing? I met them, you remember?
Yes, my daughter is right now doing a PhD in University of Calgary, Canada.
Oh,
Natalia.
she's in Calgary, the coolest place on the earth.
University of Calgary, yes. A very, very distinguished university. And she is delighted to do so. It's wonderful.
Oh, great, great. And how is Bolivia now? How are things?
Very good. In fact, the COVID is practically gone. We have almost no deaths, and life is back to normal. Truly. Of course, there is no absolute certainty for the forthcoming future, but I think that it has become more of a cold type of pathology, which is the natural evolution of a pandemic, you know. So I think
right. You know, herd immunity, and they say if about 75% or 80% is vaccinated, there will be herd immunity, and then that way we may be able to mitigate this corona effect.
Yes, I think so. I think our immune systems in the end are doing a great job.
Yes. How is the weather? What is the temperature like in Bolivia now?
Oh, we are in summer, and but it rains a lot here during the summer, so it can get a little chilly, but it's pleasant. In general, it's quite pleasant.
Okay, good. So when are you visiting India next?
Oh, I'm wondering. I have to see you again. Of course.
I also want to see you again. Yeah.
You know, and by the way, I am having a coffee, and look at this.
Oh, I remember Deepa's
You
cup.
gave it to me.
Yes. Usha,
cup.
Dr. Usha, can you see? She may not be here.
From Deepa's.
Yeah, I know. I
Deepa's
remember
coffee
this
cup.
guy. I have one with me also.
Yes. And my visits, thank you, Dr. Shashi, because our visits to Deepa's were truly outstanding. The meeting we held together with the Global Hypoxia Summit and our Chronic Hypoxia Summit has been outstanding. And in fact, we were even thinking of carrying out the 8th Chronic Hypoxia Symposium this year in India with Professor
Please do.
Venkat.
I'll be more than happy. Tell me if I can support you in any way. I'll be more than happy to do that.
Yes, but what we are doing now is we have decided to carry it out in La Paz anyway, because we have many people interested in coming to visit. So you have to come. You and your team.
I will come. When is it?
This is in October the 10th to the 15th.
Okay, I will try to come. I will. Yes.
Oh, that would be wonderful. You will be fascinated. In fact, my talk today is about these experiences.
Yeah, keep me posted. So send me the details about the conference. I will try to log in and maybe try to come. Yeah.
Okay. So
Dr. Reddy and Dr. Mullur, are you there?
And Dr. Mullur will join in 2 minutes.
Okay, no problem.
Okay, so we can continue to have fun. Congratulations on this fantastic event, Dr. Shashi. You are truly an extraordinary woman scientist in India, and I have to express my admiration for you because
thank you. I am taking it as a compliment. Yeah, thank you.
It's the truth. It's the truth. You know how always women have been always downgraded in many aspects, not only in science. So your activities, what you have achieved is truly extraordinary, and you are worthy of a great respect from scientists around the world.
Thank you, sir. Thank you.
And how is Hyderabad? Is it very nice and warm now?
2 degrees daytime.
I'm sorry?
Hyderabad is warm. It is about 42 degrees Celsius in the day.
Oh, really? Very hot.
Yes.
Oh my God. Is it more than usual?
Of the year, temperature is like this only. Nothing unusual.
Okay. Okay. So it's very typical. Very good. Excellent. So nice. I'm sorry I wasn't able to join the conferences because of the difficulties of the time, you know, the time synchronization. You are having it in the middle of the night for us. Hello, Dr. Lata.
Hello,
I see you.
good evening, sir. Good evening, Professor Gustavo. How are you, sir?
Happy to be here once again.
Yes,
to see
same
you,
here,
Dr. Lata.
same here. I'm really privileged. Like again, I am interacting with you. Very honored, sir.
Yes.
Honored. Nice seeing you, sir. How
I
are you
am
doing,
the
sir?
honored one. I am
Yes,
the
sir.
honored.
Thank you. Thank you so much, sir. Thank you so much. Good evening to ma'am, Dr. Shashibala Singh, ma'am.
Namaskar. Good evening. Good evening.
Hello, ma'am. Hello.
Good evening.
Yes, ma'am. I'm from BLD, deemed to be University, Bijapur.
Yeah, I know.
Thank you, ma'am. Thank you so much. How are you doing, ma'am? It is wonderful.
I'm fine.
It's very wonderful event, ma'am. I was going through all 3 days. It was very wonderfully
Thank you
organized.
so much. Thank you. Yeah, thank you.
Yes, ma'am. Yes, ma'am. We actually wanted to join physically, but unfortunately, because of so many reasons, we couldn't join it. But very well organized, ma'am. Even though it is virtual, very well organized. Congratulations to your team, whole team. Okay. Can we begin the session now?
So, Dr. Lata, I have 30 minutes, right?
Yes,
Time?
sir. Yes, sir. Yes, sir. Yes,
Okay.
sir.
Perfect. You know, I have my talk is for 28 minutes, and I need 2 more minutes to finish a video with a video. So I will have to make sure I can share both. You know, once I finish sharing my talk, go ahead and share the video to show you
Yes.
how La Paz.
And definitely, I think organizers can help in this regard, sir.
Okay, just in case.
Yes, sir.
Hello,
Yes, sir.
Dr. Kusal, Professor Kusal Das, my dear professor.
Yeah, Professor Gustavo, nice to see you here. Please go ahead. Yeah, I just shifted from here.
Yes.
Yeah, I am Dr. Reddy, another chairperson for this plenary lecture. Okay.
Good
Hello,
evening, sir.
Dr.
Lata
Reddy.
here, sir. I think it's 5. It's 5. If organizers permit, shall we go ahead?
Yes, ma'am. Shall we start the session?
Yes, definitely.
Okay. Good evening, everyone. I will request the chairpersons of the session of plenary lecture, Dr. Lata Mulur and Dr. S.K. Reddy, to kindly take over the session ahead.
Thank you.
Thank you. Thank you so much. Thank you so much. It's my proud privilege to introduce Professor Gustavo Jubieta Caleja. Is the director and head of High Altitude Pulmonary and Pathology Institution, La Paz, Bolivia. He's graduated from Aranska, USA, and did his MD from Bolivia University, La Paz. He was a brilliant student and received several awards for his academic excellence. He pursued further his study in pulmonary medicine and physiology at IPPA La Paz and entirely dedicated himself for research in pulmonary medicine. Dr. Gustavo has published 67 peer-reviewed research articles and written 2 books and 2 book chapters. His area of research is high altitude disorders, including pulmonary and cerebral edema, mountain sickness, etc. He is one of the world's leading expert in hypoxia. He is a clinician, researcher, and postgraduate teacher and guide for HAPPI and the president of Jubieta University, La Paz. Besides medicine, Professor Gustavo is expert in software programming in various software program languages. Interestingly, he is also a skilled hardware engineer and developed many instruments used for hypoxia research. Dr. Gustavo's greatest contribution to the pulmonary medicine research are designing and constructing Chakaltia Pyramid Laboratory at 5,300-meter altitude on Chakaltia Mountain, Bolivia. He is a great science meeting organizer and soccer lover too. Apart from that, he is a physician, hypoxia researcher, and a teacher, and great father and friend also. With this brief introduction, I'll hand over the mic to the Professor Gustavo.
Thank you very much, Dr. Lata. I am profoundly honored to be present in such a distinguished and important meeting of physiology in India. India is one of the BRIC countries of the world that is going up. And the other countries have had difficulties, like Brazil, you know, with the economy and so on, and Russia, of course. So India is filled with wisdom. So I am so happy to be present, and my talk has to do with a very important topic. Behind me, you see the city of La Paz and our mountain that's next to it called Illimani. And I would start now my talk to show you what I have been doing during all this time working at high altitude to be able to show you what our experience is and what the point of view is regarding hypoxia. The title of my talk is Human Adaptation to Chronic Hyperbaric Hypoxia. Deleterious or beneficial? You see, the concept of scientists studying from sea level, studying hypoxia at high altitude, is that it is deleterious. The evolution, my slides are being seen, right? Yes?
Yes, sir, yes, it is visible, sir.
Okay, so you see the map showing the human migration out of Africa 65,000 years ago and how it migrated to through Europe, to India and Australia and on to the Americas. And of course, that's why we are labeled as only 15,000 years ago in Americas. However, adaptation is something extraordinary, and these are the mountain regions of the world. Here's where we are located in La Paz, Bolivia. Very far from India. I have been 5 times visiting India, thanks to dear professors like Professor Tupil Venkatesh, Dr. Shashi Bala, and Professor Kushal Das. Incidentally, I am a visiting professor at BLDE University. What happens is that at sea level, we have a PaO2 arterial oxygen tension of 98 millimeters of mercury, a PaCO2 of 40, and an hematocrit around 42%, which is considered normal. So this is the city of La Paz at 3,100 to 4,100 meters. It's growing more and more. We have over 2 million people living up here. And if you come up to the city of La Paz, you arrive at the airport like Professor Kushal Das has done when he came to our 7th Chronic Hypoxia Symposium with Professor Biradar. The arterial oxygen tension drops to 60. The carbon dioxide drops to 30. You see, it's 10 less. And If you go up very fast, you could have the acute mountain sickness, HAPE or Hayes diseases, high altitude pulmonary edema, high altitude cerebral edema, which can be lethal, but these are a very tiny fraction. However, if you come slowly and you take it easy and you have knowledge about how to face hypoxia, the hematocrit gradually increases over 40 days, like shown here, to a hematocrit 50, which is my normal hematocrit all my life. My father lived to be 90 years old with a 50 hematocrit. My grandmother lived to be 99, her two sisters the same, with higher hematocrits than those at sea level. So there's a remarkable adaptation to chronic hypoxia. Human beings, animals, and plants on exposure to hypoxia can have acute exposure. That's a fast exposure to hypoxia that can produce this acute mountain sickness, high altitude pulmonary edema, high altitude edema, can be severe and deadly. So that's why it creates so much fear. However, if you do it gradually, you adapt to chronic hypoxia, then there's a remarkable adaptation and normal life at high altitude. But this depends on time. That's why we have a formula of adaptation that relates adaptation is equal to the altitude and time, the time factors. So the oxygen transport mechanisms used for transporting oxygen to the tissues is something my father created called the pneumodynamic pump, which are the lungs, the hemodynamic pump, the heart, and both the respiration and circulation have respiratory rate, heart rates, tidal volume, and stroke volume, which are similar, but one pumps air and the other one pumps blood.
Sir. Your slide is not moving. Your slide is not moving.
Are we still on the first slide, sir?
Oh, no. Right now,
actually, we are able to see the first slide yet.
Oh,
there's no movement.
Should I resume share? Is that what I'm supposed to do?
Yes.
And
I think organizers can assist.
Maybe I have to stop share and restart my slides. Is that correct?
Yes, sir. Yes, sir.
Okay. I'm sorry this happened. Let's see. Okay, have I stopped sharing?
Yes, sir.
Okay, now I'm going back to start my slide again. And let me see.
Clear. Yes, sir. It's visible.
It is
okay.
Yes, sir.
Okay, so this is the pneumodynamic pump, the hemodynamic pump, and circulation. So these two pumps I was mentioning, one is an air pump, moves air in and out of the lungs. The hemodynamic pump is the heart, but pumps the liquid blood. And then we have the same mechanisms on both sides, respiratory rate, heart rate, tidal volume, and stroke volume, and circulation and ventilation together pump oxygen to the hemoglobin and onto the tissues. And based on this idea, I created this graph, which shows the laws of physics applied to physiology and high altitude. If you look, you see you have the three, we call it now the oxygen transport triad, the pneumodynamic pump.
Sir, excuse me, sir. Slide is still not changing.
Oh, no.
Yes.
Oh, but I thought you were seeing my new slide. How can we change this? Stop
No,
again?
Now we are seeing only one slide.
Okay. Do you see this slide?
Not right now.
I'll try. Okay. I'll try. That's very strange. I never had this happen, but we go on. Do you see it now? Okay, so we're going to have a special, presentation. Okay, so this is please tell me as soon as you, because I'm changing slides, and so it should be going fast because I have to show you several things. So the pneumodynamic pump, the hemodynamic pump, and hemoglobin are a triad, the oxygen transport triad. And you see, these are the laws of physics applied to physiology. It's so beautiful because you see the Guy-Lussac's law of pressure changes, then you see Boyle's law as oxygen enters the lungs, then you have the diffusion Fick's law, and you have Henry's law, the partial pressures of oxygen, and you see the oxygen going through through the alveolo-capillary membrane into the red blood cells, and then spreading itself through the capillaries, through to the lung, to the heart. And then that's the hydraulic laws, Pascal's law of hydraulics. And then there's the fusion, the Fick's law of oxygen being transported to mitochondria itself for the generation of energy. This is very beautiful. So at high altitude, we have, when you first go to high altitude, the pneumodynamic pump and the hemodynamic pump fundamentally work. But then as time goes by, the hemoglobin increases gradually, and that compensates and reduces the work of these two pumps. But what happens what's happening of high altitude research? People are studying hypoxia, looking at microscopes, electron microscopes, physiological and molecular
Can I have
change.
next slide?
I'm sorry, this is new slide. You're not seeing it?
No.
No,
No,
still
sir,
we are
no.
seeing the hydraulic loss, gas
Somehow,
loss. Of
I think you stop sharing the full screen. You just go with your normal.
I'm sorry, what do you mean?
Not full screen, not in presentation mode, but in the screen, what it is written, the normal slide. There is some problem when you are sharing with the full screen. It is not moving.
Okay, so I'll just put it like this,
Yeah.
right?
Yeah.
Slightly increase.
Oh.
No, it is again full screen. It may not again go to the next slide.
How can I do that? Because it always, as soon as I put play, it goes to full screen.
Okay, it is. I think that your slide preparation is a 50%. I think it. Yeah, but I think now it can be ready.
Yes, sir.
Maybe I should
This
put
is
it like
fine,
this.
sir. They should be fine, sir.
Okay.
Go ahead.
Okay, I'll do it like this, right? Is that
Yeah,
okay?
yeah.
Yes, sir. Yes, sir. Manually, you can just change it.
Yeah.
I won't press play because it doesn't work. Okay. Anyway, so with all these microscopes, electron microscopes, there's the search for physiological and molecular changes of hypoxia. You will find changes, of course, but then being at sea level, the scientists assume this is bad. This is not good. This is deleterious for health. Well, we think it's the opposite. And for instance, you see this slide. Do you see it now?
Yes,
This
sir.
new slide?
Yes, yes.
Okay. In ischemic stroke, one of the fundamental recovery factors is the increased expression of the vascular endothelial growth factor. You see, when somebody has a ischemic damage or stroke, something that Professor Kusal Das has shown, that the preconditioned rats to hypoxia can have a much more resistance to stroke. So you see here in this study that the vascular endothelial growth factor plays a fundamental role in angiogenesis, and the recovery is fundamental with vascular endothelial growth factor. Well, this slide here that I have presented in one of our papers it has to do with how we adapt to high altitude. On this left side is when you go immediately up to 2 days, when you do the altitude ascent, you have the lowering of arterial oxygen tension and the pulse oximetry. You increase ventilation, the pneumodynamic pump, and the cardiac function, which is the hemodynamic pump, and your pH changes, and it's acute respiratory alkalosis. But then there's renal excretion of bicarbonates, and you have chronic respiratory alkalosis, but we don't call it chronic respiratory alkalosis. We call it a normal acid-base status at high altitude. And these are the values: pH 7.4, which is pH is fundamental for cellular work, and PaCO2 30 and PaO2 60 millimeters mercury. So what happens? You have a increase of pulmonary artery pressure, you have right ventricle dilatation, but these are normal adaptive mechanisms. You also evolve to have a right ventricle hypertrophy, which is fine. That's how it works. We have also pulmonary hypertension. Our pulmonary pressure is at sea level, it's 12 millimeters of mercury. Here it's 25 millimeters of mercury or a little less, and it's fine. That's the adaptation. Hemoglobin also goes up gradually with time, as you can see here. And we also have hypoxic induction factor, increase of nitrous oxide, increase of erythropoietin, And there you see the vascular endothelial growth factor. So that was a protective mechanism for stroke at sea level. Well, at high altitude, we have these mechanisms, and we have more vascularity all over our organism, not only in our hearts that have much higher vascularization, and we have lower incidence of cardiac myocardial infarction, but we also have brains with much more circulation. And those are adaptive, normal, beneficial changes for us. Now I want to mention, I'm here as I was a visiting professor at the University of Copenhagen. I'm here with Professor Paul Erik Paulev, a great friend. He has passed away a few years ago. And this is Ole Sigurd Andersen. And what I proposed to Professor Paulev is that we should modify the Fans-like formula, that is the acid-base balance formula, fundamental for cellular function. And I modified it by changing the hemoglobin according to the altitude and the actual altitude bicarbonates within this. So the formula. So with that information, I was able to change the Sigurd Andersen acid-base charge that has a PaCO2 of 40 here, as you can see, and changing these to different altitudes. For instance, the 2,000 to 3,000 meters is where you see this graph and then this diagram, and then this other diagram for 3,000 to 3,999 meters, and you see how the normal ranges are lowering. In fact, if we go down further, I mean, you go up higher at 40,000, 5,000 meters, it's up there, and on Mount Everest, the normal ranges are way down here. And this means that if acute adaptation to high altitude is achieved, the acid-base status of blood changes and becomes normal with no alteration of base excess or negative base excess, also previously known as base deficit. There can be no base deficit under normal conditions of adaptation to high altitude. In other words, complete acid-base adaptation to high altitude implies a normal acid-base status for that altitude with no deviations from normal. And that's why we wrote this article, do over 200 million healthy altitude residents really suffer from chronic acid-base disorders? We don't think so. We are perfectly adapted. And the question is, do we understand chronic hypoxia? I don't think we do. I think we understand it more than many other people that are at sea level because they are not living it permanently. For instance, this beautiful paper, Arterial Blood Gases and Oxygen Content in Climbers on Mount Everest by Michael Grockett and co-authors from the Caldwell Extreme Everest Research British Group and published at the New England Journal of Medicine, has shown that the interpretation of acid-base status on the summit of Mount Everest using for 4 climbers where they took blood gases at 8,400 meters and analyzed them, they used the sea level formula, which resulted in a calculation of base excess of minus 6.9 millimoles. Can an individual be walking around the mountain with such base excess? I mean, you see these changes in intensive care units where a patient is intubated and so on. So that's why we decided to show what happens at high altitude if you use the normal sea level calculation formula for the acid-base balance. And you see these blue dots are for different altitudes. You see at sea level, it's 0. The balance has to be 0 always. Acids and bases have to be balanced. So it's 0 at sea level. It works at sea level, but at 2,500, 3,000, 4,500, it starts increasing and it comes to 8. And in the case of Grockott's data, they calculated and it was 7, completely off the reality for us. So what we did is we wrote a paper called Extremely High Altitude Hypoxic Conditions During Mount Everest Expeditions, Residents at the South Pole Stations in Tibet and among the Andes. Fans-like equation modifications is crucially important for acid-base measurements. As they calculated it with the normal sea level fans-like formula and then found a base excess of minus 6.9 millimoles, we used our modified high altitude fans-like formula and it was 0.7, almost 0, perfect cellular metabolism. So this is the real acid-base status of subject at 8,500 meters. And this is why the extraordinary mountaineers are able to survive and continue to perform exercise, even at such extreme hypoxia. Now, of course, there are fundamental changes that are irrefutable. At high altitude, we have an increase of red blood cells and hemoglobin, as shown here in this graph. We also have a decrease of carbon dioxide. You see how carbon dioxide is going down, and on summit of Mount Everest, it's around 8 millimeters of mercury. So this is why we use those to create our tolerance to hypoxia formula, those two variables. One is hemoglobin, which is a long-term adaptation, fundamental in numerical position here for the tolerance to hypoxia. And then on the denominator is the arterial carbon dioxide. In other words, a lower carbon dioxide makes you more tolerant to hypoxia. And this is the way it's developed. And what this shows is that in La Paz, we are 1.7 times more tolerant to hypoxia. And in Everest is 4.86. The pressure of oxygen plays a key role on survival. At sea level, the higher pressure creates the intense biological diversity. Nevertheless, being our planet Earth provided with mountains, life has adapted to low oxygen pressures. Humans have evolved to live at higher altitudes more and more. And so my father has, for instance, proposed that man can adapt to live even on the summit of Mount Everest. And this is a very interesting thing. He said the organic system of human beings and all other species tend towards adaptation to any environmental change within an optimal period of time. And that's how we developed the adaptation formula. It never tend towards regression. There can be no loss of adaptation or de-adaptation that inevitably leads to death. Of course, when a scientist finds that the lungs are filled with fluid at high altitude, he can panic because it comes like this, you see? It comes like this with the lungs completely filled with fluids. That's high altitude pulmonary edema. And then 2 days later, we have the lungs completely cleared. So we wrote an article called Acute Mountain Sickness, High Altitude Pulmonary Edema and High Altitude Cerebral Edema, A View from the High Andes. And it was interesting because 17 sea-level high-altitude biologists wrote to the chief editor asking that the article be withdrawn. It wasn't following what they had been seeing. But this was based on 51 years of our experience treating these diseases. And we treated all of them during 51 years successfully at high altitude at our institution with the resources. I am very fortunate to say that I was I'm sorry, but I'm going to have to play this because do you see this letter of support now? Do you see my letter of support?
Yes, sir. Yes, sir.
Okay,
Yes, sir.
So please tell me if it doesn't advance again. This letter of support was fantastic. It was Professor Tupil Venkatesh Kushal Das, but we also had many scientists from around the world supporting our article. I hope you have seen all these people because many are from India and they have given us full support and we're very, very thankful. And now you see this new slide where the article was anyway retracted. Do you see it?
No, sir. No, it's not changing. You have to escape again.
Okay. So I have to go back. I'm sorry. I have a dynamic conference, so it should be active, but I guess I can't make it active, unfortunately for you. We go back, and here we are now again.
Yes.
So this article was retracted. And what happened, this was a very interesting stimulus for the oxygen transport triad in high altitude pulmonary edema, a perspective from the high Andes, where we answered some of their questions. They were all trivial questions and they were completely answerable. And a new enhanced version has been accepted in a very important journal. So going to sea level, we have fears as sea level people going to high altitude. You see, you fear hypoxia. We fear dengue, we fear chikungunya, we fear snake bites, we fear sharks, We are afraid of hyperoxia. And we have created a term called, instead of HAPE or HAZE, we call it LAPE, low altitude peripheral edema, because our feet can swell and so on. I have no time to give you full details on those things. But in any event, this is the formula of adaptation to high altitude. I'm sorry, I put it in Spanish. Adaptation to altitude is equal to time over the altitude change. And what we have written and we have observed is that there's extended longevity at high altitude. So there's benefits of exposure to chronic hypoxia. You see here in Bolivia, going from the lowlands, the departments from 220 tons in the Amazon, going up to Potosí at 4,100 meters, the city of Potosí, we can see that as we go up higher and higher, we have more people living over 100 years old. This is amazing, and this is explained by many reasons: genetics, less exposure of insects and wild animals, dryness of the environment, less incidence of asthma, less incidence of bronchial inflammatory disease, less incidence of obesity and metabolic disease. You don't see very many people with obesity here in La Paz. Less stress, healthier diets maybe, and other factors, less lesion secondary to reactive oxygen species. So is hypoxia favorable to longevity? Is a good question that we ask ourselves. Now, to go towards the final part of my presentation and to put the cherry on the cake regarding our concept of beneficial life at high altitude, where is COVID less deadly, at sea level or in the highlands? This is a very important question. Let me tell you that based on our knowledge, at the beginning, on January the 31st of 2020, based on all our knowledge of chronic hypoxia, I was interviewed by the Universidad Mayor de San Andres, radio and television, and I affirmed there that COVID would be less severe due to several factors, among them higher ultraviolet radiation at high altitude. And this was our first publication. Does the pathogenesis of SARS-CoV virus decrease at high altitude? This was by Christian Arias Reyes and Jorge Solis from the University of Laval in Canada. And of course, my daughter, Natalia, and myself participated with some other Bolivian colleagues working outside of the country. And we proposed that there was a lower amount of ACE2 receptors in the lung, so there was a less possibility of affectation from COVID virus, which, by the way, I have also introduced a new term of the attack of the lungs by the virus that's called pneumolysis. And we wrote a paper, and Professor Kushal Das, Professor Tupil Venkatesh, and Jorge Solis are also co-authors along with my daughter, Natalia. And there's also higher ultraviolet radiation at high altitude, and it's a protective effect for us. It is not harmful. And what we found there is that the global distribution of COVID-19 cases reduced over altitude. As you see, the altitude increases. We had less cases of COVID. The second publication was decreased incidence, virus transmission capacity, and severity of COVID-19 at altitude on the American continent. Here with Christian Reyes, Jorge Solis again, and here we are with Natalia. And there we showed that as you go to different altitudes, and it was in 23 countries, you can see how the incidence number of cases dropped, the incidence of COVID. As you go higher, there's less COVID. And then we wrote a letter to the editor because there was an article called mortality. They affirmed that the mortality was higher at altitude. They assumed this because we live in hypoxia, and they said, oh, this disease produces more hypoxia, so it's going to be very deadly. Well, we wrote a letter to the editors, and it was very difficult to publish it. The editor sent us 9 versions to correct because he kept erasing everything we had said in a letter to the editor at this journal. But what we showed there is, you see the green line here, this is Santa Cruz, the lowlands in Bolivia. That's the incidence of COVID during the first wave. And here is the high altitude of La Paz. They have similar populations, but look, it's 26% less at high altitude. And here in the table, we see that in the lowlands, The case fatality rate was 8.6%, but in the high areas of Bolivia, it was 3.2, almost 1/3 less. The same happened in Peru, 5.9 in the lowlands and 3.1 in the highlands. And now we have finished a paper and has been submitted, case fatality rate at COVID at high altitude, a multinational study in Bolivia, Ecuador, Colombia, Peru, and Mexico. Natalia is the first author, and I'm the leading author here with colleagues from all these countries, even colleagues from Germany and the United States that I have invited to join us, because we have studied 161 million inhabitants in all these countries to see what the case fatality rate was in COVID. And this is what we found. In Bolivia, this is but the lower of 1,000 meters of altitude, and this is about 2,500 meters. And you see that it is higher always in the lowlands. It repeats itself in Colombia, Ecuador, Mexico, and Peru. And you see the recovery rate is much higher at high altitude. And this is the same statistically showing the all significant, totally significant findings in the chi-square and odds ratio. But we also wrote another article of intensive care patients where we found that EPO and hemoglobin, those patients that had a higher EPO and a higher hemoglobin survived COVID much better. And so, in fact, they all survived when they had a higher level of EPO. We used EPO as a treatment to treat pneumolysis. The destruction, pneumolysis means the destruction of the alveoli due to COVID. And so, there are other articles supporting this concept from many areas, from Ecuador, USA, Peru, Brazil, Saudi Arabia. And so the conclusion for us is exposure to chronic hypobaric hypoxia is mostly beneficial even during the COVID pandemic. You see, and this, if life is so successful at high altitude in the cities of La Paz, El Alto, and Cerro de Pasco, and the human beings in his quest for instinctive survival will leave planet Earth and travel to space, is it important that humans adapt to chronic hypoxia? We believe so, and we have postulated it many years ago that in space, the ships should use chronic hypoxia. This would prepare for faster preparation of extravehicular activity, oxygen economy, greater possibility of survival in space. And as you all know, we carried out a football game on the summit of Sahama. And I can't show you the video right now, but they played at 6,542 meters. I show you here my father, who left us 6 years ago, and here's Natalia. We're most grateful to our collaborators. And we want to invite you to join us at the World Capital of Hypoxia, La Paz, Bolivia, during the 8th Chronic Hypoxia Symposium this year between October the 10th and 15th. And it's a fascinating meeting because you will be exposed to this life. So now I will stop sharing this screen, and I will, if you please allow me to share one video, which is very brief and short. And I hope I can do it. Let me see. Yes.
Yes, sir. Please.
This is the video. Are you seeing it?
Just play it, sir. You can play it.
Okay. It's not playing. Something is wrong. Oh.
It's not played, sir.
Yes. How strange. This is very strange. It never happened this way. I'll try one more time.
Then you click there. You click on the.
Yes, yes, yes.
Click now.
Yes, sir. It is played now. Can you maximize it? Can you maximize it?
Oh, how can I do that? I'm sorry. I'll try it one more time.
Yes, sir.
Something happened here. Very strange. Let's see.
Double click on it.
Okay. Do you see a black screen? Oh, no,
it's not shared, sir. It's
yes,
not shared.
Unfortunately, I'm sorry. I can't take any more of your time, so I can't show you. But what that was is a very interesting image of La Paz, where you would see how we are carrying out our normal life at high altitude in a very, very normal life. And I hope you come to visit us. I'm sorry for all the inconveniences of this presentation. I've given hundreds of talks. It has never happened. Murphy's Law once again. Thank you very much.
That's fine.
I have one question, Professor Jabeka.
Yes, Professor.
See, it's a very interesting topic regarding this high altitude hypoxic conditions, especially it's a boon for the COVID-19 patients, okay? Do you agree with that one? Even if we create some conditions, hypoxic conditions in a hospital for the COVID-19 patients, can they do recover fast when compared to the normal? That's my question. I don't know whether how is it possible. Creating hypoxic condition at the.
Do you mean if they recover faster? Is that the question, or I didn't quite get it? I'm sorry.
Yeah. My question is, if a COVID patient is admitted in a hospital and creating high altitude conditions like hypoxic conditions, can the patient recover quickly?
Yes. I believe that if you use erythropoietin, you are doing an artificial exposure to chronic hypoxia. You are stimulating the production of red cells. And so if you do it early with the problem, you administer EPO, you increase the possibility because it's one of the three factors of the oxygen transport triad. And since this is pneumolysis produces a reduction of oxygen, the transport, the capture of oxygen, if you have more red blood cells, it will capture much better the supplementary oxygen you are giving. So even a transfusion would be extremely beneficial as it would increase the number of red blood cells. Very important for the transport of oxygen to the tissues. Yes, that's what we have learned, Dr. Reddy. Thank you for that. That's an interesting question.
Yeah, thank you. Thank you.
Can I have a question?
Yes, Dr. Paul.
Yeah.
Yeah, Professor Paul, just I'd like to introduce you with Professor Gustavo. Professor Gustavo. Professor Gustavo?
Yes.
Yeah.
Yes, I'm listening.
Here is Dr. G.K. Pal, Professor G.K. Pal, one of the distinguished professor of India. And very renowned. His book is the most popular book among the physiologists written in India. And he's very close friend of mine. He is like a family member. He was a former professor and head of the department of JIPMER, now professor of physiology there in JIPMER, Pondicherry, and also he's the dean of faculty of University of Pondicherry. So here is Professor G.K. Pal, distinguished professor of, true sense distinguished professor of physiology of India. Professor G.K. Pal, please go ahead with Dr. Gustavo.
Thank you so much. Professor Gustavo, my heartiest congratulations for a wonderful work and a brilliant presentation you made today. It is really great, and I was really delighted to see your presentation and the publication in the journals like New England Journal of Medicine. Such great works are done by great scientists like you. I have a small comment, actually, not a question. You mentioned that VEGF, the vascular endothelial growth factor, which increases at high altitude. And what I know actually that VEGF is a marker of vascular inflammation, VEGF. And VEGF is involved in mountain sickness. So if the VEGF is high at high altitude, what you have mentioned that at high altitude, there is a higher longevity due to advantage of hypoxia, then how this VEGF, which is a vascular inflammatory marker, and how actually this does not cause the cardiovascular problem and still people live longer at high altitude. Can you just...
Dr. Pal, I am delighted to meet you. Due to the background of the presentation of Professor Cusaldaso, partly, but also because your question poses a very, very interesting thing. You see, in our paper that we wrote about acute mountain sickness, high altitude pulmonary edema, and high altitude cerebral edema, that was the 18 people that criticized it. One of the criticisms was to a point that I said there, that when I said, when we are exposed to hypoxia, the hypoxia inducible factor increases. Of course, we all know that, but the increase can be so big that it can also be deleterious, not deleterious, but aggressive. And the organism can feel an aggression from the presence of HIF. And now that you mentioned the BEGF can be thought of as an aggression if it is overproduced. And maybe that's the explanation why someone has deduced that it would be an inflammatory marker. Actually, it is a response to improve the conditions of the organism. The organism is trying to build more vessels to supply more oxygen to tissues. It's a desperate attempt to improve circulation and to transport more oxygen to tissues with vascular endothelial growth factor, more capillaries. But it cannot be done. That's why the adaptation formula carries the time factor. It requires a time factor. So you're right, and it's interesting that you point out that it would be considered an inflammatory factor, but actually inflammation is also a response of our organisms that can be favorable in many cases, because it increases for instance, when a mosquito bites you and you have an inflammation, a local inflammation in your skin, this is not bad for you. It's good because you are producing more capillaries, you're vasodilating the capillaries to produce more antibodies to protect your organism, to antihistamines and so on, to produce to lower the aggression. So inflammation can be favorable. But now that you mentioned this about BEGF as a marker, I'm a bit surprised on that aspect.
Thank you, sir. Thank you so much. Thank you so much.
Thank you. Thank you. Wonderful question.
So every time we hear from you, I heard you talking about hypoxia many times. Every time I get to learn a lot. Thoroughly enjoyed your talk, sir. Thank you so much.
Thank you.
Any other questions from the audience? Can we allow? Any questions?
Well, I would like to comment something, Dr. Lata.
So please.
Dr. Nino Ramirez is a good friend of mine. In fact, let me tell you something interesting. When we held the Global Hypoxia Summit in DPAS,
yes,
Nino came to me and talked to me and approached me and talked to me. And now since then we have become great friends. And yesterday he told me, I'm sorry, I won't be able to be in your talk because it will be 3 in the morning and I cannot be at that time. And I understand I wasn't in his talk either for the same reason, but he wished me good luck with my presentation. And in my mind, I said, I don't need good luck. I've done hundreds of presentations. I've always done it well. I needed the good luck because my slides didn't work properly.
Okay, wonderful, sir. That's wonderful, sir. We heard sir talk also. So I think this is all we have for sir's session.
Yeah, Dr.
Sir,
Lata,
yes,
Dr. Lata, I will say one word here.
Yes, sir. So please.
Because Professor Pal is here, so
yes, sir.
I had been to La Paz, where the background of Professor Gustavo is saying in 2019. Yes, that's background. So I had been there, and when I had been, very well, my hemoglobin was actually 11, and that too actually one blood transfusion that has come rise from the 8 to 11. And after 2 months, I immediately left for La Paz, and it was a long program to US also. And during this time, I was panicked, but my former vice chancellor, who is no more now, he assured me that nothing will happen. No need to further blood transfusion. 11 is enough. You can adapt. Very surprisingly, when I get down from the airport, that is the El Alto, that is further 600 meter higher than the La Paz. It is 4,100 meter altitude. Practically, I was a little thinking that whether I will be having nothing happened to me. Even when I went and I have been thoroughly checked and examined by Professor Gustavo and his medical team, my blood pressure, everything is normal, except my oxygen saturation is 90. It has come. That is because it's common for them for having a high altitude, low oxygen saturation. So that's given me a classic example. I did not have any problem in the high altitude, even though I was having a very low hemoglobin concentration in practically. So being a physiologist myself, I will ask Dr. G.K. Pal, please, Professor Pal, this is an issue happened to me and I survived. And I have a nice time in the La Paz in many places I travel, including Peru, Colombia, many places, nothing happened. So it's a wonder in the high altitude. I think until we go there, we will not realize that what happened there.
Yes, yes, very correct, Professor Das, very correct. And that is what Professor Gustavo mentioned, that his father's work, that human being can adapt to any altitude, any kind of high altitude. And you are the example. You are the example.
Yes, sir, yes.
Yes, absolutely. And
Yes.
the 2 million people living here in the city are never concerned about hypoxia, never. I mean, except when they would some kind of lung disease, of course, but never. And the truth is, if you come to high altitude like Professor Kusaldas has done, you stimulate your organism to create a defense mechanism that is favorable for you. I think that maybe you have gained some, as smoking takes away years of longevity from you. It takes away years if somebody smokes. If you come to hypoxia, you increase the possibility of living longer. So Professor Kusaldas, you did great, and it was wonderful to have you here. And we will have you again, I hope, this October, of course.
Definitely, sir. We all want to experience the same thing. Hope to see you again. Yes, sir. Thank you, sir. Thank you for your wonderful talk and interaction. At this note, I would like to thank organizers for giving this opportunity of chairing the session from Professor Gustavo. Thank you so much, sir. Thank you.
Thank you. Thank you all. I love India.
Thank you, sir.
Love for.
We would like to thank Professor Gustavo for this very informative session. We also thank Dr. Lata Mathur from BLD University and Dr. S.K. Reddy for chairing this session. Moving on to our next session, the Symposium 6, Sleep, Memory and Neuroprotection. I would request Dr. Usha Panjwani to carry forward the symposium.
Thank you. So this is the 6th symposium, the last of the scientific sessions of Fips 22. So a very good evening, afternoon, morning to all the participants and speakers of this session. And this session is on sleep, memory and neuroprotection, in which we have 5 speakers, and one by one we will go through it. I hope you will enjoy and have fruitful deliberations with the speakers who are experts in the area. So I start this session with a young researcher, Dr. Bdnf Kumar from Japan. Dr. Bdnf is a postgraduate from Jawaharlal Nehru University, JNU, Delhi. He did his PhD in physiology from the Department of Physiology, AIIMS, New Delhi. And he's been a recipient of honors and awards like B Kiran Gold Medal in 2011. And further, he has completed his postdoctoral training at Max Planck Institute of Psychiatry in Germany, where he analyzed sleep EEG of mice models of stress disorders. Subsequently, Bdnf moved to Japan, where he received a JSPS fellowship for a research project on sleep and memory. And last year, he also secured a highly competitive Japanese grant as principal investigator on a project on infantile amnesia. So without further ado, I hand over the floor to Dr. Bdnf to start his lecture.
Hello, good evening. Can you hear me?
Yes, we can hear you.
And can you see my slide?
Yes.
Yeah. Okay. Okay. Good evening, everyone. My name is Dipendra Kumar. I am thankful to the Fips organizer for giving me opportunity to present my recent work. As we all know that sleep is important for memory consolidation, but the cellular mechanism, how and when actually sleep consolidate memory is poorly understood. We have discovered a subpopulation of neuron in the hippocampus in the area called dentes gyrus. You can see in the left-hand side figure, these pink color cells are actually newborn neuron, also called adult-born neurons. And these neurons are actively participating in memory consolidation. So my title of the presentation is Young Neurons' Role in Consolidating Memory During REM Sleep. So after discovery of adult hippocampal neurogenesis by Altman and Das in 1965, it is now widely accepted that adult hippocampus can generate a new neuron. This is commonly known as adult-born neuron because adult hippocampus can generate new neuron. In the hippocampus dentes gyrus area, particularly the subgranular zone is the host of neurogenesis, where neural stem cells divide and generate progenitor cells, and then progenitor cells develop into a mature neuron, and then finally as a mature neuron. During the development, these neurons receive input from entorhinal cortex and send output to CH3 area. And finally, they become an integral part of the memory circuit or hippocampal circuit. So we wanted to track this neuron. So here what you are seeing is actually adult-born neuron or new neuron at a different developmental age. So this is a 2-week-old adult-born neuron, and then this is a 4-week and 10-week. As you can see that with maturation, they become more sending their arborization to the molecular area and developing like becoming like a mature neuron. We targeted 4-week-old neuron. The reason was because this 4-week-old adult-born neurons shows high plasticity and high excitability. Why we were interested in this adult-born neuron? Because it was shown by several papers that these adult-born neurons are actually directly involved in memory encoding. There are so many literature suggesting that these neurons are actually helping our brain to separate or called pattern separation. And also here I am showing example of memory resolution. For example, as you can see in this cartoon or a schematic diagram, the left-hand side dentes gyrus is with adult-born neuron, this green color circle, whereas the right-hand side is without adult-born neuron. As you can see that without adult-born neuron, there is no encoding of some novel information, for example, sun or word here. It is hypothesized that adult-born neuron is actually required for encoding novel feature or novel, or in other words, new memory. We were interested in sleep and memory, so we wanted to know what is the activity of this neuron during sleep. So for this purpose, we used transgenic mice to express calcium sensor on the specifically on adult-born neuron to image their activity during normal sleep-wake of the cycle of the mouse. So what you are seeing here is the activity of adult-born neuron during the non-REM stage, and this is during the red is during the REM stage. And when it goes into the wake, there is definitely high activity. So here the green line is showing the activity of adult born neuron in wake stage. But when mouse enter into the sleep stage, these neurons become more and more slower and sparse. So what about, this was the without formation of memory. What about if we induce learning and memory in the mice, and how about then the adult born neuron activity? For this purpose, we induces generated peer memory in mice using a classical contextual fear conditioning paradigm. As you know that there is a delayed shock where food shock is appeared after some delay, and then mouse can form memory of that context. On the other hand, immediate shock is a kind of negative control where when mouse is kept placed into the chamber and food shock is delivered immediately, then there is no formation of a memory as reflected in the data here. So delayed shock mouse shows higher freezing because they were able to make memory of the context. But what about the adult born neuron activity? It was for our surprise when we saw that adult born neuron activity did not change when there is no formation of memory. So it was similar to baseline situation. So after a shock, there is no memory formation and no change in the activity. On the other hand, when memory is formed, specifically during the REM sleep, we notice there is a significantly decrease in the activity of adult born neuron. So this was very interesting for us, and we thought why there is such decrease in activity of adult born neuron during the REM sleep. Is it processing memory? But before going into memory processing, we need to first confirm whether this REM active neuron, REM active adult born neuron, is actually encoding memory. To do that, we have to do again imaging of adult born neuron during learning as well as during post-learning sleep. So first we identified adult born neuron which were active during the REM sleep. So we said REM positive neuron, and then we tracked their activity during the learning period, whether those neurons are really active or during the encoding period. And here is the result. So we found indeed the REM active neurons are actually active during the learning compared to not REM active adult born neuron. And in a comparison with total percentage of REM active cells, we actually found there is a larger percentage of adult born neuron which were active during the REM sleep is also active during the learning. So this confirmed that the REM active neurons are actually involved in memory encoding. Then what about, is it a very relevant or to the memory processing? In order to know that, we wanted to silence the activity of adult born neuron, specifically during the REM sleep. So for this purpose, we again use transgenic mice to express haloropsin to silence the activity of this neuron, specifically during the REM sleep. The left-hand side is you are seeing is the schematic diagram. So after learning, mouse was allowed to sleep in their sleep chamber. But whenever they are entering into the REM stage, we silence laser light. For example, here you can see that when we notice the theta wave is coming, we silence laser light to block the activity of adult born neuron in order to know whether their activity is relevant for memory processing or memory consolidation. Before going to memory data, we have to confirm whether optogenetic silencing of adult born neuron has any effect on sleep or EEG oscillations. So we confirm that there is no effect of optogenetic intervention on sleep or EEG power spectrum. We wanted to go further deep down to see whether local field potential of the dentate gyrus area where we are targeting silencing, is there any change because of the optogenetic intervention? And we confirmed that there is no effect on LFP of the dentate gyrus area because of the optogenetic intervention. Now here comes the effect of silencing of adult one neuron during REM sleep in memory. And it was very surprising to know that there is an effect on peer memory. So the mouse is the upper one is the one which was silenced during the REM period. And the bottom one is, you can see, is a control mice. So these both mice received light, but the bottom mice has no halorhexin. So their memory is intact. They are freezing more in the contextual chamber, but the other one is freezing less. Of course, they are freezing, but it was significantly lower. So we conclude that the probably adult one neuron activity is essential for memory consolidation during REM sleep. But what about non-REM sleep or wake stage? But first of all, we wanted to know the time after learning. In the previous figure, what you have seen is the first 6-hour period where we silence the activity of adult one neuron. What about later REM episode? So in this experiment, we silence not the first 6 hours of REM episode, but later period. And we found that there is no such effect, what we have seen before. And also during silencing during the non-REM stage or wake stage in similar manner, there was no significant change in the freezing behavior. So we conclude that the adult one neuron activity is very relevant for memory consolidation during the first 6 hours from the learning. But what about mature neuron, not adult one neuron, like the developmentally one neuron? So for this purpose, to know their involvement in memory consolidation, first we wanted to image their activity during sleep. So this time we express a calcium sensor on the granular neuron or mature neuron using a virus technique, and image their activity during the sleep-wake cycle after again inducing fear memory or fear learning. But in the mature granular neuron, we found there is no such effect of the learning. The activity was comparable to the baseline situation. To confirm further, we silence actually mature granular neuron during the REM stage to validate whether this neuron's activity is really important for memory consolidation during REM sleep. And for this purpose, using various control, we confirm that mature granular neuron is not, activity is not actually relevant for memory consolidation. We wanted to go into the detail of the mechanism about what is happening when we silence the adult one neuron, what is happening at the dentate gyrus level or neural network level. For this purpose, we did transcriptome analysis of the dentate gyrus, and for our surprise, we found that because of the silencing of adult one neuron during REM stage, various immediate early gene, for example, FOS, NPS4, or NR4A1, so decreased in the gene expression, indicating that there is inhibition of the dentate gyral neural network activity because of the silencing of adult one neuron during the REM sleep. Then we went further to see about the adult one neuron morphology to find out whether there is any change because of the silencing of adult one neuron during REM stage. Using various, again, control. This is halonestin is the one where silencing happened. There is this is a light control, or this is a SAM control. This is no learning control. Using all those control, we confirm that the spine density and a spine head diameter of the adult bone neuron does not change, except the spine neck length of the adult bone neuron is elongated. You can see it's elongated because of the silencing those neurons during the REM stage. We suggested, as we know from the literature, that during REM sleep, the synaptic strengthening happens. So the spine, you can see in the upper panel, their spine neck length is elongated and it gets shortened. On the other hand, during the REM sleep deprivation, it remains elongated. So it is suggested that during the REM stage, there is a synaptic strengthening happens. So this the spine gets shortened to enhance the synaptic connections. We suggest that probably silencing of adult bone neuron during the REM stage affect the synaptic strengthening of the adult bone connection with the other neurons. And probably that is one of the possible region which may intervene the memory consolidation process. Now, adult bone neurons are, this is from the human study, is now, there is evidence that adult bone neurons number progressively decline as Alzheimer's disease advances. As you can see in the advancing stage from BRCA stage 1 to 4, there is a significant decrease in this double cortin adult bone neuron marker cells in the human. Of course, with age, there is a decline in number, but in the Alzheimer's patient, the number is decreased much.
What time is that? 7 o'clock or 6 o'clock?
Yeah, thank you. I want to thanks.
7 o'clock. Huh?
I want to thanks specifically this works was performed in Dr. Masanori Sakaguchi, the talented young PI, and he guided me through this process. But before reaching here, I'm very thankful to my training at AIIMS New Delhi and Max Planck Institute in Germany, where I get more used to with this sleep and sleep-related functions. Currently, I'm working with the IIIS director, Masashi Yanagisawa, with a neurodevelopmental circuit for the sleep and memory. Thank you, everyone. I would like to receive questions.
So thank you so much, Dr. Dipendra, for that such an interesting talk on adult bone neurons. Am I audible?
Yes.
Yeah. So thank you so much. I'm requesting all the participants to put the questions in the chat box and we can take them up in the end. Okay. So now we can move on to the next speaker, and the speakers can restrict their talk to 15 minutes each. So I invite Dr. KP Mishra. Let me give you a brief introduction about Dr. KP Mishra. He is working as a scientist in DePaz, DRDO, and he's made a significant contribution in the field of high altitude physiology, in particular immunophysiology. He was also a member of the 28th Indian Antarctic Expedition. He received several prestigious awards like ICMR Shakuntala Amirchand Award and Professor B.K. Aikath-Orishan Award from ICMR again, and several others. He has published 85 peer-reviewed research papers and articles. He has authored books and book chapters, and he's also on the editorial board of some of the prestigious journals. So I will invite Dr. KP Mishra to start his talk on the role of neuroinflammation in memory impairment. Please start, Dr. Mishra.
Thank you, madam. Can you see my slides?
Yes, we can see your slides.
Is it moving?
Yes, it's moving.
Okay, ma'am. So first of all, I am very much thankful to the convener of this symposium, Dr. Usha Panjwani, for giving me this opportunity to share some of my research with this August gathering, and of course the organizers for providing me this platform to this forum to share my research on the role of neuroinflammation in memory impairment. Well, neuroinflammation is the inflammation of central nervous system that occurs as a response to a variety of inflammatory signals. It could be stress, trauma, aging, infection, and pathogenic protein aggregates. So what makes the basis of neuroinflammation? The basis could be a combination of responses from different cells like neurons, microglia, astrocytes, even peripheral immune cells, which secrete cytokines, chemokines, and some complement proteins that are endophyllotoxins and inflammatory in nature that constitute the basis of neuroinflammation. And in brain, microglia, which is actually a resident brain macrophages that accounts for 10 to 15% of all cells in the brain, which actually responsible for the inflammatory event, and it performs the housekeeping job in brain, which clears the random cellular debris. Microglia is very plastic in nature. It can be converted to pro-inflammatory state or anti-inflammatory state depending on the need. And if it is activated by inflammatory signals, or for example, as I said, stress, trauma, aging can activate these microglia cells to a classically activated microglia cell type that is called as the M1 type of cells. So how these stress stimulate these microglia cells? We know that when we are stressed or any kind of damage occurs to the cell, a damage associated molecular patterns are secreted. These DAMs are nothing but the alarmins which stimulate the inflammatory signals by binding to the pattern recognition receptor, which are present on our sentinels of the immune system, that is the macrophages. These are the different toll proteins, like from toll-like receptor 1 to toll-like receptor 13. So far, 13 toll proteins receptors were known in human, and toll-like receptor 4 is most studied toll-like receptor in human being. And when HMGB1, which is a high mobility group box 1 alarmin, when it's secreted by the damage of the cells, it can bind to the toll-like receptor 4 and can activate the downstream signaling cascade by activating mediated IKK complex and activating the transcription factor NF-kappa-B, which is the master transcription factor of our inflammatory event, and secrete a series of cytokines which are inflammatory in nature. These are the TNF-alpha, interleukin 6, and interleukin 1 beta. It's not moving. So this is the stressful environmental, how the stressful environmental conditions activates the inflammatory events. For this, I have participated in the Antarctic expedition, one of the Antarctic expedition, that was the 28th Indian Antarctic expedition, to evaluate how the stressful environment affect our immune system. So from Cape Town to Antarctica, we went from SIP, and we measured the cytokine profiling we did, and we found that when we were under SIP in the ocean, the inflammatory cytokines were significantly enhanced. But when we came back to the station, that is Maitri station, the inflammatory level were significantly decreased. This means that our body has a kind of resolution to the inflammation. So resolution of inflammation is very important. And the inflammation is so important that this whole issue of the immunity journal was dedicated to the inflammation, focusing on the cytokines and the inflammation. and which is, as you can see, the inflammation stands for the flame or fire, and that is why this cover page of immunity was shown as the inflammatory cytokines. So when we are under stress, our brain cells secrete kind of the alarmins that activate the microglia cells, in particular the macrophage cells. You can say in the brain resident macrophages are known as the microglia, which secrete inflammatory cytokines, and these inflammatory cytokines are deleterious if persist for a longer period. So they causes the neurodegeneration, and it can be protected by some kind of the neuroprotective treatment, and these agents could be any means like endographolide, which we have tested, and we found that it is good in the modulating the M1 cell towards M2 cell by increasing the phagocytosis, repairing, remodeling, and even it enhances the memory as well. So this is the famous endocrinologist Hans Sille. He said it's not stress that kills us, it is our reaction to it that kills us. So how the reaction to the stress can be prevented? So stress reaction can be prevented by different means. Yesterday we have heard the lecture of Dr. Selvamurthy. He stressed upon how the yoga intervention can prevent the different types of stress response. So here I am going to show you how the Ayurvedic medicinal plant Endographis paniculata, which is in India known as the Kalmeg in North India, and commonly it is known as the king of bitter because of the very bitterness taste of this plant, which has a very important labdent diterpenoid that is known as the andrographolide, which prevents the inflammation. So here our objective was to find out the role of andrographolide in attenuating neuroinflammation and working memory impairment. So to fulfill this objective, we have divided our study in vitro as well as in vivo study. And for in vitro study, we have used primary cortical glia culture and microglia enriched culture with 4 groups: control, LPS treatment group, because LPS is a known positive stimulant for inflammation, and then cells which is treated with andrographolide alone, and the cells which are treated with LPS along with the andrographolide. Similarly, in vivo study, we used biopsy mice, and there were 4 groups: control, LPS, andrographolide, and LPS treated with andrographolide. And the methods which we have used were the flow cytometry, immunocytochemistry, immunofluorescence, ELISA, immunoblotting, and radial arm image for behavioral study. And this is the 8-arm radial image assay we have conducted for behavioral study to find out the working memory scores. Here we have the methodology were used: 3 trials per day for 5 days, then training, 3 trials per day, 15 days, and followed by the testing for 26 days. This is the primary glia culture protocol, where we have isolated the primary glia culture from the neonatal rat pups, and you can see the mixed glia culture were containing the 45 to 50% astrocyte, 35 to 40% microglia, and 10 to 20% oligodendrocytes. And enriched culture, more than 95% of the cells were positive for microglia marker. So to begin with, we have done the cell viability assay because it is important to identify which dose is not cytotoxic. So we found that andrographolide treated with 1 microgram per ml was not causing any kind of cytotoxicity at the 24-hour, 48-hour, 72 hours tested. It is also not causing any kind of cytotoxicity when we have given it with the LPS. So now the question is how the HMGB1 stimulated by LPS and how the andrographolide take care of the HMGB1 expression. As you know, HMGB1 is the alarmin which stimulates the inflammatory event. So we measured the HMGB1 expression by Western blotting and by real-time PCR. We found that HMGB1 expression was significantly reduced in the LPS treated group along with the andrographolide. As you can see over here, the LPS induced HMGB1 expression was significantly reduced both in vitro as well as in vivo culture. You can see this is the prefrontal cortex where the mouse were treated with the LPS, and when it was treated with the andrographolide, the level of HMGB1 was significantly reduced. We measured it by the other means, like at the transcription level, the HMGB1 level was significantly reduced. That means the endographylide is able to decrease the HMGB1 level. Now, whether it enhances the pattern recognition receptor or not, that is also a question. So we have measured the toll-like receptor 4 in vitro as well as in vivo model. We found the increased toll-like receptor 4, and when it was treated with the endographylide, we found significant decrease in the toll-like receptor 4 expression. As you know, the toll-like receptor 4 is one of the pathways for stimulating the inflammatory signals, and CD284, or you can say the toll-like receptor 4, is significantly reduced by the treatment of the endographylide. It was further confirmed in the prefrontal cortex region that the downstream cascade of the toll-like receptor 4, that is NF-kappa B, is significantly reduced by the treatment of endographylide. So we concluded that endographylide is able to suppress the toll-like receptor 4's expression on the macrophage surface. And it can also be able to prevent the inflammatory activation, or you can say the transcription factor stimulation. Now, what happened to the different types of markers present on the macrophage cells? Is it enhanced or it is suppressed? So for that, we measured the CD14 expression, CD11B expression, CD68 expression, because CD68 is telling us the type of microglia, whether it is activated or not activated. So we measured and we found that endographylide is able to suppress the CD68 as well as CD14 and CD11B expression. The CD14 is able to bind to LPS binding protein and along with the LPS, so it is decreased, and therefore the inflammatory event can be prevented by the treatment of the endographylide. Now, one important cell is the astrocyte, which is also playing important role in inflammation. And a glial fibrillary acidic protein were measured in the LPS treated group as well as the endographylide and LPS treated group. We found that GFAP expression was significantly reduced in the endographylide treatment group. So it is clearly indicating that all the inflammatory stimuli or signals which are produced by the cells can be prevented by endographylide. And endographylide we have also tested in another model like rheumatoid arthritis model. There also we found that endographylide is able to reduce the inflammatory cytokines in the synovial joint. So therefore, it is very good as an option for the treatment of the inflammatory diseases, because the anti-TNF therapy is very expensive therapy for the inflammatory diseases. Now, the question is the another pathway of the inflammation is very important to look into, because not only the toll-like receptor, but there are many other pathways of inflammation, like inflammasome pathway, which is not activated by the toll-like receptor, but it is activated by the purinergic receptor, which is like P2X7, that can be enhanced by the alarmins, like ATP and other purinergic ligands. So P2X7 receptor we measured, and in its downstream pathway like inflammasome pathway we measured, and particularly the NALP3 inflammasome, or you can say the NLRP3 inflammasome, is very important for producing interleukin-1 beta. The interleukin-1 beta is present in pro-interleukin-1 beta form, which when cleaved by the caspases, it becomes a mature interleukin-1 beta. So we found that endographylide is able to suppress the caspase 1 as well as the inflammasome NLRP3 and P2X7, which also suppress the interleukin-1 beta production. So it is very important to see the holistic aspects of the pro and anti-inflammatory cytokines. So for that, we measure the TNF alpha, interleukin-1 beta, and TGF beta, interleukin 10. As you know, the TGF beta and interleukin 10 playing against the inflammatory signals. So we measure the anti-inflammatory cytokine TGF beta as well as interleukin 10 over here. And you can see the endographylide is able to enhance the TGF beta level in the endographylide and LPS treated group as compared to the LPS alone group. Interleukin 10 is similarly enhanced by the endographylide and as compared to the LPS. Similarly, in the pro-inflammatory cytokine level, the level of TNF alpha and interleukin-1 beta was significantly enhanced in the LPS treated group. When treated with the endographylide along with LPS, we found the reduction in the TNF alpha and interleukin-1 beta level. So we concluded that endographylide significantly altered LPS-induced expression of TNF-alpha, interleukin-1 beta, interleukin-10, and TGF-beta in cell supernatant of primary mixed culture and in vivo the mouse prefrontal cortex. Now the question is, as you know, the TNF-alpha causes apoptosis. So whether the endographylide suppress the apoptosis or not. But as you know, the BAX protein is pro-apoptotic in nature and BCL2 is anti-apoptotic in nature. So we measured the BAX expression in the LPS treated group and compared it with the LPS and endographylide treated group. We found reduction in the BAX expression, while BCL2 expression, which is anti-apoptotic protein, was significantly enhanced in endographylide treated group. We further measured the FZV staining in prefrontal cortex region. Here also we found that the mean fluorescence intensity of the cells positive for FZV was significantly reduced in endographylide and LPS treated group as compared to the LPS. Now coming to the different aspects of the neurodegenerative proteins. As you can see, the neurodegenerative proteins stimulate the neurodegeneration process. And these proteins are the amyloid protein precursors, beta amyloid, phosphorylated tau. And we measured all these neurodegenerative proteins in all 4 groups, that is control, LPS, endographylide treated group, and LPS plus endographylide treated group, in vitro as well as in vivo. And both the data said that the amyloid protein precursor, phosphorylated tau, and beta amyloid is significantly reduced by the treatment of the endographylide as compared to the LPS treated group. Now, we measured the beta secretase and post-synaptic density protein 95. As you know, beta secretase is also a neurodegenerative enzymes which enhances the neurodegeneration process, while PSD95 is post-synaptic density protein which strengthens the synaptic strength. We found that beta secretase level was significantly enhanced in LPS, but it was suppressed in the LPS and endographylide treated group, while the synaptic density protein 95 was significantly enhanced in the LPS and endographylide treated group as compared to the LPS. We further measured the memory scores by the atom radial major assay, and we found that LPS treated group showing the higher scores of the error, while when it was treated with the endographylide, the memory error score was significantly reduced. So therefore, we concluded that endographylide is an important terpenoid or labdent diterpenoid, which can significantly reduce the LPS or inflammatory event, and it can be able to enhance the working memory. And therefore, we concluded that endographylide prevented neuroinflammation associated neurodegeneration, and endographylide improved synaptic plasticity markers in cortical as well as hippocampal region, and it could be a novel pharmacological countermeasure for the treatment of neuroinflammation and neurological disorders related to memory impairment. With this, I come to the end of my talk. I would like to acknowledge Sudeshna Das, who has done this studies. He was a student, a PhD student who performed this study. I would like to thank Madam Dr. Sasiwala Singh, Hyderabad, and former DG Life Sciences, DRDO. Under her supervision, this work was performed. I was the co-supervisor for this study of Dr. Sudeshna Das. I would like to thank Director DPAS for providing the infrastructure and all the support for conducting this study. And of course, my funding agency, Defense Research and Development Organization, in the form of a project DEEP 265. Thank you. Thank you all for giving me this chance.
Thank you so much, Dr. KP Mishra. Such an interesting talk on a plant-based active principle in counteracting neuroinflammation. So I would request the participants to please put their questions in the chat box and interact with these expert lecturers, these speakers at the end of the session. So now, thank you, Dr. Mishra, once again, and moving on to our next speaker. Am I audible? Hello? Hello?
Yes. Yes, ma'am. Thank you.
Okay. Thank you. So our next speaker is Dr. Meetu Wadhwa. Dr. Meetu Wadhwa is, I'll just give a very brief introduction. She is M.Pharm from Bhimrao Ambedkar University, Lucknow, and also she's done her PhD from DIPAS, our Institute. She has been our research scholar. Presently, she's a postdoctoral scholar in the University of California, San Francisco. So Dr. Meetu today is going to talk about some of these countermeasures, caffeine, modafinil, and minocycline, in ameliorating the sleep deprivation-induced neuroinflammation. So I hand over the floor to Dr. Meetu.
Thank you so much, ma'am. So now I'm going to share my slides. So can you see the slides?
Yes.
So first of all, I would like to thank the Fips organizing team for giving me this platform to present this work. So I'm sure most of us love to sleep, but due to increasing day by day stress, heavy workload, and increasing use of the electronic appliances, people are not able to take proper sleep. That mostly affect their health and mental well-being. So today we are going to discuss more about how sleep deprivation affects the cognition and the related countermeasures. So our main focus will be on the neuroinflammation. Before going on to the real problem, sleep deprivation, let's try to understand the sleep. In brief, sleep is a state of immobility with reduced responsiveness. It can be distinguished from coma by its rapid reversibility. So we sleep daily. Why we sleep? Previously, it is believed that sleep gives us energy gain from day's activity. Is it true? No. In reality, if we see, the energy gain during sleep is very small. So why? Why we sleep? So sleep is very important as it is necessary for the health and survival, brain development, and sustaining cognitive skills. There is a range of 2 hours to 20 hours of sleep time per day in different mammals. If we, among which in humans, 8 hours of sleep per day is mostly seen. Generally, people ask how much sleep is required. The answer is there is no particular set of sleep time required. So it varies from person to person. It depends on different age groups. Like in infant, the total sleep time is maximum as compared to the adults. Coming back to the real problem, the sleep deprivation. What do we mean by the sleep deprivation? So in humans, sleep less than 8 hours in 24-hour period is sleep deprivation, while in rodents, any time duration the animal kept awakened forcefully is called sleep deprivation. In general, we think there is no side effect of sleep deprivation, there is no negative consequences, but this is not true. So there are several studies in humans which have shown that sleep deprivation decreases the attention, memory performance, and decision-making power. Also, in rodents, sleep deprivation has shown decrease in body weight, food intake, and cognitive performance deterioration. So this is not true that sleep deprivation doesn't affect health. Sleep deprivation affects different major organs in our body. So all of these organs are important, but today we will focus mainly on the brain and immune system. If we see brain and immune system together, we will see that there is a bidirectional communication between these two systems. The cerebral microglia, which are the resident immune cells of the brain, release several sleep regulatory substances. These substances act on the hypothalamus, basal forebrain, and regulate the sleep. But whether sleep deprivation affects this brain immune interaction or not? If yes, then how? With this focus, so microglial cells is actually the glial component of the brain, which release the increased amount of the pro-inflammatory cytokines upon activation and which ultimately affect this brain-immune interaction. Under physiological condition, these microglia survey the surrounding, and if it finds any harmful stimuli, they act as a phagocytic cells and do the neuroprotection. But what happens under the stressful condition? Under stress, the microglia become more activated, release increased amount of the pro-inflammatory molecules, causing neuroinflammatory response. If we see the literature, there are a number of human studies which have shown the increased amount of pro-inflammatory cytokines in plasma at different sleep deprivation duration. Yet, the cellular signaling that trigger these changes remain to be determined. So with this question, we hypothesize that there might be a role of neuroinflammation in sleep deprivation induced cognitive impairment. So we used automated sleep deprivation apparatus that is developed by our own lab in DePass. So this apparatus is worked by Animate software. It detects the animal activity. If the software detects the animal immobile for more than 30 seconds and freezing more than 5 seconds, it gives signal to the interface and shaking controller, causing some shaking and vibration, which makes the animal awake. In this way, we made the animal sleep deprived and evaluated behavioral as well as the neuroinflammatory changes. Following standardization of the sleep deprivation duration from 24-hour, 48-hour, and 72-hour, we found that 48-hour of sleep deprivation effectively maximum produced changes in the spatial memory decline, along with the increased amount of the pro-inflammatory cytokines and increased microglia activation as seen in this correlation analysis. So after a positive correlation analysis found between microglia activation, pro-inflammatory cytokines, and spatial memory, we come to know that 48 hours of sleep deprivation activate the microglial cells, which disturb the inflammatory cytokines level, causing spatial memory impairment. As we all know that neurogenesis is useful for the cognition. So what happens to neurogenesis in sleep deprivation and neuroinflammation? In literature, it has been shown that sleep deprivation and neuroinflammation is detrimental for the neurogenesis. And in neuroinflammation, neurogenesis is decreased via modulation of the inflammatory cytokines. So it will be, there is a possibility that sleep deprivation induced neuroinflammation affects the cognition via changes in neurogenesis. Till now, we have understood the problem, but our aim is not only to find out the problem, but also to find out some therapeutics. So we focus on two categories of therapeutics. First is selective microglia activation inhibitor, minocycline, and second is the psychostimulants, caffeine and modafinil. Minocycline is a well-known antibiotic, although its anti-inflammatory action is not well known, but it has been shown to attenuate microglia activation in several neurodegenerative disorders. On the second hand, caffeine and modafinil have been shown to promote adult neurogenesis during 48 hours of sleep deprivation in one of our previous study from our lab. Although all of these three therapeutics will be effective against the neuroinflammatory changes during sleep deprivation is still not known. Before going on to the molecular level or cellular level changes, it is important to see the behavior changes with the treatment of these drugs. So for this, we tested the spatial memory first with the help of Morris Water maze, and we found that minocycline treatment at 5 mg per kg dose effectively improved the spatial memory changes during sleep deprivation. Also, caffeine at 60 mg per kg and modafinil at 100 mg per kg improved the spatial memory test parameters such as latency, path length, path efficiency, number of entries, and time spent following sleep deprivation. Cytokines are pleiotropic in nature. They affect sleep as well as affected by the sleep.
Meetu, can I interrupt you for a minute? Can we wind up in a few minutes, 2-3 minutes? Because we have 2 more speakers.
Yeah, sure. Just give me 2 to 3 minutes.
Okay. Thank you.
Thank you. So we measured a few inflammatory, pro-inflammatory, and some anti-inflammatory cytokines and found that minocycline, caffeine, and modafinil is effective against the disturbed balance of these inflammatory cytokines during sleep deprivation. Glial cell activation is a significant source for these upregulated pro-inflammatory cytokines. So we next evaluated the gliosis in hippocampus via counting the microglial cell count at different stages, resting, intermediate, and activated. Interestingly, we have found that minocycline, caffeine, and modafinil effectively decrease the number of activated stage microglial cells with increase in the resting stage microglia. There is no change in the intermediate stage microglial cells during sleep deprivation. So neurogenesis is useful for the cognition. It involves different stages like proliferation, differentiation, and maturation. We evaluated this stage by different markers, and we found that sleep deprivation negatively affects these stages, and minocycline treatment is helpful in ameliorating these changes during sleep deprivation. So to sum up, we come to know that sleep deprivation affects the cognition via neuroinflammation. Microglial cell activation serves as a putative mechanism. Minocycline, caffeine, and modafinil served as an effective therapeutics against the above consequences during sleep deprivation. With this, I would like to thank Director DePass, my supervisor, and the entire DePass team for supporting this research. And at last, I would like to thank the Fips organizing team for giving me this opportunity to share this work. Take-home message is sleep well, be well. Thank you.
Thank you, Meetu, for finishing in time. And I know we have sleep deprived you a bit on pulling you out on a Sunday morning. So now I call upon my next speaker. I have great pleasure in introducing to you Professor Uday Pandey. Professor Uday Pandey is an associate professor, Department of Pediatrics, Human Genetics, and Department of Neurology. He is an associate director, Children's Neuroscience Institute, Children's Hospital of Pittsburgh. Dr. Pandey has done his graduation and post-graduation from Dr. Ram Manohar Lohia, Awadh University, UP, and his PhD he has obtained from Sanjay Gandhi Postgraduate Institute of Medical Sciences, UP. So he is a very celebrated scientist. He's had a checkered career and has received many awards and honors. Because of paucity of time, I'm not reading out those awards and honors. And he has published several articles. In fact, he has a very high citation index. He has 17,451 citations, which are with an H index of 32. He's published books and monographs as well. So without standing in between you and Dr. Uday Pandey, I invite you, Dr. Pandey, to deliver your lecture.
Thank you very much, Dr. Panjwani. I really appreciate such a generous introduction. Can you see my slides?
Sure, yes, we can see.
Perfect. And it's a really pleasure to be part of the symposium. And I loved previous talks. And thank you so much for inviting me and keeping us on track with your emails. Really appreciate it. So my lab is located in Pittsburgh, and Pittsburgh City is known for a number of tunnels and bridges. So there are 446 bridges and tunnels in Pittsburgh. I think this is the maximum number in the world. And the whole city is connected through these tunnels and bridges. And our university was founded in 1787. So it's a pretty old university and medical center. This is a cathedral of learning, a long, tall building where on top floors we have rooms for every country. And one of the room has been dedicated to my beloved country, which is India. So please visit if you ever get a chance to come to Pittsburgh. So for today's talk, what I would like to accomplish is I would like to give a brief introduction about amyotrophic lateral sclerosis and our recent efforts that we took to identify genetic modifiers of ALS using in vitro and in vivo model systems. And then some work towards understanding the molecular mechanisms associated with ALS. So what is this ALS? Why this funny name? So this disease is also known as Lou Gehrig's disease. And Lou Gehrig was a famous baseball player from New York City. And he was diagnosed with Lou Gehrig's disease when he was at the prime of his career. And because of this devastating disease, he could not continue his sports career. Another celebrity who was a football player in New Orleans, Steve Gleason, he was diagnosed with ALS at the age of 34. And he could not continue. Within 2 years, he became wheelchair bound. And now he cannot move any part of his body except his eyes and a couple of fingers to control his computer assisted device. So this term ALS is called, came from a Greek term, which is called A means no, myo means muscles, and trophic means nourishment. So there is no muscle nourishment. Lateral, it defines the location of nerve cells that signal and controls the muscles, and sclerosis means scarring or hardening of the degenerative regions. So this disease is caused by loss or dysfunction of upper and lower motor neurons, which comes with muscle atrophy. And normally people die within 3 to 5 years after being diagnosed due to respiratory failure. So at present, there are no effective therapies available for ALS, and this disease is poorly understood. So a lot need to be done in order to understand the molecular mechanisms. So as a little bit more about this disease, there are two components. Most of the ALS patients, they are sporadic. That means randomly in a family, somebody would get diagnosed with ALS. There are about 10 to 20% of cases which are familial in nature. We know that there are genetic mutations which right now there are several of them. First one was identified in 1992, and then recently we had so many ALS causing genes that were identified. But the message from this slide is sporadic and familial cases, they are clinically indistinguishable. That means a neurologist cannot distinguish them unless you tell your genetic report or your family history. And they share common pathological features. So the type of ALS that we are studying is caused by mutations in this gene, which is an RNA binding protein called fused sarcoma. And this is an DNA-RNA binding protein, which is predominantly nuclear. And there are many missense mutations have been reported in this gene. This gene has been shown to be involved in regulating different aspects of RNA metabolism. And because of pathogenic mutation, a small, tiny fraction of this protein goes to the cytoplasm, where it forms aggregates and it becomes incorporated into cytoplasmic stress granules, which I will talk a little more in next few slides. So why fuss? the age of onset of patients with ALS with FUS mutations is way more shorter as compared to sporadic and other types of ALS. So normally you see this in this graph, the red bars, they show you that patients have been diagnosed in very early stages, and people call it juvenile onset ALS. So suggesting that if we can find molecular pathways, it would be very interesting and applicable to other types of ALS. And you can see a survey done by one group where they found about 61% of FUS cases, they are less than 40 years of age. So what happens when you have mutations? So in FUS protein. So FUS is predominantly nuclear protein, and because of mutations, it goes to the cytoplasm, and generally every cell has a defense mechanism, which is called stress granule, RNA granule formation. And if you take just control cells and challenge them with any stress, they form these granules, and they try to halt translation by sequestering all actively translating messenger RNAs. And when stress condition is over, they try to resolve these structures, and then translation resumes. But mutations are different types of mutation, aging. This is a type of stress that these cells go through, and majority of the components of these stress granules are RNA binding proteins, and most of them have been linked with ALS pathogenesis as well as other types of human neurodegenerative diseases. So my lab specializes in using complementary model systems, including fruit flies that we use in conjunction with mouse models, induced pluripotent stem cells, and postmortem tissues. So there are many advantages of using fruit flies, including the availability of tools, shorter lifespans, and many of the important pathways, including sleep cycle or circadian rhythm, have been mapped in Drosophila, and then later on people found it to be true in human patients, here a human being. So we developed multiple models of ALS by introducing ALS causing genes in Drosophila. And we targeted expression of these transgenes to different tissue type ubiquitously. And what you are seeing here is Drosophila I, which is control I, and this is wild type FUS, human FUS, and these are three different types of mutations. And you can see that there is these eye tissues are made up of neurons. So you can easily see that there is a necrosis and there is external eye degeneration, suggesting that these point mutations are highly detrimental to these neurons. And same thing happens with other types of ALS models. You can see that there is a neuromuscular defect in different types of ALS. And we published all these findings over the last 10 years. So for today's story, what I would like to talk about is we took an unbiased approach where we used a conditional model. We drove expression of a wild type or mutant FUS in Drosophila brain, and then we took out their brains. We extracted RNA and performed RNA sequencing just to look the impact of expression of these expression of mutant FUS on brain transcriptome. And what we observed here is interesting, that about 64% of the transcriptome is downregulated in response to expression of mutant FUS, whereas 36% of the transcripts are upregulated. And what we found is about 280 transcripts, they were specific to mutant FUS. And there were like 131 that was different between mutant wild type FUS and mutant FUS. So what from here, what we did is we took a human data set where somebody performed similar analysis in human cells. And we compared our transcriptomic analysis that how many of these transcripts are conserved in human versus flies. So one of the transcript groups that came out, and they were the topmost one, is called a DDX group, or dead box helicages group. So we started working more towards this, and what we did was we this is heat map showing downregulated and upregulated transcripts in Drosophila and humans. So what you can see here is we measured, we decided to look for the levels of DDX protein here. And here you can see these double bands, which is 2 isoforms of DDX17. And you can see in 2 different ALS causing mutations, DDX17 has been drastically downregulated. And not only in human embryonic kidney cells, but also what we found is in ALS patient iPSC neurons. And these iPSC neurons were generated from patient cells, from fibroblasts. And you can clearly see here that there is a significant reduction in DDX17 levels, which is more obvious in the quantification panel. So what does it mean? What is the physiological consequence of this change in context of ALS? So DDX17 protein is a multifunctional protein, and it is involved in regulating different aspects of RNA processing. It's a predominantly nuclear protein, and it has been shown to be involved in regulating different RNA processes, including liquid phase separation and other processes. And it has been shown to be recruited in DNA damage site, and it interacts with other DNA damage causing proteins, including HDAC1. So in our initial work, we first looked for the relationship between cytoplasmic granules that
would show your name instead it would show mine, sir.
In our mammalian cells. So this is wild type cell, and you can clearly see that FUS, which is green, is predominantly a nuclear, and this is mutant cell. You can see that these punctate structure, they represent cytoplasmic aggregates, and these aggregates, they co-localize in the bottom panel with arrows with DDX17. And this is not only true in HEC cells, but also in neuronal cell type, which is more obvious here. So we asked whether it is true or whether it is a consequence of overexpressing wild type or mutant protein in these cells. So we used iPSC neurons from patients where we get endogenous mutant FUS expression. And what we found here is you can clearly see in isogenic control where these are a punctate structure that represent cytoplasmic stress granule because they are positive for G3BP, which is marker of a stress granule and co-localizing with mutant FUS. You can clearly see co-localization of DDX17 with mutant FUS in the cytoplasmic stress granules. So now, next question is whether DDX17 and FUS, they have any biochemical interaction. So in order to do that, we took FUS construct and we made a bunch of point mutations in this. So first mutation that we made was in the RGG domain, which has been shown to be part of many proteins, which is similar to DDX17. We also made mutation in the RNA recognition motif. And we performed a biochemical analysis where we transfected cells, mammalian cells, with HA tagged constructs. And what I'm showing you here is there is an interaction of FUS with a DDX17. So long story short, after a bunch of analysis, what we found is if we mutate RGG1 domain, this interaction between FUS and DDX17 is gone. So there is RS1 mutation, which is in RGG1, and RS4 mutation, which abolishes all four, completely abolishes RGG domain structures. So now what we did was we started investigating this further, and we found that upregulation of DDX17, it reduces the insoluble formation of these cytoplasmic FUS aggregates. And that we found using two different approaches. Number one, by IF, what you can easily appreciate in this quantification panel, that there is increased punctate structure, which represents FUS in insoluble aggregates. And this also is true by biochemical methods, where we found that there is a clear reduction in the insoluble aggregation, and that co-localizes with DDX17. So now, what is the molecular mechanisms? So in order to further understand the molecular mechanism, we found it has been previously shown that there is increased DNA damage and double standard break formation, and that has been linked with a mutant FUS associated ALS, and it has been shown in ALS FUS patient iPSC neurons. So and then also DDX17 has been implicated as an epigenetic regulator, which is recruited to DNA damage site by microradiation. So now what we did was we used two different markers of DNA damage, which is gamma H2AX and 53B1. And what we found is there is a significant increase in DNA damage in iPSC neurons, which is true, which can be also seen by Western blot analysis. So when we overexpressed DDX17, you can clearly see that there is a reduction in the double distended BRAC and the staining of these two DNA damage markers. So now I would like to just go to the next slide, because I think I'm running out of time. So we looked for the consequences of DDX17 overexpression in iPSC neurons, whether it can protect against degenerative phenotypes. So previous studies have shown that apoptosis is a secondary response to DNA damage. Whenever you do DNA damage, you get apoptotic signals upregulated and cells die with apoptosis. So we used markers of apoptosis, and we looked for whether there is an increase of apoptosis in these iPSC neurons. So in mutant FUS expressing cells, you can clearly see that there is an increase in this apoptotic marker. And when we overexpress DDX17, the apoptotic staining goes down, further suggesting that DDX17 overexpression protects against neurodegeneration. So now, last slide that I wanted to show, what is the in vivo relevance of these findings? So here in the first panel, you can see there are two mutants that we expressed in fly eyes. And this is control eye, where you can easily see there is a nice architecture, and expression of FUS leads to external eye degeneration. So when we knock down DDX17 in these fly eyes, you can see there is enhancement in external eye degeneration. But if you overexpress DDX17, you can see there is a suppression in the external eye degenerative phenotype. So we did a bunch of quantification, biochemical analysis to further support these findings. And lastly, we performed motor function assays in these animals by driving expression to neurons. And we found that the motor function of these animals expressing mutant FUS protein is restored when you overexpress DDX17. So whether it is true for all type of ALS, is not, because we did the similar experiment with other types of ALS, and we found that it is specific for only FUS mediated ALS. So this is just a summary that we identified DDX17 as a modifier of mutant FUS toxicity, and we believe that it is it helps or suppresses neurodegeneration by DNA damage, regulating DNA damage process. So this work was primarily done by my graduate student Tyler Fortuna, and I'm grateful to my collaborators and funding support, and I'm very thankful for listening to me. I'm ready to take any questions during the break. Thank you.
So you'll all agree with me that Professor Uday Pandey has taken us on this lovely journey of ALS. And thank you so much, Professor Pandey, for sparing this time on a Sunday morning in US. We are indeed very grateful to you. So since we are running short of time, we'll quickly move to the last speaker, Dr. Anita Jagota. I'll just give a very brief introduction. She is done BSc and MSc from Delhi University and done her postdoctoral work in CCMB Hyderabad and also University of Massachusetts, US. She joined Hyderabad University in the year 2000. Presently, she's the head Department of Animal Biology in University of Hyderabad. And her lab is working on understanding the mechanisms involved in neural regulation of circadian timing system and linking its complexities to various physiological processes. She has more than 50 papers in high-impact journals, and she is a recipient of awards and honors, and is a member of many national and international bodies as well. So I would request Dr. Anita to immediately start her talk. And if you could restrict to 10 minutes, because I think we are getting late for the valedictory function.
Okay.
So that'll be nice. So over to you, Dr. Anita.
I'll share my screen. I'll share my screen.
Yeah.
Can you see my screen?
Yes. Yes, and you can, yeah.
Is it clear?
Yes, it is clear.
Yeah.
Yes.
First of all, I would like to thank the organizers, Professor Dr. Shashi Bala Madam and other organizers, including Dr. Khatri, and to include me and give me this opportunity. And it's my really great pleasure to speak in the session, which is chaired by one of my very good friend, Professor Usha, Dr. Usha, and I'm seeing her after many years on the virtual screen. So today, and I understand we are running out of time, so I will try to be as fast as possible and give you the glimpse of the work done in my laboratory, and we can always interact later. So I am presenting here today age-induced neurodegeneration in SCN, therapeutic effects of hydroalcoholic leaf extract of Withania somnifera, which is called commonly as ashwagandha. These are the various kind of projects going on in my laboratory. I have put this slide for the interest of the younger students who may be looking for the opportunities joining various laboratory. So in my lab, we are focusing on identification of biomarkers of aging, neurodegeneration, Parkinson disease model, AD model, sleep deprivation, and we study anxiety and deprivation in sleep deprived rats, and also certain aspects of inflammation, cognition, memory, and we are also into the therapeutic interventions using the various methods, one of which today I'm going to talk.
Yeah, good evening. Good evening. How are you?
And yeah, that's important.
How do you do? How
And
is
there
your
are
family?
others. So
And how is...
next, the biological clocks synchronize their activities with the day-night cycle caused by Earth's rotation. And these biological clocks, they are remarkable timing system as they measure local time of the day and lapse of the time, and the biological clocks are synchronized to the environmental cues. Various activities in our body, they peak at certain time of the day. Therefore, it is very important to understand the genetics, physiology, genomics, phenomics, and the metabolomics, the proteomics and epigenetics involved. So we are looking at different angles as it has direct medical applications for planning many chronotherapies for many disorders like sleep disorders, neurodegeneration, many physiological disorders, mental disorders, etc. Technological and development or small and medium scale enterprises, like certain formulations from plant need. For that, the plant part has to be picked at certain time of the day to have the desired product, and industrial application, especially for bioremedial processes. These biological clocks are localized in the SCN in the case of the mammals, and they regulate all the peripheral tissues which also act as the clock, and they are called as peripheral clocks. And the SCN is the suprachiasmatic nucleus, is the main pacemaker. This SCN receives the light through retinohypothalamic pathway and regulates the release of the hormone melatonin, which is neurohormone, through pineal. And I'm skipping some of the details to save the time. The SCN regulates rhythmicity via interactions of clock genes and their protein products, characterized by transcriptional and translational feedback loops. This is the central loop and this is the auxiliary loop. The BMAN and clock are the positive regulators, whereas PER and CRY and the REV-ERB alphas, they are the negative regulators of the clock, and then there are many other players. Increase in elderly population is the major concern in 21st century, because especially the neurological disorders, they are the diseases of the elderly, as you can see. And the age-related pathologies have been attributed to the circadian dysfunction. Due to aging, the reduced physiological function, leading to neurological disorders, leading to reduced social cues, reduced exposure to light and environmental cues together results in the disrupted circadian function and sleep disorders. You can see the normal sleep-wake pattern advanced and fragmented with the aging. The aging and the chrono description, many labs are working internationally, and we are one among them. There are various reports from our labs and also from across the world. I have only included few here. So, but I would like to highlight is, in the young, the number of molecules which are interacting, they are more, and the stoichiometry of interaction and the buffering capacity decreases with the aging. You can see the number of molecules that could be interacting decrease with the age. So synchronize the desynchronized rhythms with age and neurodegeneration is the biggest challenge. Can these age-related changes in rhythmic pattern be prevented in normal aging using antioxidants, especially melatonin and herbal formulations? We have done a lot of work with effect of melatonin on various parameters in SCN upon aging. Instead of going in the details, I have only put the titles of the publications. So effect of melatonin, we have shown that in the it helps in restoration of the daily serotonin rhythms upon aging, and especially if given early in middle age, and also in differentially regulating the clock gene expression, the chronomics of serotonin metabolism, SOX1 rhythms, and many antioxidant enzymes in lipid peroxidation in liver. And then the NO rhythms we have shown in the case of the peripheral clocks, and also in the proteomics, like we studied the proteomic profile and the number of proteins which were disoriented with aging. We could restore the circadian pattern with the therapeutic interventions using melatonin. And then there were other methods like restricted feeding and all. Then we wanted to look at the herbal formulation, and we studied curcumin effects, and we could show that in SIRT1 expression it helps. And also in the kidney, we had studied the immune gene expression, and in other dysfunctions also in the clock, the curcumin had helped. So here I only wanted to say the cell damage and the reactive oxygen species, the cell damage by ROS increases with the age, and we have to regulate this damage to slow down the aging process. And the antioxidant Withania somnifera has withanol, withaferin, and withanolides, which are non-toxic, neuroprotective, anti-stress, antioxidant, anti-carcinogenic activities, and also scavenging activity was used for the current study. There are both people studying leaf extract and root extract. We preferred leaf extract with the hydroalcoholic leaf extract because it has the components of both aqueous extract as well as the alcohol extract. And and we found that in the leaf extract, the scavenging activity was 80% as compared to the root extract, which showed only 35%. Then we have used various age groups, 3-month, 12-month, and 24-month old male Vistar rats. And hydroalcoholic leaf extract formulation was used, 100 milligram per kg body weight, and the vehicle being carboxymethyl cellulose, 0.5%. Then these are the various serotonin metabolism components which were studied in one go using reverse phase HPLC with EC detector. And these are the standards. And then we also studied MT1 and MT2 receptors, G protein coupled receptors present on the SCN. And these are only dissociation curves. And we could find the black bold lines are the controls that with the age there is disintegration in the circadian rhythm pattern. Either they are getting delayed or they are getting advanced. Like you can see in this particular case, in 24 months, the PER1 expression is getting advanced in the old age, whereas it is getting delayed in the 12 months. And this is the green dotted line.
Anita, I'm so
Yeah,
sorry
only
to
one
interrupt you.
example. Sure.
We can
Yeah,
wind up.
only one example to
Okay.
explain.
Thanks so much.
So the green line, you can see that with Withania somnifera intervention, you can see the phase of the rhythm is restored. It is again showing maximum at the same time point, that is CT6, the mid-subjective day. So similarly, I'm not going to the details of all the molecules studied. This is all published. So like if anybody interested can go to the publication, I can send PDF. So this is the mean 24-hour levels showing the restorations, and this is the we had done the correlations, and where the red means the negative correlation and blue means the positive correlation of the various molecules studied. And you can see at a glance that the expression pattern and the correlation pattern is changing with the age. And with the Withania somnifera intervention, you can see these green rings I have put. The restorations are quite close to the 3-month in the case of 12 months. And in case of 24 months, again, you can see it's a very beautiful restoration which we have achieved. We have published this recently. And this is the MT1, this is the CERT1 and NRF2 protein studies also. Here also we established the similar things. And then we studied the locomotor activity rhythms, and we could establish with Withania somnifera these disoriented rhythms have been restored. And this is the last slide. So I just quickly have walked through this. So, and you can see we could get differential restoration using ashwagandha, for the clock gene expression, gross locomotor activity, serotonin metabolism, and melatonin receptor expression and clock gene expression. And this is my team. I must thank my student Kaushik who did this work. And without funding, nothing is possible. And the project students we have a MOU with the University of Munster. So I do get students from there, and also our own project students plus our PhD students. And this is my current team. Thank you very much. And this is our beautiful campus. Thank you.
So thank you so much, Dr. Anita. No, we enjoyed your talk, such a nice talk on our biological clock, and with so many of these countermeasures in restoring the biological clock. So thank you so much. So with this, we come to the end of this session. For the paucity of time, I don't think we can take any questions. So indeed grateful to Dr. Anita, Dr. Uday Pandey, Dr. KP Mishra, Dr. Dipendra, and Dr. Meetu. So thank you one and all. And we've come to the end of the session. I hand over the floor to the organizers for the valedictory session. Thank you.
Thank you.
Chairperson, Dr. Usha, and also I want to thank all the speakers who made excellent presentation, and thank you so much. I think now we are at the end of this 3 days conference. We are now going to start valedictory session. डॉक्टर राव ने ज्वाइन किया है तो दिखाओ सर गुड इवनिंग डॉक्टर राव वेलकम
Yes, good evening, madam. I'm joined already.
Thank you so much, sir. Thank you so much.
Thank you, madam. Thank you, madam.
क्या प्रोग्राम क्या था सबसे पहले मुझे ही बोलना
Hello, Dr. Sambasiva Rao. Good evening. Nice to see you, Selvamurthy here. How are you?
Namaste, sir. How are you, sir?
Fine, thank you. Pleasure seeing you. How
Yes,
is
sir,
your
thanks.
university doing, Mizoram
Yes,
University?
it's very good, sir. It is doing very well, sir.
I'll visit sometime soon. I'll come to
Yes,
your
sir. Actually, I have to give a one memento, sir. It was with me long back. It was actually you are supposed to come as one of our association conference. No, you are not turned up actually that time. You could not come. So that memento is still with me. That honorary fellow award actually, it was still with me, sir.
I have to come. You tell me when I have to come. I'll be there.
Yes, yes, sir. Sir, you are still at Delhi, sir?
Yes, yes. I am with the Amity University now.
Yes, sir. Yes, sir. I know that you informed me once, sir. Actually, yes, sir. Nice, sir.
Nice seeing you.
Nice meeting, sir. Thank you.
So good evening. I think we have come to the end of 3 days conference, and it was attended by a large number of people on each day. That gives me really good, good feeling that in spite of being online, I was not expecting attendees to be in such large number. That has really made us feel better and made us feel more motivated that whatever we did became part of a large number of physiologists, not only animal physiologists, but human, and even there were people from other walks of science. So really great. And it gives me immense pleasure to welcome Professor Rao, who is Vice Chancellor of Mizoram University. I know him for almost about a year or so, and I keep reading about his accomplishment in the university because the way he is steering the university system, and he has brought in a lot of projects, a lot of papers, and even his faculty members are putting a very good progress in terms of publication, in terms of number of projects, and the law, and so much of decorated faculty member he has. Really great, sir. And the way he has been sort of, I've seen him, the way he pushes everyone, and then everyone performs to the best of their ability. Though you all know him, but I would just like to tell you a few things about his accomplishment. As I said, presently he is a Vice Chancellor of Mizoram University, Central University. He is also the General Secretary of Association of Biotechnology and Pharmacy, and also the editor of Current Trends in Biotechnology and Pharmacy. So, sir, we are happy that you are, you are part of pharmaceutical sciences, where we are also hosting this conference in Naipur, Hyderabad. So that gives me more, affiliation to what is your
Actually,
expertise.
madam, it is not a forum to inform you that actually I'm shortlisted for Naipur, Hyderabad director post, but I did not come for the interaction actually on the
I
day.
know that. Yes, sir. Thank you.
Yes.
Yeah. So he held various prestigious positions in many universities, not only in India but abroad. He is a visiting scientist at VCU Medical College, USA. Also, he is a visiting scientist at University of Germany. And not only this, he by profession he is a man with pharmaceutical background, but he is also have been able to establish a lot of academic program not only in India but outside India also. Some of the programs which were established outside India is PhD in biotechnology with Republic of Panama, then PhD Master of Science in Computer Applications in Tanzania, then tuning program of MS Biology with University of West Georgia, and also he has a lot of such kind of activities which he has been doing it. If I have to read everything, it will run into pages. And then he has experience 35 years in research in the field of environmental toxicology, physiology, environmental and aquaculture biotechnology. He has 7 patents to his credit. Really great, sir. And also he has received several prestigious awards, and he is a very decorated professor, both in teaching and both in research. And also, the one thing which is really great about him, that he was the Vice President of Indian Society of Comparative Animal Physiology, SCAP. So really great. In last almost 2 months, we have been looking for the, office bearers and the member of SCAP and SEPI. So we have found one today, besides few which we know. Really great.
Yes,
so
madam.
thank you, sir, for joining us at a very short notice.
Yes, madam.
We feel obliged. I will just recap what all Fips is, since you are here for the first time. So this Fips is a Federation of Indian Physiological Society, and this federation came into existence about 3 decades ago, and there were few pioneer physiologists in India who came together and formed this Federation of Indian Physiological Society. It's a consortium of 4 physiological society: PSI, Physiological Society of India; SEPI, Society of Animal Physiologists of India; APPI, Association of Pharmacologists and Physiologists of India; and SCAP, Indian Society of Comparative Animal Physiology. So these are the 4 societies which are part of Federation of Indian Physiological Society. However, today we have one more society which will be part of Fips, and we welcome Professor G.K. Paul, who is the President of Association of Physiologists of India. So this is another society which will be coming under the umbrella of Fips.
Yes.
Then We had organized 3 days conference. We had a lot of participants. And we also had some oral presentation where, depending on the performance, we had jury members, and the presentations were evaluated, and some of the awardees also will be honored during this valedictory function. So this is about Fips, and I want to thank each one of you who have joined. There are many people who are there, a lot of bright young students, the office bearers of Fips and of different society. I want to thank each one of you for joining us for this valedictory function. Now I will request chief guest, the Professor K.R.S. Sambasiva Rao, to kindly address the gathering.
Good evening, madam. Good evening, madam. Good evening, madam Shashibala Singh and Professor Salamurthy sir, and other presidents of the other associated associations, and all the invited speakers and faculty members of NIPER Hyderabad. It was a great opportunity for me to be participating in this 9th annual conference and also international symposium to attend in the valedictory function. I had a wonderful interaction with the madam since several years, and it was really a memorable time. I would have been allowed to come physically there during this convention if I would have known earlier. But anyhow, it is a good opportunity for me to interact with all of you. And it was the National Institute, which has been one of the best pioneering institute, National Institute of Pharmaceutical Education and Research. Hyderabad is one of the best institutes in the country, and it is growing very well in one of the authoritative institute in the country, particularly in pharmaceutical research. And it was the vision of the institute is to serve as a leadership in the particular pharmaceutical education, and to become as a global institution for higher learning and research in pharmacy. And this is I'm very happy that the 3-day meeting of 9th International Symposium of Federation of Indian Physiological Societies has been organized by NIPER Hyderabad very successfully during this March 25 to 27 on the theme of reconnecting physiology and nature of a healthy life. I understand that the conference invited many experts from various fields, especially from partnering societies such as PSI, SAPI, APPI, and SCAP. The conference has various discussions and knowledge transfer from eminent scientists. Madam was describing about that. And it was various categories of areas like biological sciences, pharmaceutical sciences, and physiology, and advanced physiology, pharmacology, and all these areas were discussed, and 350 national and international participants were participating in this international event. It was really wonderful, and I'm very happy to see that. The Fips International Symposium is very important because I've been listening about this since many years, and I've been following the various symposiums which have been organized by Fips for the past several years. I could not get an opportunity to participate in any of them, but in the context of physiology, particularly in understanding the human's abilities to sense heat, cold, smell, pressure, and position are vital for perceiving and reacting to our surroundings. Understanding how this physiology works in a particularly critical situation and in human physiological challenges actually which we come across, it is very important, and also in the context of corona pandemic in the last 2 years, and understanding the human physiology challenges. In 2021, Nobel Prize was also given for this particular one. So it was a very important area of science for humans, human health science, and it is one of the very important area for the people. And it is an important, particularly in COVID episode, where smell and taste biology has also been very complex, and it is also been a very important, prominent place in COVID pandemic situation. And understanding the human physiology from cell to molecular biology is also very essential to resolve many human diseases. This physiology is very, very important, particularly to like neurodegenerative disorders, neuro diseases, and several other challenging issues, targets which are not clear. Many diseases, problems, and their targets are not clear. the drug discovery is a very important area for these particular diseases, and the management of the disease is also very important. And it is a big issue, and physiology plays a very crucial role in particularly in addressing all these changes. And it is a multidisciplinary science, physiology, and without physiology, nothing can be worked out, and no disease can be addressed, and disease targets can be addressed, and it is one of the important area of science, and particularly in the angle of medicine and various other human health issues. So this subject of physiology has particularly the 5 national associations which has been together organizing. It is a wonderful event that particularly for young researchers and young scientists and young faculty members to understand what are the developments of the science in the recent times. And this can open a new directions for the young scientists to investigate what kind of research they have to carry out, and what kind of objectives they have to set up. And this will be very important event platform for the young scientists. And the physiological association are very, very prominent role they are playing in this particular area of research, and they are very ideal societies, and they are playing the Fips, one of the important societies, a congregation of societies playing very crucial role in exchanging ideas and propagating the knowledge, and motivating the young scientists, particularly in developing collaborative research from the in between the various institutes. So particularly in developing drug discovery and a novel way how to new discoveries. So novel ways how we can discover new drugs and also new targets for the control of various diseases. It is also very important. I wholeheartedly congratulate Madam, the organizing chairperson, Dr. Shishubala Singh, who is a wonderful administrator, and ever since she was working in DRDO, and I've been knowing her and how dynamic she is, and she's leading the entire team of NIBR Hyderabad, and organizing this wonderful event, international symposium, in a very efficient manner. And I congratulate all the members of the NIBR, and also the presidents, secretaries of all the collaborating societies, and participants especially, and speakers, and very senior person, Salomati Garu, for its grand success of Fips 2022. I especially thank Madam and other people for giving me an opportunity to say a few words on this occasion in the valedictory function. Thank you very much.
For a very nice message, and emphasizing the role of physiology in pharmaceutical sciences, and also in all the areas of science. I think physiology plays a very important role. If we understand the normal physiology only, then we can appreciate the abnormal physiology or the pathology. Really great, sir. I'm sure all the young participants who made oral presentation must be waiting for this moment where we will be announcing the awards. In this categories, we had about more than 200 presentation in 3 categories. There were 3 categories: student oral presentations, faculty oral presentation, and young scientist colloquium. And in each category, we had a large number of participants. The jury was drawn from NIPER and also from other institutes, and they were there online to listen to the presentation, and they made an independent evaluation. And after the evaluation, first I am going to talk about student oral presentation. In this presentation, we have people who are doing PhD. So all the participants were there who were in the PhD program. And we had a very good response, and sometimes it was very difficult to say who is the best. To be very frank, if you ask me personally, all are the best. Only thing is, just just from the point of view that we have already said that first, second, third. So I will just name the winners in student oral presentation. out of first, second, third number for in oral presentation for in a student category goes to Sanap Sachin Nasik. He is from Pharmaceutical and Pharmacokinetics Division of CSIR CDRI Lucknow. Sanap Sachin Nasik. I hope if he is there, he may be listening to us. Otherwise, we will be sending you your e-certificate along with some award to you on by mail. Then in second category, we have two very good presenter, and there was a good strong tie. So we want to announce the result, and both of them are second. Shiv Kumar Samita. He's from Department of Pharmacology. It is Kishori Tai Bohar College of Pharmacy in Nagpur. And the second award in the same category at second position is Vishnu Kant. He is from Pharmacoinformatics of Naipur, Hajipur, one of our Naipur which is located in Naipur. Congratulations to all. However, the first, again, we have a tie on the first. We have Ashish Kumar Srivastav, who is in Immunomodulation Division of Defense Institute of Physiology and Allied Sciences, DPAS, Delhi. And with him, there is another in the same category and holding by rank number first position is to Anika Sood, who is from Department of Pharmacology, Toxicology, Naipur, Hyderabad. So congratulations to all the winners in student oral presentation. Now we move on to faculty oral presentation. These faculty were young faculty, and they made a presentation, and again we had a jury. So in this, the third position in the young faculty is to Dr. Devrata Mandal. He is from Biotechnology Division of Naipur, Hajipur. Congratulations, Dr. Mandal. Second is to Dr. Arjita Banerjee. She is from Department of Physiology, Dr. B.C. Roy, Multi-Specialty Medical Research Center, IIT Kharagpur. And the first goes to Dr. Rahul Kumar, who is from Department of Pharmacology and Toxicology, Naipur, Hyderabad. Congratulations. Hearty congratulations to all the three winners. Thank you for participating. The third category was Young Scientist Colloquium. Again, we had large number of presentations in this. It went almost for 8 to 12 hours every day, 12 hours. So 2 days, it was almost 12 hours, and for day one, we ended this presentation by 10:30 in the night. So in the Young Scientist Colloquium, we had third rank was to Akhil Nair, who is from...
It is second, madam.
Sorry, what I have is slightly different. Yeah, so third is to Prakash Kumar, who is from Department of Biotechnology, Naipur, Hajipur. Congratulations. Second is to Akhil Nair, who is from Department of Pharmaceutics, Manipal College of Pharmaceutical Sciences. And the first one is to Valencia Fernandez. She is a PhD student in our, she is a postdoctorate fellow with Department of Pharmacology or Toxicology, Naipur, Hyderabad. So congratulations to all three. Great job done. We are really proud to announce these awards and look forward, for their future. I'm sure this award will make them more motivated, and they will be the future leaders in the science which they are pursuing. So we will like to see them as a leader in next 5 to 10 years, and they will be, I'm sure, will be taking much more better responsibility in pursuing science for helping mankind at large. So with this, I would request Dr. Dharmender Khantri, who is the main person who has been organizing this conference. As I said, I am the last person. I did the least. Otherwise, it was all the program was organized by Dr. Dharmender Khantri, Dr. Madan, and a young team of students. They were there for 24/7. I want to really compliment, congratulate them, and it is their day. And I'm sure, please celebrate after the valedictory session so that you will know that you worked very hard. And we really look forward for such kind of participation even in the future. Now I will request Dr. Dharmendra Khatri to propose vote of
Dr. Sashi, can I just make 2 minutes? I want to thank you and appreciate you. Can I just have 2 minutes?
Yes, sir, please.
Yeah, thank you so much. Dr. Shashi Bala Singh, the president of Fips, and also Dr. Rao, K.R. Sambasiva Rao, the chief guest of the valedictory function, and all the office bearers of the Fips, very distinguished participants of these 3-day events of the Federation of Indian Physiological Societies. My dear students, first of all, compliments to Dr. Shashi for wonderfully organizing this conference. And it was such a short notice. The whole international conference, I can tell you, it was organized in 3 weeks' time. And bringing all eminent people from across, even less than 3 weeks. And she worked very hard along with the team, Dr. Khatri, Dr. Madan, and all other team. So compliments to you, Shashi. Let's give a big hand to Dr. Shashi Balasa. Wonderful. A great effort. And I have been following all the deliberations. And every expert in the field of physiology came in, from all the human physiology, medical physiologists, molecular biologists, whichever, she has picked the best people, best brain from across the globe. So my compliments to you. And as president of Fips, she has done a remarkable job. Compliments to you. Dr. Sambasiva Rao address was fantastic because he is a doer. And he has done it. And in the Mizoram University, he is giving a new paradigm there. And as a chief guest of this valedictory function, he has given a wonderful message to all of us. The importance of the federation as well as the importance of physiology. He brought those two issues very, very aptly and also very lucidly. And I would like to build on that. Why federation came in, because I was involved right from the beginning when the Fips was formed along with Dr. Manchanda. S.K. Manchanda was the main architect of this. And I was fortunate to be associated with him. So the whole purpose of this, physiology is the basis of human life, animal life, and even a small life like invertebrates. So there are many societies in India, 4 or 5 societies separately. How do we bring synergy? How do we integrate them together as a single force? So that was the purpose. And now I would like to give a clarion call to everyone as the past president of Fips, to everyone that let us revive this. Synergy is strength. Unity is strength. So if we unite together, that will be a force multiplier in all our endeavors. So I would like to particularly tell young physiologists, young biologists, that the first thing is you should understand that the physiology is the basis of human life. So, and as well as the biosphere itself, whether animal, plants, everything is physiology. So that is why even the Nobel Prize is given to physiology and medicine. It is physiology and medicine. So, and even if you are doing a immunology, molecular biology, genetics, everything is physiology. It came from the parent is integrative physiology. So always try to understand, even if you are working at molecular level, cellular level, and assess and take it to the man, molecule to man. So this is what is integrative physiology. This is what is Fips. So I would again, let us increase the membership from all the societies. And I'm sure more activities, more integrated research programs, workshops, I would like to see from all these Fips. I'm sure that you have already started thinking about all this. So to conclude, I would say that let us make a final recommendation. What have we learned? Because ultimately, every time 9th conference is held, what are the salient takeaways from these 3-day deliberations? Circulate to all the constituent members of our Fips so that everybody will come to know. All members should come to know, not just only the office bearers. So it should go percolate down to each and every member of this federation and the constituent societies. And also look at the way ahead. based on what we have learned from this 3-day very important deliberations, where do we go from here? When we meet next time, what is that we want to do? So strategies. So I would request Shashi and the whole team, Dr. Khatri, Dr. Madan, and the constituent societies, where whomsoever was the chairperson, and let us make a gist of it, and then the small committee of these group of all the societies can meet together and look at what are the major recommendations, implementable recommendations, which emerge out of it, which can be circulated. Maybe it should not be more than 2-3 pages, which everyone will read. So this will benefit everyone who may not have even attended. So I would request Shashi to kindly do that, so that this will even go to, let's say, PMO, it'll go to ICMR, it'll go to Agriculture, Ministry of Agriculture, the Animal Husbandry part, Animal Physiology. So kindly churn out from the deliberations some of the specific recommendations. Then it becomes meaningful. Otherwise, just another proceedings will come, and then if Shashi, if you kindly can do it, you are a great leader, and I am sure you will do. And then we can send it to various places. Niti Ayog will send, Ministry of HRD we will send. So I would request with that, once again, compliments to you and congratulations to all the awardees who have got brilliant young scientists, posters, oral presentation, young scientists. My compliments to you. You are the future leaders. So understand that you are going to be leader not only in India, but across the globe. So your responsibility to emerge as a leader in science, technology, physiology, biology, medicine is going to be much higher. So understand that responsibility on your shoulder, and best wishes, blessings to all of you, and congratulations to all of you. And thank you, Shashi, once again for this opportunity to share some thoughts. Dr. Khatri, Dr. Madan, thank you very much for this wonderful organization.
Over to Professor.
Good evening to the Honorable Chief Guest, Professor K.R.S. Subbarao, Vice Chancellor of Mizoram University, of the today's valedictory function of Fips 2022, the organizing chairperson, the Fips committee, professors, scientists, teachers, and my dear friends. First of all, I would like to thank the driving force, the brain behind the 9th Fips, Dr. Sasibala Singh, the organizing chairperson, and you gave me the opportunity to be the organizing secretary of this conference. And it's a matter of honor for me to extend a vote of thanks to all those who are present today for the valedictory functions of 9th FIPS International E-Conference. The conference went for 3 days from 25th to 27 March 2022. We had more than 350 participants, include 280 attendees, 73 speakers and chairpersons from the India and globe. We had 5 keynote lectures, 3 plenary lectures, 6 symposium with more than 30 presentations from the expert speaker around the world. We had 150 presentations in 3 different categories, 73 student oral presentations, 41 faculty oral presentations, 33 young scientists oral presentations. I, on behalf of NIPER Hyderabad and entire supporting sponsoring organizations, team, speakers, chairperson, participants, and presenter, let me call it as fraternity of physiological scientists here together, and on my own behalf extend a very hearty vote of thanks to all the speakers for gracing your important work and sharing with us findings and opinions during 9th Fips conference. Ladies and gentlemen, an event like this cannot happen overnight. It requires planning and a bird eye for the details. It is just because of the energy, none other than the madam who has worked hard day and night to make this event successful. Her macro and micro management making this 9th Fips grand success is highly impressive, and it gives me a lot of learning and honing my management and administrative skill as well. We have been fortunate enough to be backed by a team of very motivated and dedicated colleagues of NIPER Hyderabad who gave their valuable time for evaluation of all our oral presentations. I would like to extend my gratitude to all the volunteer students, structures, and tech support team of NIPER Hyderabad who work hard in setting this up for the arrangement of live streaming. Thanks to all the head of the departments of NIPER Hyderabad for their support in a smooth and seamless conference, starting from the JPN Misra, the registrar of NIPER Hyderabad, Dr. Nanduri, Dr. Saurav, Dr. Amit Asthana, Dr. Neeraj Kumar, Dr. N. Sankriya, Dr. Chandriya, Dr. Vasundhara, Dr. Lakshmi, and Dr. Ganadhan. A special mention to my team. Without them, the event couldn't have been possible. They all have worked hard by staying back the late nights. Dr. Rahul, Preeti, Anika, Valencia, Ankita, Savia, Nusrat, Gurpreet, Pushpa, and Anuradha. They have been the team because I'm not complete. I alone is not a organizing secretary. They all are the organizing secretary of this conference. I would like to thank all the associate society of PSI, SAPI, APPI, SAP, and SCAP and their office bearer for their supporting in making this event successful. I would like to also thank the advisory committee, Dr. Rajiv Vasne, Dr. Salamurthy, Dr. Kushal Das, Dr. Rajendra, Dr. KK Deepak, Usha Panjwani ma'am, Dr. Gopal Krishnapal, Dr. Ramad Chandra, SK Reddy, Dr. SK Reddy, Dr. Ajay Kedang, Dr. KP Mishra, Dr. Madhuri, Dr. Latika Mohan, Dr. MS Paul, Dr. Sunil K. Hota. And this is with, I would like to thank everyone who has joined today for the validity function, and Jai Hind. Thank you very much.
Thank you. Namaskar.
Thank you, Shashi. Congratulations. Thank you.
Thank you so much for all the support from all of you. And it is because of your support we could do it. Thank you so much. Namaskar.
Thank you, madam. Thank you very
Jai
much.
Hind.
Thank you, one and all. Thank you, Team Fips and Organizing Secretary and President, Madam, for such a nice conference.
Thank you, Usha. Thank you, Usha.
Thank you very much, Madam, and all your team. I am Dr. Kushal Das. Thank you very much.
Thank you for
And
helping us
congratulations.
in making the program. Thank you.
Thank you, ma'am.
Congratulations, madam.
Thank you, Anita. Thank you. So kind of you to be here. Thank you.
Thank you.